Slide show पे जाना चाहिए. Resume slideshow. And नहीं उधर वो कुछ और आ गया. So set करें. And okay. यहाँ पे ठीक है. All right. And this biochemistry, you know, is a central scientific subject in which in biological sciences. So students from different departments, they take this biochemistry course as minor, compulsory minor, and we have about 5,000 students per semester, which take courses of biochemistry as elective minor or different type of courses. So we have now quite a good number of research labs, thanks to the university administration and different research projects. We have established some research labs, and we have also an old building of biochemistry from which our most of the alumni aware of, and we also have a now new building at the postgraduate biochemistry block. R&D, I'm just give you an overview of different labs which are working there. Like they have molecular biochemistry lab, natural product biochemistry lab. We have health biotechnology. We have clinical molecular biochemistry. We have plant genome editing lab in CAS. We have bioactive molecules research lab, cellular biochemistry lab, and we are recently we are developing this with Dr. Ali and his team, mammalian molecular genetics lab. That is also we are establishing in CAS. We have different ongoing and recently completed research projects, which our faculty is quite interactive in that, getting these projects. And there are always, we try to get projects. Of course, without the funding, you cannot do anything. Economics is the main key factor for all such types of activities. We hold a number of scientific events and different scientific activities. In the past few years, we had 3 international conferences with more than 1,200 participants and a number of foreign delegates. And then we have symposia like this one, national, international, and seminars. And we have workshops on practical trainings. And we are also quite famous for our hands-on trainings, because we take very few number of participants in the hands-on training so that we can deliver real hands-on training to them, so they can and there are people who are like going for postdocs, for our students for PhD. These hands-on trainings are very good for them, so they get good training out of it. And we have a number of students' activities like study tours, sports gala, annual dinners, special day celebrations, and especially this CESCOP. We have one session today after 3 talks on our alumni. That was a CESCOP that was initiated a quarter century ago when Professor Mukhtar was here as a young faculty, as a lecturer. He was here in this unit in biochemistry department. So we initiated this scientific, educational, and social council of biochemists. So it kept on working, went some hibernation, and then it emerged out again during the last couple of years. But I will go more when in our alumni session. So these are just glimpses of our student activities. We are up for the quality education. Is our vision of our Honorable Vice Chancellor and our Dean Faculty of Sciences, we have revised our curricula based on the OPE concept, and we also have introduced skill-oriented courses for our students, graduates, so that they can be aware of, so they can be working with in practical situation. And we have institution of advanced research facilities, as I already mentioned, that we are in research labs, we are trying to maintain, and development of international collaboration. This symposium, we are thankful to our guests from Poland. We are going to sign an MOU with this university in the Poland, and also they are offering us like internships and scholarships, and we will have collaborative projects. And we are going to have alumni fund, alumni advisory board. Today we are going to announce the first alumni advisory board in our alumni meet after the 3 lectures. And then a long standing problem of recognition of biochemistry from PMDC, that is also on our agenda. So alumni board, along with the members, I think they can work more on it. This symposium already, you know, is along with the UF Biochemist alumni meet. This is, we planned it for 2 days activities. So you already have the schedule. First day, the technical sessions, you have already like, we have different like keynote speakers from different parts of different institutions within country and abroad. And this technical session 3, that was also a big part of it was included in, it was physical and hybrid type of thing, but this time we got a scarcity of funding. 2 of our speakers, they were ready to come to Pakistan, one from UK, one from China, but the economic situation didn't allow us we could offer but we are very thankful to Endowment Fund Secretariat, who has allowed and delivered the funding for our honorable guest from Poland, Professor Bhatti, who is also Dean Faculty of Sciences, Executive Director of the Endowment Fund Secretariat. So they very kindly have provided us funding, so we could have him there. The second day, it will be now webinar. We converted into webinar, so we have speaker from US. He is also, Dr. Atarata is professor and chairman of one of the departments, and he is also a very big name now in natural product chemistry. So his lecture will be online. And then we have Hoi Zhuang. They are also looking for a signing up of an MOU with us. And Dr. Azhar Hussain from UK, from Zahiruddin Babar. He is from UK. So he will be also delivering a talk on. And we also have a poster session, and we have about 50 posters which are available. You can visit around, and there will be a poster evaluation, and there is a committee that will decide the first, second, third prizes on the posters as well. And for the social evening, you know, today is the Qawwali night, and many of our alumni, they requested me, they informed me that it would not be possible to join us in the morning, but many of them will be coming in the Qawwali night at least. So, and for our Honorable Patron-in-Chief, Professor Dr. Khalid M. Khan, he will be here anytime. He will be joining us in this morning session, and then he will also be with us. So hope you will enjoy this. So I urge everyone to it will commence at around 6:30 p.m. We are thankful to Endowment Fund Secretariat once again. It is our only funding agency, but it is for this symposium, but we are highly thankful. And I'm also thankful to the participants for their timely registration and all things that made it possible. I'm thankful to our UF administration for allowing us to hold such kind of activities. I'm thankful to all the keynote speakers who have come all the way from different institutions and from abroad. And I'm also thankful to online. There are how many number? There are about many number are online participants from different. There are participants from China, from different parts, and there are more than 70 participants. They have joined online this session, today's session as well. We are sharing the screens and all the activities going on there. And our honorable keynote speakers from different countries, we are also thankful to them for giving their time. And I am thankful to whole of my team in the Department of Biochemistry, that without this team, it is not possible. So I won't take any of the name here because everyone is performing tireless job for the last many, you can say, couple of months. And also our laboratory and office staff, who is also very active under the you can say Mr. Tariq and his team is working very hard with us, like 24 hours type of thing. And also, and our enthusiastic youngsters, the CESCOP teams, which are very enthusiastically, they have been working to make this possible. And I am thankful to all of our guests from NIAB. Dr. Amjad is here, Dr. Sitan. He has joined back from US in BZU and he's here today, so you will have interaction with him as well. Dr. Abda, Dr. Saima, you will have a lecture talk also of the Dr. Saima. And we are thankful to one of my older colleague, you can say, or he has been my session fellow, Dr. Nadeem Akhtar. You will also be listening to him. And we are thankful to Dr. Hira Ijaz from Haripur. And we are also thankful to Dr. Muhammad Asghar. He is from NAMS. And we are very thankful. We were looking forward to a very informative talk from him so we can move further in this arena. And Professor Dr. Mushtaq Ahmed from Quaid-e-Azam University, we are highly thankful to him as well to deliver today's talk. And all the guests, all the participants, all the students, we are thankful. And in the end, once again, I am highly thankful to Professor Mukhtar, who is always here whenever we request him to join, and Dr. Javed Iqbal. He is the pioneer of, one of the pioneers. His logo of CESCOP was designed by Dr. Javed Iqbal. So we are thankful to him from ComSets and a group of students from ComSets. So with these few words, see you around with an hoping very interactive session. Thank you very much.
Ladies and gentlemen, this was Professor Ahmed Jamil, the chairman of the biochemistry department. And while he was addressing to you, our principal of Paras campus and a very humble professor of our department, Professor Dr. Anjum Zia, joined us. Please give a big round of applause for him. And now I would like to call here on the rostrum our the Dean, Faculty of Sciences, Professor Dr. Ijaz Ahmed Bhatti. He's paying respect to our ex-chairman. Please give a big round of applause. Dr. Tahir Abad.
Yes, to our alumni, one announcement and good news that one of our colleagues has become professor of biochemistry now, Professor Dr. Anjum Zia. So you already know it, but he's also principal Paras. So all 3 semesters of biochemistry, he's looking after those as a principal, not only as a principal, but as a biochemist as well. So we welcome him as well.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. Honorable Dr. Professor Dr. Muhtaram, the Vice Chancellor, ex-Vice Chancellor Islamia University, and currently is also Vice Chancellor serving in Islamabad. Professor Dr. Tahra Iqbal, ex-Chairman and ex-Dean, Faculty of Sciences. Professor Dr. Amar Jamil, Chairman Biochemistry. Professor Dr. Hakmal Bhatti, Professor Dr. Anjum Zia, my colleagues and dear students who are the students, the real beauty and asset of our institution, our university. So very good morning to all. So I'm really very happy to see this morning Dr. Saptan, who spent a lot of time in US and the land attracted him back. Now he's serving in Pakistan in BZU as associate professor. So I'm really impressed. The pupils will learn, go abroad, learn a lot, and then come back to serve the community. So it is really act. We can salute this. So I'm very happy to see Dr. Javed from ComSet and the colleagues, the national delegates and as well as international delegates from Poland. And I'm really grateful to these people who traveled a lot to join this uh, special event organized by the Department of Biochemistry. So as Professor Amarjavil mentioned, biochemistry is the central science actually addressing the all kinds of reactions, activities, excitement, which are present in nature in terms of animals and plants. All kinds of living activities is being governed through the biochemistry. So biochemists are the real interpreter of the universe. What is going on in the plants and animals? So unfortunately, I remain always deficit to understand the biochemistry because I'm physical chemist. So I did not get any opportunity in my life to learn properly by sitting in the class to learn the biochemistry. But due to the close interaction with the biochemistry, chemistry and biochemistry departments were together, they remain up to 2010. Chemistry and biochemistry. So through my colleagues and interaction, so I am still learning. Biochemistry is really very fascinating subject who can give true idea about the study of nature. So now I will give you the special thanks to the Department of Biochemistry for organizing such a symposium, national and international conferences, workshops, and skill development courses for the community, for the faculty, and for the faculty at large to give special training to the other universities and institutions as well. So I will give you a very fundamental, my opinion about the 2 day seminar, drug delivery and development. So I have taken the word that drug. The word the drug has been derived from the French word rouge. That means dry herb. So conventionally, in the primitive ages, unconventionally, the treatment was being done through the plants, through the animal products, through the minerals. But even in that ages, the preference was given to the plant origin substances. They were always given the preference. So the first modern pharmaceutical and the potent compound was isolated by the German chemist from the opium plant, which was morphine, which was first labeled compound, which was used as a drug or a medicine to treat many kinds of diseases. So in the actually the drug delivery and its development is a multidisciplinary task. So the chemists, biochemists, pharmacologists, pharmacists, biotechnologists. So all are striving their hard to identify and extract and elucidate such type of compounds which can be used as medicine. It is a multidisciplinary task. So everyone is working hard to get and impart specific labor in the identification and then it development and sending it to the markets. So drug delivery and development in this process, first of all, the compound has been identified in any source. Mostly the people try to get from the plant sources. So after identification, the preclinical trials were performed, and then after that, post material and clinical trials were identified, and then they were performed, and finally the drug was marketed after the some regulatory processes. So these are the fundamental processes through which the drug is identified, discovered, and then it was developed and marketed. So in this age, many computational experts, one group is working in the ComSat Islamabad, they are using theoretical approaches for the drug identification, their delivery, and hypothetical computational approach for that. And they are producing a lot of publication. But after that, even single drug has not been marketed in Pakistan using that computational tool because it is a much more, I think, sensitive and much more significant to have a practical approach rather than the theoretical one. So in the process of drug delivery, being a chemist, we feel that any drug which has a molecular mass less than 300 could be served as a drug. So if its molecular mass will go up to 300, it will not be so successful or significant that material which can be used as a drug. So this is the first prerequisite for any material to be used as a drug. And the number of hydrogen bond donor and acceptor must be less than 3. So, and the hydrophobicity and its hydrophobic and hydrophilic character is also evaluated through C log P value that must also be less than 3, and the rotatable bond must be less than 3. If such any material which will have this type of characteristics, that can be further used to trial for that. After discovery, they can be used for clinical and preclinical trial to come as a drug. So in the end, I will give you the idea that UF actually has opened multidisciplinary research opportunities for the faculties and the students. There are centers available biotechnology center, food security center, nutrition centers. These are the center which has been established for the horizontal growth of research, and multidisciplinary research opportunities are available. So there is a lot of opportunity for the students and faculties to go for the multidisciplinary research and for the international collaboration. So this symposium will surely open the opportunity of collaboration with Poland, with the other institution, ComSat, Kaiser University, and the other very prestigious institution of our land. So this type of conferences and symposia will actually have a collaboration opportunities of the students and the faculty, teachers with the other institutions as well. So this is the need of the Pakistan to develop the socio-economic status through such type of practical activities. So at the end, I will thank to Department of Biochemistry for organizing such a beautiful and very productive and very applicable forum for the researchers, students, and the faculties. So I again, at the end, I will thank to Professor Dr. Mukhtar Ahmed. He served the Islamia University Bahawalpur in the way, and he will give a new name of the university. He actually gave the paradigm shift, and he realized the community that universities build the economies and universities are for the societies and communities. So I appreciate and acknowledge his task he performed in the university for its revamping. Thank you so much.
Thank you, Mr. Ijaz Ahmed Bhatti. And now I would like to call Professor Dr. Muhammad Mukhtar, the Vice Chancellor of the National Skills University, Islamabad, to please, sir, please come here and serve the shields, present the shields to our worthy guests. I would like to call upon stage Dr. Hira Ijaz. Dr. Hira Ijaz. Okay. Dr. Saima Siddiqui. Please, ma'am, please come up on stage. Students, please give a big round of applause. Dr. Saima Siddiqui.
Amal, you have to come up on stage. जी आपने आना नमल कर लो
This is Dr. Saima Siddiqui. Next to her is Professor Dr. Muhammad Nadeem Akhtar. Sir, Dr. Mukhtar wants to say something.
I think we have to have a big clap and appreciation for Dr. Saima Siddiqui because she has been recently promoted. On top of this, we be proud that there is some specific syndrome, जैसे हम Krebs cycle को पढ़ते हैं, that syndrome is in her name, Siddiqui syndrome. So Pakistan should be really proud for that. Thank you.
And now the shield is being presented to the Professor Dr. Nadeem Akhtar from Ghazi University, Dera Ghazi Khan. Please, ma'am, please join us.
कुछ लोग बड़े खुश होते हैं
सेशन को कैंसिल नहीं आना इवनिंग
Now I would like to call Professor Dr. Mushtaq Ahmed, Tamga Imtiaz, Qaid-e-Azam University, Islamabad. So we see you have a huge fan following sitting over there, chanting for you. And now it's Dr. Muhammad Asghar, National University of Medical Sciences, Rawalpindi. He's working at the importance of bioequivalence in generic drugs, ensuring safety and efficacy. Dr. Muhammad Asghar. And now it's time to present the shields to our worthy foreign guest. I would like to call Professor Dr. Lukas Fizolkowski to come up on stage, sir.
Oh my God, I was looking for you. I thought
He's from Nicholas Copernicus University in Torun, Poland. And now it's time to present the shield to Professor Dr. Alicja Nowacek from the Nicholas Copernicus University in Poland. Dr. Alicja. मैं पूछता हूँ ये अभी अभी जैसा टॉक है या सेशन है टेक्निकल now I would like to call Professor Dr. Mohammad Mukhtar, the Vice Chancellor of the National Skills University Islamabad, to come over here and share a few words with us. So please come here. We welcome you. Please, another round of applause.
Bismillah ar-Rahman ar-Rahim. It's a real great morning for me that we have our international colleagues as well as a lot more dignitaries. Of course, Dean Saab, who spoke so eloquently about drug development. I think you know better biochemistry than a lot more others, you know. I mean, you know, he was confessing that has a problem with the biochemistry. But anyway, I mean, I'm also one of the biochemists. And by the way, I'm so glad to see even I was a lecturer, I used to sit over there on the backside because that's the place where we usually get the place. I think Dr. Abda will confirm this one. I don't know in Harira how was the things. So I'm glad to see that biochemistry is evolving at least at University of Agriculture, Faisalabad. Because when I was a student, we were 5 total in the class in MSc Biochemistry, and among them, the relative ratio of girls, there was 1 girl and 4 boys. And last night I talked to Dr. Ahmed Jamil, and he mentioned there is total change. So I mean, it has immensely evolved. So there are 80% girls and it has reversed. I mean, girls जिनको मैं अक्सर जो है ना, I mean, I basically floated the idea कि they should not be called as a girl, उनको बेटियां कहा जाए. That sounds much better जो है ना. So that goes. और एक चीज जो कि, I mean, I know you have evolved, but I just want to give you a advice. That advice is like, I think that one, our colleague, because we five, one is sitting in California, Dr. Javed, the other one is sitting in UK, Dr. Kaiser, Dr. Jamshed के बारे में मुझे पता चला कि वो इस दुनिया से चले गए जो है ना. He was our colleague also. And I am here. Kullu nafsin za'iqatul maut. Everybody has to go. With due apology, something I have to. This is not a technical speech. Don't you worry at all. You won't miss anything. This is. So anyway, we were having just one student, and I think Dr. Saima and Dr. Abda will, will echo with me. But that one was more than four. Like her voice was so powerful. She is so strong. Everybody know about this one. I just want to share with you. I think couple of months before we had a dinner. तो उस डिनर में हमारा ऑल बायोकेमिस्ट गेट टुगेदर इन इस्लामाबाद. तो उस डिनर में खुशकिस्मती से वो मेरी क्लास फेलो आ गई जो है ना. So there were like I think 5 or 7 other ladies sitting over there, but she did not give a chance not to talk to anybody else. She was dominating all around. So you would have to work very hard. Anyway, I mean, I know this conference is very timely. आमिर साहब को हम जो है ना बहुत ही मासूम और बहुत ही भोला भाला और ऐसे जो है ना समझा करते थे जो है ना. लेकिन ये जिस इनमें जो कुछ भी आया है ना, उसके पीछे जो कैटलिस्ट है वो जावेद साहब हैं. ये सिटिंग ओवर हियर, यू नो, he has been struggling such a good things. So Doctor of Chemistry is working over here. Don't you worry at all. You know, we are doing like, I mean, we did not use enzyme. We use catalyst over here, you know. So all that is moving in a great way. By the way, I mean, I just want to share with you कि मैंने दुनिया के बहुत सारे ममालिक में पढ़ाया है. जैसे अभी मैं बात कर रहा था, ये हमारे डॉक्टर साहब तशरीफ रखते हैं. ये प्रोफेसर साहब मलेशिया में होते हैं. तो मैं अभी इनको उधर बैठे हुए बता रहा था जो है ना कि इनकी मैडम जो है ना, जिनको मैंने तकलीफ भी दी जो है ना, तो मलेशिया में अगर आप जाएं तो आपको ओनली चावल ही मिलते हैं खाने के लिए, बॉइल्ड राइस, दैट्स इट, यू कैन ईट. सो आई मीन ए पर्सन लाइक मी कि जो कि खाने की पीने के बगैर जिंदा नहीं रह सकता, इफ ही वुड हैव नॉट बीन देयर एंड मैडम ना होती, क्योंकि मैडम सुबह सुबह बल्कि पूरे रमजान में, अभी रमजान भी आने वाला है, अफ्तारी और सहरी का अरेंज किया करती थी. सो दीज आर द टाइप्स ऑफ लाइक आई मीन links that we keep enjoying, we keep moving, and I have millions of stories. मुझे टाइम बता दें मेरा कितना है आमिर साहब, मुझे बता दीजिएगा जब मैंने खत्म करना है. 2 मिनट, 3 मिनट, 4 मिनट, हाउ मच टाइम डू आई हैव इट? यार ये जाती है 5-7 मिनट तो मुझे दे जो है. So you know, I am good in telling stories. I mean, and I'm proud of this one. ऐसे हुआ कि इसलिए मैं आपको कुछ स्टोरीज ही बता दूंगा. वैसे साइंस और टेक्नोलॉजी के बारे में मुझे बहुत सारे लोगों ने बताया कि आप बड़ी अच्छी कहानियां बता लेते हैं जो है ना, यू स्टार्ट ए चैनल, यूट्यूब चैनल. तो मैंने कहा यार दिस इज नॉट गुड, सो इफ आई डू, आई विल डू समथिंग साइंटिफिक. सो देयर इज ए स्मॉल थिंग, बायोमेड ग्लोबल जो है ना, अगर आप देखें जो है ना, एक ए स्मॉल चैनल दैट आई रन जो है ना, बायोमेड ग्लोबल. I mean, over there, I read every day what is new science and try to make it simplified way. Now, the people over here, there are a lot more biochemists sitting over here, not the faculty. What was the great invention of year 2023? Anybody can tell me? So this is the world of information. And we have to get involved with this one. We be proud that was from the biochemistry and biotechnology. That is the treatment for hemophilia, like a boy or a girl, because I have seen the people dying in front of me because I used to work in a medical university. Now there is a treatment exists for that one. I'm sorry, don't you worry. I mean, if I ask you something, that was a tough question. मुझे इस वक्त मुनीर साहब याद आ रहे हैं, अल्लाह मियां उनको जन्नत नसीब करे. He is the one basically biochemist जो है ना, उनको उन्होंने जान दी. और ये सारी जो बेटियां इधर बैठी हैं जो है ना, they should be thankful to मुनीर साहब कि because he was so much welcoming, bringing the girls in biochemistry. मुझे I mean, for everything I have a new story. तो ये था कि मैं और मेरी वाइफ उस वक्त हमारी शादी नहीं हुई थी और मुनीर साहब का ऑफिस तीनों का इकट्ठा था. तो एक दिन जो है ना, 3 बड़ी वेल ड्रेस बेटियां आई. तो आई, उन्होंने देखा, उन्होंने कहा मुनीर साहब की हाल जे, अल्लाह मियां उनको जन्नत नसीब करे. आई मीन इट्स नॉट समथिंग जो कहते जी ठीक जे, वो सर को मिठाई वगैरह तो खवा दे जड़ा ना. अच्छा, मुनीर साहब का एक वो मुलाजिम होता था, मेरे ख्याल में आप में से किसी को नाम याद है या नहीं है जो है ना, तो जा यार तो मिठाई लेके आ. क्या था मैडम आपका उसका, भूल गई, देखे ना जी उसका जो है ना. बट एनीवे वो गया, मिठाई लेके आया. मैंने भी खाई, जाहिदा ने भी खाई और मुनीर साहब ने, और तीनों बच्चियों ने बड़ी मजे से. तो बाकी जो बची हुई थी जो है ना, तो उन्होंने कहा सर हम ये ले लें थोड़ी थोड़ी मिठाई. ने कहा ले लें जो है ना. वो बच्चियां चली गई तो हमने मुनीर साहब से पूछा कि मुनीर साहब ਤੁਹਾਡੇ ਜਾਣ वाले ने. मुनीर साहब कहते हैं नहीं यार मेरे तो जाने वाले नहीं जड़े ना. तो मैंने पूछा जायदा से, मैंने कहा जायदा ये आपके हॉस्टल की बच्चियां हैं? कहते नहीं मुझे तो पता नहीं है. तो मेरे तो जानने वाले वैसे ही नहीं थे क्योंकि वी वर द मोस्ट डिसलाइक पीपल जो है ना उस वक्त. ऐसा इशू होता था. एनीवे हुआ ये कि मुनीर साहब ने उसको भगाया, उसका बच्चियों ने बुलाओ यार. तो उनको बुलाया गया तो उनसे पूछा गया, हाँ भाई आप कहाँ से हैं, कौन सी डिपार्टमेंट में आपका एडमिशन हुआ है? कहते हैं सर हमारा तो उधर वेटनरी में एडमिशन हुआ है, हमें किसी ने बताया है कि मुनीर साहब के पास चले जाओ, जो उन्होंने को मिठाई खानी है तो वो खिला देते हैं. तो हम ये टेस्ट करने आए थे कि क्या ये साइंस वर्क करती है या नहीं करती है. सो दीज आर ब्यूटीफुल साइंसेस. और इसके अलावा जैसा कि डॉक्टर साहब माशाल्लाह भट्टी साहब बड़े अच्छे तरीके से बता रहे थे कि देयर आर लॉट मोर अपॉर्चुनिटीज. वन मोर टेक्नोलॉजी जो कि अभी पिछले हफ्ते बल्कि आई तो काफी अरसे से है, कि आप में से हीमाटोलॉजी का कोर्स बहुत लोग पढ़ते होंगे, आइसोलेशन ऑफ ब्लड और ब्लड से ल्यूकोफोरेसिस जो है ना. नाउ द टेक्नोलॉजी इज, हमारे कॉलीग्स जानते हैं कि कैंसर कैन ओनली बी ट्रीटेड विद CAR T cell. You take the T cells out of person and you transform them in the lab, put them back into the person, and the person will fully recover from cancer, for leukemia, for number of other cancer types. But the problem is the companies cannot monopolize because those T cell transportation is issue. So there was article in Nature also a few days before, like you can only work with that one if you get involved with the things. You develop a simple technology, isolate the T cell, and then you their transfection. I think most of the people know that you transfect the genes and then take those cells and take them. Here we go. So that is a good way. Anyway, I mean, coming back to sir, 3 more minutes, Amir साहब. तो मैं इनको आमिर साहब को बड़ा अप्रिशिएट भी करता हूँ हमेशा जो है ना. आमिर साहब मासूम होते थे, भोले भाले होते थे, बड़े हैंडसम होते थे जो है ना, माशाल्लाह जो है ना. सो आई मीन इट वाज लाइक ए फैसिनेशन स्टोरी जो है ना. हुआ ये कि हमारे बायोकेमिस्ट्री में सुबह सुबह जो है ना क्लासेस, वी वर फेमस के पहला जो है ना पहला पीरियड लगा करेगा, तो फर्स्ट पीरियड विल बी देयर. कुछ लिखा हुआ बेटे? ओह थैंक यू वेरी मच फॉर हेल्पिंग मी. मैंने कहा मैं शायद बूढ़ा हूँ, मुझे पता नहीं चल रहा था. एनीवे सो हमारे इधर वाइस चांसलर उस वक्त होते थे, अल्लाह में उन्हें जन्नत नसीब करे, मियां मुमताज साहब, मेरे ख्याल में वो भी इस दुनिया से चले गए हैं. उनको बताया गया कि यार ये बायोकेमिस्ट्री वाले सारे फड्डा कर देंगे, कोई भी फर्स्ट पीरियड नहीं होता सर जड़ा एवं क्लासा जड़ा ने लाया। उस वक्त आई वॉज ए रिसर्च ऑफिसर था, और मेरे साथ अगर कुछ लोग जानते हैं अब्दुल रऊफ रूफी साहब जो है ना, एक इधर ह्यूमन एनवायरनमेंट कंट्रोल सेल बना था जिसमें रूफी साहब और मैं थे। और उन दिनों मैं एमफिल भी कर रहा था। आई वॉज ट्राइंग टू फिनिश माई डिग्री ओवर देयर। मेरे ख्याल में हमारे उस्ताद मोहतरम तशरीफ लाएंगे, डॉक्टर खालिद साहब जो है ना, जिनने मेरी जान बचाई, नहीं तो आज आई वुड नेवर हैव बीन हियर, यू नो, द प्लेस वेयर आई एम। एनीवे, सुबह सुबह अपना वीसी साहब आ गए, उनके साथ जुबैर सदीकी साहब चले गए इस दुनिया से, हैं जो हैं, ठीक चले गए। इन्ना लिल्लाहि व इन्ना इलैहि राजिऊन, पता नहीं है जो है ना बैड सूरत। एनीवे, तो वो वीसी साहब आए एंड आई वाज मेकिंग माय ब्रेकफास्ट इन माय लैब बिकॉज आई वाज डूइंग एक्सपेरिमेंट। और ब्रेकफास्ट क्या था अपना डॉक्टर साहब जानते हैं कि वो जाके जो है ना बन लेके आना और लस्सी बना लेना, और उसके साथ जो है ना, देहाती लोग हैं हम जो है ना, आई वाज मेकिंग दैट एंड इन द मीनवाइल द वाइस चांसलर एंटर्स। सो आई मीन ही कम्स ओवर देयर, हाँ भाई क्या कर रहे हो? मैंने कहा सर लस्सी बना रहा हूँ। उन्होंने कहा ये रिसर्च है। मैंने कहा सर नहीं, ये रिसर्च नहीं, ये मेरा नाश्ता है। सो एनीवे, तो अब वो जब जाने लगे तो मेरे हम आए में फ्रॉम बहावलपुर, वेरी विलेजर पर्सन, तो हमारे पास कोई आया तो मैंने कहा सर आप लस्सी पियेंगे? कहता नहीं नहीं थैंक यू वेरी मच। ही लुक अराउंड, there was nothing in the class except that जो मेरी वाइफ है जो है ना, वो आ रही थी उस वक्त जो है ना, उस वक्त 7:15 या 20 हो चुके थे, तो things were 20 minutes late. He went back and a bureaucrat guy, I mean, he right away put the things in high alert. So, I mean, these teachers, I mean, just I'm trying to tell these students that how things move, how life moves, and don't get worried at all. Life is with you. Enjoy this one. जितना इसको अच्छा आप कर सकते हैं जो है ना करें, कभी डिप्रेस नहीं होना चाहिए. So what happened? Like the chairman called me, रियाज साहब केमिस्ट्री वाले ठीक ठाक हैं, अल्लाह की उनको सेहत तंदुरुस्ती दे जो है ना, बड़े बड़े ही औखे चेयरमैन होते थे, बड़ा मुश्किल था। अल्लाह करे अब जो है ना डॉक्टर साहब और ये जो है ना अच्छे चेयरमैन हूँ जो है ना, तो उन्होंने मुझे बुलाया। उन्होंने कहा असल में ये टेक्निकलिटीज जो है ना, उन्होंने कहा जी वीसी साहब नाराज किस लिए हो गए जो है ना, उन्होंने छाड़ा क्यों है सबको जो है ना। उन्होंने कहा जी वो जो लड़का जो उधर लस्सी बना रहा था, उसने लड़की लस्सी ऑफर कर दी जो है ना, इस वास्ते वीसी साहब नाराज हो गए हैं, दैट इज द रीजन। तो खैर मुझे उन्हें बुलाया, ने कहा हाँ भाई तुम लस्सियां बनाते रहते हो सारी रात? मैंने कहा नहीं सर हजूर मैं लस्सी नहीं बनाता, रात को जलदाज अपरेटस चल रहा था, उस पर नाइट्रोजन एस्टिमेशन हो रही थी, दैट आई वाज डूइंग। उन्होंने कहा अच्छा ठीक है, आज से तुम्हारी छुट्टी है, जाओ मैंने तुम्हें टर्मिनेट कर दिया। सो आई मीन दैट इज इफ समबडी हियर द वर्ड टर्मिनेशन, जो इधर कलीग बैठे हैं, व्हाट दे विल डू? डॉक्टर साहब आप ही बता दें क्या करेंगे सर, है नहीं, खबर नहीं, परेशान होना है। अच्छा ये मुझे जब टर्मिनेशन के ऑर्डर मिल गए तो मैं उनके कॉरिडोर में, वो पुराना जो कॉरिडोर है उसमें जो है ना मैं चल रहा था, मुझे आज भी याद है कि उधर मैं गुनगुना रहा था बड़े जोर से, और क्योंकि थोड़ा सा टेंशन भी थी जो है ना कि आज पुरानी राहों से कोई मुझे आवाज ना दे, वो जो गाना है वो गुनगुना रहा था। तो इतनी देर में डॉक्टर रियाज बाहर निकले, कहते हैं यार मैंने खुदकशी करनी है या तुम्हें मैंने निकालना है जो है ना, अभी मैंने तुम्हें समझ नहीं है कि मैंने तुम्हें निकाल दिया है इस यूनिवर्सिटी से? तो मैंने कहा सर मुझे समझ तो आ गई है, तो इसलिए तो मैं गा रहा हूं इधर जो है ना। सो दीज वर लाइक आई मीन ए गुड स्टोरी, एंजॉय योर लाइफ। अपनी इस यूनिवर्सिटी को अपना जितना भी अब आप कहेंगे कि मैंने वाइस चांसलर की आपको स्टोरी नहीं बताई, वन लास्ट स्टोरी एंड दैट्स इट। इस्लामिया यूनिवर्सिटी बहावलपुर का वाइस चांसलर था डॉक्टर साहब उधर ही होते थे माशाल्लाह जो है ना, तो हमें जो सबसे बड़ा आया मसला आया वेटनरी वालों का। अब वेटनरी वाले जो है ना बड़े ये स्मार्ट लोग होते हैं। हम जो अपना मैडम के और हमारे जो है ना कंबाइन मेंटर थे डॉक्टर अफजाल साहब जो है ना, अल्लाह में उन्हें जन्नत नसीब करे, बड़े अच्छे इंसान। तो उनकी एक वर्डिंग थी, डॉक्टर अपने बी साहब अभी मैंने कहीं आ रहे हूँ तो अभी मेरी जो है ना छुट्टी करा देंगे एनीवे जो है ना, उन्होंने कहा यार एग्रीकल्चर इज कंबाइंड ऑफ टू वर्ड, एग्री एंड कल्चर, लेकिन इनके साथ नो लगा था कि ये ना तो ये एग्री होते हैं ना ये कल्चर रहते हैं जो है ना। सो दैट्स व्हाट इट इज जो है ना। तो बैचलर जो वेटनरी वाले हैं, आई मीन ऑफ कोर्स हर डिपार्टमेंट की अपनी एक जो ना होती है। तो मैं वाइस चांसलर था, एक दिन मुझे पता चला कि सर वो वेटनरी वाले रोजाना जुलूस निकाला होता था, बच्चियां थी काफी सारी, वाइस चांसलर मुर्दाबाद। अच्छा फिर ये जो है ना इनके एक दो नारे ऐसे होते हैं एनिमल से रेलेवेंट होते हैं कि वाइस चांसलर कुत्ता है जी, ये है वो है, माजरत के साथ आई एम यूजिंग दिस बैड वर्ड जो है ना। सो एनीवे उनको पता नहीं था कि मैंने जो है ना अपना जो है ना, वो सर्दी थी थोड़ी सी, मैंने वो टोपी पहन ली जो है ना, तो मैं भी उनके साथ आ गया। उन्होंने कहा वाइस चांसलर मुर्दाबाद जो है ना, वो जैसे वो कह रहे हैं मैं साथ वाइस चांसलर भी वैसे ही कह रहा है जो है ना। तो वो जो कुछ कह रहे थे एनीवे, तो उनमें से किसी को पता चला, उन्होंने कहा यार वो तो भी साहब आए नहीं जड़ा है ना। तो आई सैट विद देम, आई टोल्ड देम कि बेटा ये जो आपके प्रॉब्लम है ये हमारे ही प्रॉब्लम है, वी हैव टू सॉल्व these issues and Allah has been very kind. This is my 5th university as a vice chancellor. बल्कि अभी क्योंकि ये जो करंट प्रेसिडेंट अभी बैठे हैं मेरे ख्याल में, आई मीन इट्स वेरी काइंड ऑफ हिम, ही आस्क्ड मी टू कंटिन्यू टिल न्यू वाइस चांसलर्स कम इन। What I have done, I have tried to listen to everybody. Everybody has a problem. You don't be part of the problem. You try to help how you can bring some good solutions. So this is my last message to you. I mean, I will be here. Inshallah, we will keep talking. We will keep enjoying. We will have better life. तो ये जो मेरे बेटे बेटियां बैठे हैं, you are lucky कि आप बहुत जल्दी फ्रंट सीटों पर आ गए। So the world is moving fast. Just work hard, listen to your teachers, listen to your mentors. We are here to help you. I mean, and be the best on the earth. बहुत सारी दुआओं के साथ अल्लाह हाफिज।
Thank you so much, sir. This was Professor Dr. Muhammad Mukhtar, the Vice Chancellor, the National Skills University, Islamabad. So I just got to know that your hometown is Bahawalpur. Actually, I was confused at whether you belong to Islamabad or Faisalabad because the sense of wit you have, it just exposed to everybody that you belong to here. The 2 years you spent here in Faisalabad.
जो मुझे अच्छा खाना खिलाते मैं उस जगह का।
आप वहाँ गए।
Anybody feed me good, so I'm a Polish also because I'm thinking that they have a good dumpling.
And for our foreign guest, I just want to tell you that our worthy speaker was sharing her exuberant nostalgia.
We are thankful to Honorable Dean, Faculty of Sciences. He has to go to some meeting. So now our technical session, first technical session is going to start. So with our first speaker, I think you know. So change picture.
Right.
And Professor Mukhtar and Dr. Taira. Professor Mukhtar and Dr. Taira, they will be chairing and co-chairing this session.
So, ladies and gentlemen, let's begin our technical session. Please put your hands together to welcome our foreign guest, Professor Dr. Alexia Novacek. Please, ma'am. We do have her introduction, but it's in the Polish accent, so which I didn't know, and I got to know her accent, both their names before this, before the beginning of this event, and she helped me with it. But I cannot do any more because there is a long introduction. So I asked her, and she said that she will introduce herself before you. So please, ma'am. Okay.
Thank you so much. As we know, my name is Alicja Nowaczyk. I would like sincerely thank for Professor Amer Jamil and authorities of Agriculture University for inviting me to this symposium. I'm the head of the Department of Pharmacometric and Molecular Modeling. I have come to you from Poland with my colleague Łukasz Kiełkowski. Where is Łukasz? Hello. We are currently working at the Faculty of Pharmacy, Ludwig Rüdiger Collegium Medicum in Bydgoszcz, Nicolaus Copernicus University in Toruń. Such a long name. And we had some headache with this name. The research interests of my team are focused on medical science and pharmaceutical science in the field of predictive modeling and detailed mechanism of action and pharmacological efficiency of biological active compounds. In the area of neuropharmacology, cardiovascular, musculoskeletal, and safety pharmacology. The scientific research carried out in interdisciplinary in nature and need a larger research group. I actively collaborate with specialists from Poland, Lithuania, Denmark, Spanish, and German. And I hope that the current event will give an opportunity to start new cooperation with Professor Jamil's group. Okay, thank you so much. The research presented today focused in acetyl salicylic acid tetramorphism. The choice of the topic was determined by two facts. First, it was a research project recently completed in my group. And secondly, the topics related to aspirin is somewhat related to the Department of Biochemistry in Faisalabad. The drug we used to treat various diseases usually come in tablet form, contain the active pharmaceutical ingredients in the form of fairly grounded crystal. Polymorph is a phenomenon in which most pharmaceutical molecules can adopt more than one crystal structure. This phenomenon is important in pharmaceutical chemistry because different polymorphs can differ in physical stability and biological efficiency. Regulatory controls require defined robust process for producing a particular crystalline form. Predicting crystal structure using experimental method and computational tools has proved to be useful in polymorphism research, depending on difference in crystal lattice parameter, crystal packing, and some molecular conformation. Polymorphs can be divided in two categories, conformational and packing polymorphism. Understanding those differences is important for both a patent and process perspective in pharmaceutical industries. In packing polymorphism, the molecules share the same molecular conformation, but are packed differently in three-dimensional space. The typical example of packing polymorphism is a paracetamol. This is a common analgesic medicine. Paracetamol is the standard and first-time treatment for fever and acute pain, and is believed to remain so far many years to come. It is usually obtained by monoclinic prism. We have view left side picture, and less stable orthorhombic polymorphs having better physical properties for pressing in tablets. This is shown in the right side picture. Conformational polymorphism arise when multiple molecular conformation can be stabilized in solid state. In that case, crystallizing gives rise to polymorphisms that differ not only by mode of packing, but also in molecular conformation. The crystallization tendency for flexible molecule arises from the presence of multiple conformers in crystallizing media. It is an important phenomenon because its impact on the properties of all solid materials. I choose chloroproamide as example of a drug with proven conformational polymorphism. Chloroproamide is an anti-hyperglycemic agent. It is a sulfonylurea derivative presenting prolonged pharmacological action. Chloroproamide has 5 stable forms at room temperature and pressure, all accessible through solution crystallizing media. This means the alpha, beta, gamma, delta, epsilon form. Chloropromide molecule can hydrogen bond in an orientation that resembles some Z or pi letter. The acetyl salicylic acid with chemical abbreviation ACA in the pharmaceutical has trend name aspirin. ACA is one of the most widely used drugs in the world. The history of ACA is very rich, but ACA as an approved drug spans 3 centuries. It was first synthesized in the 19th century, 1897. It appeared to pharmaceutical market at the beginning of 20th century since 1900. In the 21st century, there was a therapeutic switch. Exact in 2015, ACA was repurposed to treatment of colorectal cancer. In the 21st century, various polymorphic forms were also confirmed by spectroscopic and crystallographic studies. In the 1930s, Spanish philosopher José Ortega Gasset said that modern man is living in the age of aspirin. He was not wrong, even though this drug is not a panacea for all disease. However, it is difficult to image modern medicine without aspirin. It is extremely attractive to many patients because it's cheap, easy to use, and effective drug. It is also still a very attractive drug for scientists and researchers looking for antidotes to even new treats. Even today, it is one of the most studied drugs in the world, with some 700 to 1,000 clinical projects during by year. What about some crystallographic data? To date, a total of 30 ACA crystal structures has been deposited in Cambridge structural database and associated with ref code remaining from AC Sala to AC Sala 29. As we can see, 8 form 1 structure, 5 form 2 structure, and 3 form 3 structure, and 2 form 4 structure are known. In this noteworthy that form 3 is not only for spectroscopic data, and so far its molecular structure has not been plotted. For this reason, we can analyze only 3 structures. The structure with lowest value of divergence index were selected for our analysis performed in this study. The structure with ref code AC Sala 21 is form 1. Form 2 is have ref code AC Sala 20. and four have Rivcode AC Sala 23. In order to obtain information about some structural similarity of the studied ACA form, table conformation was selected for the ACA crystal for similarity determination in the superimposition procedure. Carbon and oxygen atom expect hydrogen atoms forming the basic skeleton of ACA were selected to matching procedure. The structural similarity was calculated as RMS matching. In the general, the lower RMS value, the better similarity. Comparison of the present geometries of the studies ACA form indicates a high similarity in the orientation of the points for all of crystals. The RMS deviation for each group are for form 1 and 2, 0.51, for form 2 and 4, 0.49, and for form 1 and 4, 0.33, respectively. Those results indicate that the pair of ACA form between 1, 2, and 2, 4 have highest RMS value in the set. This is due to the fact that the acetyl group show different orientation with respect the plane of the benzene ring. In contrast, the ACA form 4 differ from both form 1 and form 2 by mutually reverse orientation some carboxylic group. Based on this result, we conclude that three different conformation is observed in acetyl salicylic acids, and this proves that acetyl salicylic acids have present conformation polymorphism phenomenon. The study used Hirschfeld sulfate analysis to investigate non-covalent interaction responsible for crystal packing. The structure and electronic properties of the crystal were verified using density functional theory. The Hirschfeld surface were mapped using geometric Hirschfeld surface distance and fragment patch plot. The shape index of coordinates plots the recurrent quantum mechanical properties for their evolution. The Hirschfeld surface were mapped over a d-norm ranging from minus 0.77 to 1.20 for form 1 and minus 0.74 to 1.31 for form 2 and minus 0.72 to 1.3 for form 4. As we can see, those are similar values. A compression of the volume and surface area of Hirschfeld surface shows that respective value for form 1 and form 2 are comparable. Their respective volume and surface area value for form 4, on the other hand, are approximately twice as large. This is due to the fact different packing of ACA molecule in the crystal cell. Form 1 and form 2, respectively, each conformation contain one molecule ACA in the elementary cell. The elementary cell of form 4, on the other hand, contains two ACA molecules. This proves the packing of molecule is observed. Okay. As we see on the screen, the electrostatic energy contribution significantly prevails over the dispersion energy contribution for the molecule in interacting through the hydrogen bond and with interaction. The energy compression of the study's centrosymmetric dimers decrease in series. For one, more is less for form 4 and form 2. This proves that the most stable structure is formed by form 2 with energy equal minus 83 kilocalories. Form 4 is about 7 kilocalories less stable. A compression of the calculated energy for the dimers of form 2 and 1 respectively reveals differences in their stability about 10 kilocalories. In all AC forms, the similarity of fingerprint lines between ACA form 1 and 2 is revealed. Form 4, on the other hand, is characterized not only by different distribution of fingerprint line, but also a different percent of corresponding contacts in the coverage of Hirschfeld surface. For all three forms, we observe sharp spikes appear in the fingerprint diagram, resulting from strong interaction contact between oxygen and hydrogen. Those are intermolecular hydrogen bonds, carbonyl hydrogen bonds, and hydrogen. The next most significant contacts come from H8 and CH interaction. The structure of ACA fields 1 and 2 are stabilized mainly by the reciprocal interaction hydrogen bond, accounting for about 42%. The intermolecular HH contacts accounting for approximately 34%, revealing a slight spike on the diagonal axis. In contrast, form 4 structure is mainly stabilized by inverse intermolecular HH contacts, generating the largest share of Hirschfeld's surface coverage. Is approximately about 38%, revealing a slight spike on diagonal axis. In addition to sharp spike appearing in form fingerprints, the diagram also due to strong oxygen-hydrogen interaction contacts. However, they give about 10% smaller contribution to Hirschfeld surface coverage compared to corresponding interaction in form 1 and form 2. In contrast, CH intermolecular interaction for form 1 and form 4 account for about 19% of the contribution, while form 2 is characterized by about 1% reduction to the contribution of such contacts in Hirschfeld surface coverage. This observation clearly indicates that in adoption to carbonyl group and hydrogen oxygen, hydrogen and C hydrogen contacts have significant effect on the molecular packing of ACA molecule. Human serum albumin is a major protein component of blood plasma and plays an important role in the regulation of colloidal osmotic pressure and transport numerous endogenous compounds such as fatty acids, hormones, and some another. Albumin also brings a wide variety of drug molecules. During drug molecules in the general circulation are either bound to plasma protein or exist as unbonded form. Depending on chemical properties of the drug molecules, unbonding drugs can passively diffuse through the barriers constituted by endothelial cells and tissue cells into organ tissue and undergo metabolism. Such serum albumin drug binding is an important factor in our understanding of pharmacokinetics and pharmacological effects of the drugs. Crystal structure of albumin is shown on the screen. As can we see, the albumin is subdivided into three domains. 1, 2, and 3, and subdomains A and B. Human serum albumin has a limited number of high affinity binding seeds. Current crystallographic studies have provided that majority of drugs bind to the specific binding seeds on human albumin. They are SID1, also known as warfarin binding seed, and SID2, also known as the diazepine binding seed. On the screen, the approximate location of SID1 and SID2 is shown in the green and red squares. In the center of the image, the X-ray structure of the albumin crystal squares showing the two specific binding seeds for SID1 and SID2 in this protein. Docking results of individual ACA polymers are shown on the right and left respectively. From top, ACA form 1, ACA form 2, and ACA form 4. Red and green big squares identify equivalent reservoir in superimposition of 3D structure. As we can see, the obtained data of binding energy and affinities in SID1 and SID2 are similar for investigate form of ACA. However, the most desirable value are observed for form 1. From a pharmacokinetic point of view, ACA will be the best distributed from the blood to the desired seat of action in form 1. As a validation experiment, we choose to perform a docking experiment for drugs with documented affinity for human albumin binding seed, such as varafin. Varafin for SID1 binding affinity is equal 4.47. Phenylbutazone binding affinity is equal 5.89. The result of the individual for SID2 diazepine is for SID2 and binding affinity have 5.89. The result of a validation experiment for the individual drug molecule are shown on the right and left respectively. From top we have varafin, phenylbutazone, and diazepine. Red and green big square identify equivalent reservoir in superimposition of 3D structures. Comparison of the references value from the literature data to the value obtained in our docking experiment shows that reproduction of the trend of the respective value. This demonstrates the higher accuracy of our prediction of specific interaction in our model. Based on our research and calculation, we conclude that ACA exists in 4 polymorphic forms. For 3 forms, the crystallographic structure is known, and 1 has a desirable spectral structure only. ACA form, conformational and packing polymorphism, the energy stability of studied polymorphs decrease in series. Form 1 more than form 4, more than form 2. Binding affinity of studies ACS human serum albumin complex change in order. Form 2 more than form 4, more than form 1. As ACA form 2 will be the best for absorption from the gastrointestinal tract into the blood stream. ACA form 1 will be the best choice for distribution from blood to active seed. From the pharmacological point of view, the form 1 of ACA is the most desirable form. As what I need more for this study, as the author of the research project presented to you, I feel some certain research deficiency. Personally, I feel that present research lacks experiment in fields of biology or biochemistry. It seems to me that biological study assessing the interaction of individual ACA polymers with albumin proteins would be valuable enrichment to our study. Thank you for your time and attention.
Okay, thank you, Professor Dr. Alexa Novak-Zak. I think I did a good job.
Thank you so much.
So, what a beautiful lecture. So now the session is open for question. Okay.
Thank you for your presentation. So I have a very basic questions. Do you see the difference in melting point of this four polypharmaceutical compound ASA? Difference in melting point?
In medical way, right now we have some big question. Which form is the best choice for medical use? And in already in market, we sell only form 1, but this study proved that this form is the best choice for medical use.
No, my question is that, so you have a four polymer compound, huh? Different crystal form. Have you observed the melting point of this four compound?
I think he's asking for melting point of four polymers.
I don't know melting point right now for aspirin.
Because I think it should be the same melting point of all four polymers. Because I have the same issue with me also. And I have also paper. So I observed that if you have a different crystal form, but they have a same melting point.
He's facing same issue like that. Melting point.
Thank you, Dr. I think that's a very, because she is telling about 4 compounds, so you think they should be having the same melting point. Thank you very much. Thank you. So, okay, next question, please.
Nice talking, Professor Alicia. It was good to learn more thing about the old compound of APC, aspirin, and you're talking about that. Do you have done some biological trial or animal trial for your study?
No, I don't have. As I say to you, I feel some disappointing for this lost experiment. I need some biological and or biochemical study.
Do you have success? This compound is, is logically done in the market, doing a job more than a painkiller?
This is in the market. We use form 1 is already, but this study is some theoretical study with enable crystallographic structure of aspirin, and we need some answer which form is the best choice for the market. Right now we can buy this crystallographic form, and we'll try some biological tests and some biochemical tests and find answer for question what is the best active with albumin, for example. We need this.
Thanks.
Yeah, there is a question from this side of you.
Okay.
Please.
Thank you very much. Very nice presentation. I have one question. You have said that the acetyl salicylic acid form 2, it is best for absorption from the gastrointestinal tract. What could be the reason for this absorption, and which animal model you have used for this study?
I don't use any physical models. I use only molecular modeling models, and we test by Doppler procedure.
Okay. Thank you.
Question.
It's not a question, actually, just a comment that this will be the major part of the MOU we were discussing, that the molecular modeling models we will be looking for testing in animal models and in cell cultures so we will be collaborating with this. So this we will be have interaction for the future. So far they are working on molecular modelings.
Yeah, I hope so.
Any more question? नहीं है? So I will request Dr. Taina to say word of thanks to our honorable speaker, please.
Thank you very much, Dr. Alicia.
Yeah.
Sorry. Next name. No. Exit. Thank you very much for your presentation.
Thank you so much for your attention.
Thank you so much, Dr. Alexia. And now here is an announcement, ladies and gentlemen, that now we have here with us the master of the masters, the teacher of the teachers. Please give a big round of applause for Professor Dr. Khalid Mahmood Khan. The patron in chief says forward. So it's a great honor for us for having you here. Now we'll move towards our next lecture, which is of Dr. Muhammad Asghar. He is from National University of Medical Sciences, has more than 17 years of experience in the pharmaceutical industry and academia. He has been holding esteemed positions such as General Manager, Genome Pharma Hatter, General Manager at Madeira Pharma Islamabad, and Head of Quality Operations at Biogen Rawalpindi. His expertise extends beyond the realms of academia and research into practical applications within the industry. He has developed over 80 common technical document CTD dossiers, facilitating the regulatory approval process for numerous pharmaceutical products. One of his notable achievements includes the establishment of a 64-bed multi-study drug bioequivalence center and drug testing lab, underscoring his commitment to advancing pharmaceutical research and innovation. Furthermore, his dedication to quality and process improvement is exemplified by his certification in Six Sigma methodologies from Kansas State University, Georgia. He is also actively engaged in professional organizations such as the American Association of Colleges of Pharmacy and the American Lung Association. Please give a big round of applause for Dr. Muhammad Asghar.
बिस्मिल्लाहिर्रहमानिर्रहीम अस्सलाम वालेकुम। इस सारे तारुफ के अंदर जो एक चीज रह गई वो ये कि आई हैव आल्सो ऑनर टू बी ग्रेजुएटेड फ्रॉम दिस यूनिवर्सिटी एज 98। ये यूनिवर्सिटी आज भी उसी तरह हमारे दिल में बसती है। टुडे आई विल लेट्स talk on the topic of this, the importance of bioequivalence in generic drug, ensuring safety and efficacy. But I will take the opportunity to discuss the gap between the students, researchers, the senior researchers, and will suggest a little to our high-ups those who can administer or can decisions to step ahead where we are stuck in between the research, a real research, and the development of the drug. In the last talks, sir told that still by using AI, by using computational programs, by using a lot of things, but still we are lacking to introduce a single drug in the international market that is being made by Pakistan. Where is the gap? All of you, okay. Where we are lacking, we have to narrow that gap. I will start with one example. All of you use the vehicles to move from one place to the other place. What is the most important thing in driving that vehicle? That is the use of the seat belt. That saves your life and have been saved millions of lives. In the same way, when you use any drug, what is the safety? What is the safety level and what is the efficacy? Somebody once asked me, what is chemistry? It was a very difficult question for me. But my answer was very simple. The science of everything. If you think the science of everything is the chemistry. And when you talk about what is biochemistry, the science of living thing. And what is the drug then? Anything, any element that affects your mental and physical health. We can take it as a drug. Early, we have been told that 33 PhD students are here in the biochemistry department, if I'm true. They all are mostly in biotechnology, in chemistry, in chemical engineering, in all molecular biology and genetics. We work on trying to discover a new compound, especially in our third world countries in Pakistan. We always try to discover a new compound, and many of them got succeeded doing their NMR, doing their electron microscopy, doing their qualitative and quantitative checks and everything they do. But what's the next? What is the next step? Where we are? Have we ever tried to take it towards a safety and efficacy level? In Pakistan, the same scenario is with the pharmaceutical industry. They make the generics, but they cannot recognize internationally because of the lacking of this bioequivalence facility in Pakistan. There are few bioequivalence centers in Pakistan, but unfortunately, none of it is internationally recognized by WHO, FDA, USA, MHRA, JICA, even PICS. Okay. Before starting this, if you take the drug, how you confirm, how you are sure that the pill you have taken, the capsule you have taken will affect properly and will go to the bloodstream or will pass away? Have you ever think the drug you are taking, what is the background of the efficacy and safety of that drug? Have you ever tried to learn about it? The drug you are taking will cure you or will give you some side effects? We will discuss in the later stage why the generic drugs needs all these things. But when the safety concerns, if I go back to 1937, sulfanilamide, One disaster happens in 1937. So sulfanilamide was thinking at that time as a miracle drug and was being used as an antibacterial drug. Some pharma companies decided to market it as a liquid drug for sore throat. In development, most of the chemists sometimes do unethical things. The problem with that salt is the solubility of that salt. It was not hydrophobic, it was not hydrophilic, or it was not soluble both in water and alcohol. They tried, the chemists discovered to dissolve it in diethylene glycol, the chemical used as antifreeze in car coolants. They made the drug and marketed it, and resulted in a disaster of killing of a lot of people. When I was making this slide, I just got to remember, not far away, in November 2023, I read a report that the WHO have banned 6 cough syrups from Pakistan. And the only reason was they contain the traces of ethylene glycol in them. Someone tried to use the active material first in ethylene glycol, then they make and they just evaporate it, but the traces remain in that drug and they marketed it. Even they exported it from Pakistan. The second very famous case you have heard in 2022, when the cardio drug was being made and accidentally they put perimethrin, which was used as an antibacterial drug, and killed 130 people. So in case of raspberries was case of death in 1937, which forced administrators to legislate food, drug, and cosmetics. The second case was thalidomide in the late 1950s and 60s, initially marketed as a sedative and treatment of morning sickness in pregnant women. It was later discovered that the drug caused severe birth defects, in particular limb deformities when taken during pregnancy. The disaster resulted in thousands of babies being born with a disability around the world. So safety, while I have told you what is the chemistry, the science of everything. So what is the safety? Again, the safety is the life. First, concern the safety always. When you discover a new compound, doesn't mean it's a discovery, but take it to the efficacy and safety level. Many of the thousands compounds discovered, but a few we took till the preclinical studies. An approximate of the 10% of the discovered compounds goes through the preclinical studies. And from that, 5 out of 5,000 new drug candidates are released, are tested in human, and only been approved. One is approved, it's by chance. Not even one is considered to be approved. Average cost of bringing one medicine in the market from scratch, from preclinical to clinical to the market, from phase 0 to 1, 2, and from the pharmacovigilance, it takes about 800 million a lot of money, a lot of time, and then it is patent for 20 years or more. Sometimes it can be reduced or patents can be sold. Now, comes to the generic drug. The generic drug is the is the copy of the drug from the innovator. It may contain the same content of the active, but when it is made, for example, you take a paracetamol or ciprofloxacin tablet in Pakistan, they all are generic made. When the drug is made, each company develops its own process, its own quality control checks, or its own quality assurance and method development checks. Copy of the brand doesn't mean that it has same efficacy or same repeatability or the same effects on your body. We can, okay, once the ciprofloxacin as a ciproxin made in somewhere in the Europe, they do the clinical studies, preclinical studies, effects, after effects, drug-drug interaction, even drug-food interaction. They perform all the studies at the time of the development of that drug. But when the generic drug is made, that part is assumptive. Especially in Pakistan, we discussed with the Drug Regulatory Authority of Pakistan, the DRAP. 3 times we discussed that where is the bioequivalence part in the chemical technical dossier? It is a part of CTD, but still not applicable in Pakistan. This is the main gap or the red line we have to cross to market our product in the international market, because without this bioequivalence, nobody accepts that your drug is safe. Pakistan exports about $300 million in pharma sector, as compared to the Germany $115 billion export of the pharma. So what is the huge economical loss of the country? Just because of the once that were a bioequivalence center in Pakistan that can be certified nationally and internationally, where will be the industry of Pakistan? While developing the generic drug, we have the myths. Generics are not so safe. Why? Because they may contain the excipients that have not been tested for their trace elements. What might be the effect of these trace elements on the body? Are not so potent. Where is the drug bioavailability? Take longer to act in the body. Extent of release, the exposure at the site of action. Made in substandard manufacturing facilities. The disasters happens in the last years is because of this substandard manufacturing facilities, the not validated processes. Bioequivalence is the only most important factor that we need to consider. Provide a safe and effective drug, equivalent in therapeutic value, in efficacy and safety as with the innovator drug. The permission of substitution with a full expectation of the same clinical effect, safety profile, tolerability as the innovator product. The same again, the generic drugs doesn't mean that they have to perform all the clinical testing as the NDA new drug application provided. There is a little difference in bioavailability and the bioequivalence. Bioavailability is simply the rate and extent to which a drug is absorbed from a pharmaceutical formation and becomes available at the site of action. But in bioequivalence, we have to compare 2 drugs. We develop a drug, scientists discover a salt, it goes to the further research in animal modeling. The animal modeling, it goes, we normally use the 15th part of that animal modeling drug to the human drugs. And then this human drugs then passes the clinical trials, goes to patent and market feedback. And after patent, it goes to the generic drug. And this generic drug must be checked through bioequivalence or clinical trial that it is safe and with the same therapeutic value the innovator drug have. Simply taken by mouth, goes to the stomach and the pathway everybody knows and they excreted. But at the elimination sites, it may go to the kidney, liver, maybe metabolites are formed. Now, no clinical studies are being performed in patients with a generic product to support the efficacy and safety. Therefore, the trials must be conducted to ensure two products do not differ. Is it there any example in Pakistan how many percentage of the drugs? There are 750 companies in Pakistan, pharmaceutical companies in Pakistan. Out of 30 to 45 are exporting to the world. The big names are also there, but 80% of the pharmaceutical requirement being completed by the local industry still have only 5%, 3 to 5% of bioavailability available for the generic drugs in Pakistan. Where we are standing. By taking this, I requested to suggest one thing in our universities, in our research stations, in our academia. We're all doing the basic research. And my aim and suggestion is to request, please step ahead to make bioequivalence centers, bioequivalence studies, bioequivalence courses, so we can know what is the requirement of the international market. We took the next step, our compounds to the clinical trials. We have taken the initiative in National Institute of Medical Sciences. This is the first university that has developed the bioequivalence center. We are going to accredit with WHO and FDA USA inshallah very soon. So please, I request all the administrators to consider to develop the local industry by the courses, by the research to the next level. This is the official definition by 21 CFR and ICH guidelines. Absence of the significant difference in the rate and action to which the active ingredient and active moiety in pharmaceutical equivalent or pharmaceutical alternative becomes available at the site of the action. The whole thing goes again and again in a circle that the drug safety and efficacy can be achieved only by the bioequivalence. Pharmacokinetic and pharmacodynamics in the bioequivalence is simply what the drug did in the body and what the body did with the drug. Usually when the drug levels cannot be measured are not relevant. Pharmacokinetics is the main key of achieving the bioequivalence, the Cmax, the concentration when the drug get the maximum concentration in the blood or the exposure site. And what time it will take to get the maximum concentration. Sometimes some drugs are prolonged or multiple dosage forms, so we cannot get the Cmax so early or their half-life is so long. So we sometimes drugs convert into some metabolites, and we use the pharmacodynamic studies for, which is not much recommended, but sometimes we do the metabolite studies for bioequivalence. In vitro, in the pharmaceutical industry, when the drug passes through the quality control, is one part is the dissolution, the comparative dissolution, which is mandatory that it gives the same conditions for innovator drug, 14 basket dissolution, gives the same condition, same parameters, same requirements for the innovator drug as well as the generic drug, which must be compliant in between 80% of the resolution testing to the 120% of the assay of the active drug. But this in vitro dissolution again does not means that it gives us the same result within the body because it contains many excipients. That excipients may cause hindrance in absorption or enhance the absorption of that drug. Even sometimes when we make the drugs, we consider the particle size of excipients, the shape of the particle size of the excipient, and the processing method of that drug with the drug.
Participants are humbly requested not to talk, please.
जब आप ये दवाई खाएंगे और आप पे असर नहीं करेगी ना, फिर सोचिएगा इन सारी बातों को what you are doing. You must know the drug you are taking, what it may affect on you. This is again the same thing. When you drive the car, you are unseen to any incident. So you use the safety belt. In the same way, you are unseen. What you are taking to the drug, what will happen inside your body until you develop the sense or we develop the culture, create the safety and efficacy, ensure that safety and efficacy. Anyhow, in the dissolution, we just compare the innovator drug with the excipient drug, with all the excipients, with all the process, with all the validations, everything that they have the same conditions, they have the same release value. But when we take in the body, they may differ because of the excipients. They may hinder or they may affect the absorption of the drug inside the body, especially the PGP proteins. The biological drugs with high permeability and high solubility drugs sometimes are exempted from all these things. There are different test methods for assessing the bioequivalence. The comparative bioequivalence is to compare the two drugs. In vivo measurement of active moiety in the biological weights, either it may be or preferably it is blood, but sometimes it is the urine samples that can be taken for this study. The comparative pharmacodynamic studies in humans for those drugs where the active may convert into some metabolites, it cannot be extracted as it is in the bloodstreams. In vitro dissolution test, which is the basis of first release of the drug into the market after manufacturing in the industry. When new drugs are to be administered, sometimes not all the drugs are required for bioequivalence, especially for the parenteral drugs. The drugs that are injected directly into the bloodstream or into the body because their bioavailability is 100%. It doesn't have to pass through all the liver and through all the system. It is directly injected to the body, so they are readily available 100% to the bloodstream. Or sometimes the other topical drugs and some other drugs, rectal drugs that are not passed through the systematic way, so they are not required for the bioequivalence studies. What is the protocol and why, how we perform? I will just give the overview how we perform the BA studies. The study objects. Bioequivalence and clinical trials are two different things. Sometimes we mix it. In the clinical trials, we do trials mostly on the patients, but in bioequivalence, we do trial in healthy volunteers, preferably males. But both genders, male and females, can be the part of the study. The scientific question to be answered, the bioequivalent, the availability of that drug into the body. Study design, it's a parallel or crossover. Study circumstances, the circumstances where the study is going on. Subjects informed consent. If I am going to perform a study, I will take the consent and will inform very clearly the side effects may have and many things. I was in University of Rochester, New York. We were doing a study on AIDS. सबसे मुश्किल काम था उसके अंदर volunteers ढूंढना. Nobody was ready to be the part of that study, especially for the clinical trial and bioequivalence. They have done a vector vaccine for that, but then again we decided to do all the studies in the patients. So we inform everything to the patients and the volunteers, what might be the side effect and what is the remuneration and what can be done after the study. So the most important part of that is the bioanalytical method. At the moment, the most common thing we are using is the LCMS studies. The study design, again, single dose, two-phase, two-way crossover designs. Alternatively, single dose parallel designs or the clinical trial designs. Minimum, the regulatory requirement is 12 subjects, but we take normally the 16 subjects. Sometime one or two may be excluded, healthy adult volunteers, gender male or female. Women should not be pregnant or should assure not to become pregnant during the study. Essentially, the Cmax, again, the three common things we have to consider in the bioequivalence studies is the Cmax, area, achieve the Cmax, and area under curve. This is the exposure of the drug. The curves we got during the study, varying absorption rates. If the geometric ratio we got 80 to 125%, we may consider the drug is available or bioequivalent to the innovator. Area under curve is a measure to expose the drug of the body, test sampling time, and infinity is the theoretical measure of the total exposed drug of the body from administration till the drug is eliminated. Extent of absorption. The coefficient of variation is always important among the subjects when we're taking the samples. The difference, how much get sample to sample. Possible results with a 90% interval, confidence interval is 80 to 125, 0.8 to 1.25%. Some clinical effects, some safety profile, and some bioavailability formations are said to be bioequivalent. Again, this is all the game of the drug efficacy and the concentration. आपने दवाई खाई, जिससे मैंने बताया कि what happens with your body. When you take the drug, if it absorbs only 10% to 20%, what will be the efficacy of that drug? Though it passes all the qualitative and quantitative quality control checks, quality assurance checks, all the validations, but still it absorbs only 20% into your body. What will be the efficacy of the drug? Only method is bioequivalence. when you claim that our drug is bioequivalent certified with the innovator drugs, the doctor and pharmacist may substitute the more expensive product. Sorry. All samples used in the BAB studies should be retained. This is all the methods I will go through only. And the record must be kept legally for 2 years. Bioequivalence study is clinical research study which facilitates to get drugs at low cost with comparing its safety and efficacy. Currently saves consumers 60 billion, 30,000 crore rupees. In developing countries like Pakistan, it assumes a high significance because it reduces approximate cost less than 50% of the drug. Again, when you discover a new molecule, take it to the next level. When we manufacture a new drug, when you develop a generic drug, take it to the next level with the certification of bioequivalence and be confident our drug is equivalent or better than the innovator. Thank you.
Thank you, Professor Asghar. You said very well, bioequivalence is very much needed. I think your elaborate lecture definitely might have enthused of these youth, like how much opportunities exist and why we are not pursuing those ones. So for the sake of time, because we have a Friday prayer coming in also, So thank you. We will save our question for you in the Kawali evening because that's afternoon session. Yeah. So, people can hold on to their question. I know there could be hundreds of questions on bioequivalence. So what I will do, the next presenter is here. Okay. So, I will request Dr. Mushtaq Ahmed from Quaid-e-Azam University. And my humble request to Dr. Mushtaq will also be please try to be brief so that
So this is Professor Dr. Mushtaq Ahmed, obtained his PhD in plant sciences from Quaid-e-Azam University, Islamabad, Pakistan. He is renowned world top plant scientist in the field of plant systematics and biodiversity. He is currently working as Professor, Director QAU Botanical Garden and Herbarium, and Chairman Department of Plant Sciences at Quaid-e-Azam University, Islamabad, Pakistan. He is honorary distinguished professor in China and Uzbekistan. Recently, in 2023, the government of Pakistan decorated him with highest civil award of Pakistan, that is Tamgha-e-Imtiyaz. He is an elected fellow Pakistan Academy of Sciences, which we usually call PAS, and is the member of awardees of international bodies. That is, I have multiple names in front of me, including HEC, PBS, BSP, etc.
Thank you.
Over to you, sir.
Thank you. Thank you so much for inviting me. I'm very honored that my mentor, Professor Khalid Mahmood Khan, when I was MSc in Narad University and he was the Vice Chancellor, when I was elected as a fellow, he was the President of Pakistan Academy of Fellow. And when I was decorated with Tamgha-e-Imtiyaz, he was the President of Academy of Sciences. Thank you so much, sir, for your mentorship. And thank you so much, my respected Professor Dr. Mukhtar, the Vice Chancellor of the National Skills University, Professor Dr. Amir and Madam, and our guests from the Poland, ladies and gentlemen. I just give you the glimpses for the students of the biochemistry, biotechnology, and the biological sciences. What will be your futures after BS, MS, or PhD? I'm from Quaid-e-Azam University, and you are familiar that Quaid-e-Azam University is the only top-ranked university in Pakistan that is included in top 400 universities of the world. It's the 86th in Asia and 9th position in the South Asia. Do you know that Pakistan is a very beautiful country with the rich diversity of the plants, natural products,
We have the plains, we have the mountains, we have the glaciers, we have the deserts, moist temperate. So Pakistan is a rich cultural heritage with respect to the plant diversity. What is interdisciplinary biological sciences? Now it is a time that the biological sciences, biochemistry, biotechnology, agriculture sciences, and the social sciences work together on natural products for the drug discovery in order to make the pharmaceutical to the nutraceutical industry. Now it is a time that the medicines, they are moving toward the nutraceutical, that food you use is act as a food as well as the medicines. And the biomedicines are the medicines that are obtained from plants, animals, microbes, minerals. So the time of the nanomedicine, now the world is moving from pharmaceutical drug to the eco-friendly nano and biomedicines. And the bioprocessing, that is the biotechnology, they play a very crucial role for the synthesis and the bioproductions of the plants medicines that are effectively useful for the sustainable as per the SDGs. As far as my research group is concerned, my research group is working in Italy, Pakistan, China, and the United States of America. We are basically focusing on the natural drugs from the medicinal plants. We are focusing on the nanomedicines from the medicinal plants and the natural active ingredients based on the chemical characterization, and they are best molecules for the drug discovery with respect to the biochemistry and the biotechnology. Diversity of Pakistan. Pakistan is a rich, diverse country with agricultural crops, medicinal plant, and number of other essential oils that are used for the aromatherapy. Cosmeceutical industry, pharmaceutical industry, and the nutraceutical industry, they use these raw materials for the production of the drugs, for the production of the eco-friendly cosmeceutical products, as well as the natural products for the dietary, the nutritionists, that the world is now moving from medical sciences to the dietary. So the plant, they provide a number of the raw materials for such type of the value-added product to the industries of pharmacy, nutraceutical, and other industries. What are the emerging technologies for the drug discovery development? Everyone talk about the molecular docking, drug discovery development, chemical characterization. So all these things, they combine for the innovation for the SDGs, that is the safe health to the community, that's the green health for green community. So this is the time that you think about the interdisciplinary science. Not only the botanists, not only the biochemists to do this job, it's a joint venture of the biologists, the agriculturists, and the social scientists to develop side drugs which have acceptability to the communities, and they have an impact to the social society. Just to see that the barbaris, everyone know when they have some urinary infection or the blood problems, the hypertension and the diabetes, you have the natural dietary plants in Pakistan. This is the just example of the barbaris. It's the superfood which is used for the hypertension and the blood pressure by a proven model. So this is the natural drug that is an anti-cancer as well as the one of the important nutraceutical diet that have a great impact to your health. Another important local medicinal part is the Caressa. Caressa is one of the important rich vitamin C. During the COVID, every medical doctor, they advise you to take the vitamins C or the immuno boost. So you have the natural drugs in the form of medicinal plant. Caressa is the local medicinal plant, which is a natural immuno boost, and a number of the drugs, they are derived from this plant by a number of chemists and the biochemists. Everyone know about the pongam, that is the everyone in Pakistan know about the great production of pomegranate, that's Indica granatum. But the wild pomegranate is a natural anti-cancer source for the vitamin C and the number of the natural active ingredients that have a great impact on your health. Everyone know about the black nightshade, that is a solanum. It is one of the important medicinal vegetables in Pakistan that have a number of the active ingredients which are used to cure the cancer. So these are the important raw materials available for biochemists, for biotechnology, and the chemists and the pharmacists to pick these local medicinal plants for the drug discovery, for the active ingredient extraction, and then they lead toward the natural dietary compound and the drugs. Just see the title of this paper. We have recently published in December. Bitter is better. Bitter is better means there are a number of the medicinal and wild vegetables. So this is a joint venture of the European and Asian peoples to develop such natural drugs or compound from bitter plants, the plant which have a little bit bitter taste. So they provide a number of the active ingredients for the cure of the diabetes, blood pressure, and so this is a joint venture multidisciplinary sciences from botany, from biology, biochemistry, and biotechnology. You have a number of the natural active wild edible foods. Everyone know about the cultivated foods, but Pakistan has a great diversity of the wild edibles. So these wild edibles are the precursors, are the raw material for natural drug discovery, for nutraceutical and the pharmaceutical drugs for the society. First step is the correct identification of the medicinal plant. When you are thinking about the drug discovery development, you have a correct identification of such natural medicinal plant in the field with the expert opinion of the botanist. Next step is the extraction of your desirable chemical compounds or the active ingredients. So it depends upon the part used by the local communities, either the fruit, either the leaves or the stems. Next step is the extraction of these active ingredients by the biochemist and chemist, because the botanists, they identify the plant, while the biochemists and chemists, they extract the active ingredient or the natural compound from these medicinal plants. Then it is the work by the chemist to isolate and characterize these active ingredients. For the next step is the drug discovery development. So where these natural compounds they are applied, you know that they are used for the nutraceutical products. They are used for the pharmaceutical product, that is cosmeceutics and the medicine. And they are also used for the veterinary diseases, livestock diseases. There's the animal feeding, because we are a meat eating nation. We must improve the milk production and the meat production by using such active ingredient. And the agriculture, apiculture, bioenergy, biochar, all these are multidisciplinary sciences to produce such natural food, natural drugs from these medicinal plants. So this is I added a slide for you students. When you are thinking about the drug discovery development, you have to start from local medicinal part, then move toward the chemical and biological screening by involving the zoologists for the clinical trial by using the animals, and then the biochemists and biotechnology, they develop the drugs, and the pharmaceutical industries, they develop the drugs and market these drugs to the community. So this is just a bottom-up approach from medicinal plant to the pharmaceutical industry by involving the chemists, biochemists, biotechnologists, and the biologists and the society. So this is just a slide for you. Bottom-up approach, pharmacology leads, ethnopharmacology leads toward the chemical and biological screening of these natural active ingredients, and then they move to the pharmaceutical development of the drugs for the community for the marketing. So this is just paper study. We have a joint venture of the Turkey. How the enzyme, the biochemists, they play a very important role for the inhibitory and the drug discovery development. So this is the joint venture of the botanists, biochemists, and chemists to develop the drugs. So this drug is developed by our joint venture of Turkey and Pakistan, and is equally patentable to your country as well. It's a very common medicinal part in Pakistan and also in Europe and Poland. That is the Paeonia. Paeonia is one of the important medicinal plant. We have developed a drug, Paeonia drug, from this medicinal part by a joint venture of Chinese Academy of Sciences and Qaid-e-Azam University scientists for the epilepsy and as well as the rheumatism. So this is a natural drug we have developed from this medicinal plant. That's a bioactive drugs for the epilepsy and anti-cancer from your local medicinal plant, is involving all the stakeholder or interdisciplinary science. Another important area for Pakistan is the diversity of the saline soil, because Pakistan is a great area for the saline soil, waterlogged soil. But this area has a number of medicinal plants. They are called as salt tolerant plants. So this is another important breakthrough for your country, for the minor cash crop for the medicinal plant to develop the drug from the salt affected soil from these medicinal plants. Another important medicinal plant from Pakistan is the Salvadora. Everyone know the Muslim use the toothbrush, that is miswak. is one of the important medicinal part that is used for the oral disease and the gum disease. We have developed a drug that is used for the oral cancer from this medicinal part by the joint venture of the Italian scientists, Chinese Academy of Sciences and Pakistan. Pakistan is also hosted a number of the medicinal plant. We have developed a catalog or the database about the medicinal part of the Asia that include Russia, China, Uzbekistan, Pakistan, Asia. So this book is a database about the raw material of the medicinal plants available for the biochemists, chemists, and the biotechnologists to develop the drug from a number of the wide range of the medicinal plants. Just in 2024, last week, we have published a paper about the bacteria, how they enhance the active ingredients within the plant, and then we have moved toward the drug discovery by involving the microbiology, biochemistry, and chemistry. So this is a joint venture of the European scientists and the Pakistani scientists, how the bacteria-based mediated natural compound from the medicinal plants. Another important plant is the Virginia. Everyone is conscious about the beauty. So the cosmeceutical product that obtained from the medicinal plant. So we have developed a drug that is a Virginia drug from this medicinal plant, which have a anti-cancer activity on your skin. Some people who have a cancer in your skin, that is a very natural moisturizer as well as an anti-cancer drug we have developed with a joint adventure of Qaid-e-Azam University and the Chinese Academy of Sciences. Last government said that we are invest a lot of money on the export of the black tea. So you think about the natural organic teas. That is the Chinese, they are familiar to the traditional Chinese medicine. So Pakistan is a rich diversity of the natural herbal teas that they are compared to the black tea. So you think about these herbal teas to develop some natural drugs for the treatment of the number of the diseases. During COVID, we have faced a lot of problems due to the lack of vaccine. No vaccine was developed in Pakistan, but nature gives you a lot of natural vaccine in your country. There are large number of the minor medicinal plant in Pakistan. They have a great natural vaccine impact. So this is the hibiscus. Hibiscus is one of the important medicinal plant that is present in Pakistan. But the Chinese, they develop a natural vaccine from this plant is an anti-COVID. So you have a great potential in Pakistan. You think about these plants for the natural drug discovery by using your local natural resources. So we have published just a study in 2024. The great player of the natural compound of the medicinal plant is Kashmir and Himalaya. So Pakistan has a great potential for the rural development because the local people still rely on those. So the people who are living in Gilgit-Baltistan and Kashmir, they are average age is about 80 years. Why? Because they use the natural compound from the medicinal plants. Everyone know about the muscular docking and everything. You know that in Pakistan, most of the people, they are facing a problem, especially the women's breast cancer. So we have developed some drugs from the Figunia cretica, which is an important medicinal part in our Punjab, that is called as Damasa. So this is one of the important drugs that is used for the breast cancer. So this is another important patent from our group is used for the breast cancer. So this is a male contraceptive because Pakistan is the 5th largest population country. So my research group is also focusing on another important group is working on the natural compounds obtained from the medicinal plant. They are used for the male contraceptive to control the population. So this paper is published in this about the population control by using natural drugs from the medicinal plant. So these are the few studies that are published by a joint venture of the zoologists and the botanists and the biochemists to control the population by using the natural male contraceptive from the medicinal plants in Pakistan. Pakistan has a great diversity of agriculture as in the livestock. So you think about the great meat production and milk production to discover and identify such veterinary drugs from the medicinal part. They are used to improve the livestock industry in Pakistan. So this is another important study published from my research group about the improvement of the livestock diseases by using the natural medicinal plants. So what is it's a need of the time that we work in interdisciplinary approaches by involving the botanists, the biologists, the biochemists, and the pharmacists and the nutritionists to develop such natural drugs for your use. Everyone know about the green nanotechnology. What is green nanotechnology? Green nanotechnology is actually the biochemists and the chemists to extract the natural nanoparticles from the medicinal plants. They have a great impact to your health. So that's called as green nanotechnology. So you have a great potential for students, especially the BS and the MS and the PhD. Think about the green nano catalyst, green nanoparticles from the medicinal plant, which have a green impact to your health in your pharmaceutical industry and the nutraceutical industry. So these are the very common steps for the student. You do these type of small experiment in your BS laboratory as well as the MS lab to develop some nanoparticles for your use, for your community use from these medicinal plants. Everyone know about the hemp, that is a bhang. Bhang is also used by some other people by the drugs, but bhang is one of the important medicinal plant in Pakistan, which have anti-AIDS activities. So the people working in the European countries, they develop the drugs from this hemp plant for use for the AIDS. It is also used for the liver cancer. It is also one of the important plants that is used for the tuberculosis. You think about the natural drug discovery from this, your local medicinal plant for the treatment of number of the diseases. So the natural drug discovery development then leads toward the chemical characterization. You are familiar about these sophisticated techniques, NMR, EDX, SRDs. So these are the basically techniques that are every student use to characterize these natural active ingredients from the medicinal plants. So these are the few, just I added these few slides for you, student. You involve these sophisticated techniques during your experiments in order to make your study more reliable, more presentable, more acceptable to the world. So these are the X-ray differentiations, energy, scanning electron microscopy, FTIR. These are the common techniques that every chemist uses. But the biologist and the biochemist must use these techniques in order to make your more study reliable according to the science which is needed. And you also involve the mathematicians and the statistician in your research. When you are doing an experiment, you must optimize your data by involving the mathematical modeling. In 2023, one of the Brazilian group published a paper in Nature. That paper was published about the DNA sequencing by the mathematicians. The work which is done by the biochemist, but it was transformed by the mathematicians. So it is a time to work with a joint venture of the mathematician, biochemist to presentable your data in the world. So the just paper published last night, is carotenoids they are cure for a number of the diseases? Yes. In Pakistan, there are a large number of medicinal plants that are the natural source of carotenoids. So this group, we have published a study about in last week in a bioresource technology, that carotenoids are the natural compounds that they combat a number of the diseases. In Punjab, especially, there's a great range of the salt range. So salt range area is a very neglected area with respect to the natural compound from the medicinal plant and number of the drugs. So you focus about your area, whatever nature gifted you, just think, work and produce something for your community. This is a just study published the nutraceutical potential of the fodder grasses to improve the livestock industry. So just only not focus on the medicinal plant they are used for the human being. Medicinal plant, natural compounds, they are also used for the veterinary diseases as well. So the superfood honey. So the people, especially the biochemists, they quantify and qualify the original honey. Honey is superfood. Another group is working on the production of the value-added product from the original organic honey by involving these biochemical and biotechnological techniques. So this is the common local industry. You think about this for the superfood. So significance of the bee flora, the ladies, they play a very important role in this field because in my research group, number of the females, there were marvelous studies about these superfoods, especially the superfood they are used for your personal use, they are used for the making the cosmeceutical product from these so these are the few glimpses from the local medicinal plant that the honeybee visited these things. So these are the chemical and biochemical characterization of the honey to identify the authenticity or the original superfood from honey. So these are just few glimpses. We have published a study with a mechanical and chemical engineer. How the biologists they link their work to the chemical engineers. Membrane reactor technologies is used for the production of these superfood from local raw material. So this is a joint venture, just example for you people. You think to work with a multidisciplinary science with your related science. So we have also started a work with the Turkish government to develop some super drugs for your food. They have no side effect, just like synthetic drugs have some number of side effects. But when you get a drugs from the plants, animals, minerals with known uses, they have a lesser side effect. Also, we are working about the European Union to develop such natural drugs from these medicinal plants. So this is just my citation, not a my citation, my group citation, where we are working, we have published, because every study should be acceptable when it's original, presentable, acceptable, and publishable. So in last year, government of Pakistan awarded me Tamga Imtiaz, and I become a fellow Pakistan Academy of Sciences. Nature wants love and care. So care the nature, love the nature, respect the nature. Nature gives you food, health, medicine, everything. Nature gives you everything if you care and respect your nature. Thank you so much for your attention.
Thank you, Professor Mushtaq, for a very impressive and beautiful presentation. So we will be having question answer session when the next session start. So from these two presentations, there will be a question answer session. And of course, we will be talking about bioequivalence as well as plant as a medicine. With a very short comment, in US, food as medicine has been approved. Like there was a special waiver that US government has given. Now doctor can prescribe food for the people who are suffering from disease. This is a wonderful initiative. And my last is to the students. When you come back, we will be asking you question also that how good listener you are. So there is a one paper that is ranked #1 in the Google. That is like we recently have gone through these viral infection. Type on the Google antiviral and plants. See which paper come in, and you be proud that paper has been published. Two of the people, they are graduate of this university. Chinese has been just asking again and again why these people come on #1. So I will be asking you the name, who those two people are. Okay? Thank you very much.
Okay, so from here onwards, I would like to call Professor Amir Jamil to take the lead and further proceed the evening. This event, sorry for that, because we are about to do the CESCO proceedings here. So over to you, sir.
Thank you very much. We had wonderful 3 talks, and the question raised by yesterday at dinner by the Alicia, I hope that what is the reason that we are not developing any sorts of medicines, or are we developing any medicines also? And also Lucas was interested in that, and I hope we will have more answers from both of these scientists, especially bioequivalence and the botanicals both. So due to just shortage of time, so we couldn't have the question answer session as Professor Mukhtar has told, but in the next session, first we will have question answer session. So you can, I hope, I also have quite a good questions for the bioequivalence as well, because this is a very important area that we are spending so much money and so much research is going on, but it's all in the shelves, in the theses and all that and publications. Why we are not translating? So one answer we may get from the bioequivalence. So thank you very much, all the 3 speakers. So now coming to the second part is our alumni, CESCOB alumni. So for this, I request Honorable Professor Dr. Khalid Khan Stara Imtiaz, Pattern in Chief of CESCOB, to please come on the stage. No, no, please.
Okay.
नहीं आप बैठिए. One of our senior alumnus is also here. I request Dr. Zahid Mahmood to please come on the stage. He is also one of the ex-president of SESCO in the earlier part. SESCO, SESCO is Scientific, Educational and Social Council of Biochemists. Those who do not know about it, we initiated it in 1990 and rather 89. So when I was MSc student and Dr. Javed was with me, Professor Khalid Mahmood Khan Saab, he was our patron. He instigated and he encouraged us to start with something like society. So we as enthusiastic students, we just followed the path and we initiated this SESCO. And that went very well. Like we used to have lots of activities, even at MSc and MPhil students. We used to have international seminars. We used to have different cultural events, sports activities and all that. And that our seniors like Professor Taira Iqbal and Dr. Mukhtar and Dr. Zaida, she's now in US. She was his first advisor. So we continued this path a few years, but and lots of people they are coming in and going. But after about 10 years or so, it went a little bit in hibernation and other societies emerged. As Professor Mukhtar has told us that there were only a few number of students at that time, but now with the passage of time, we have a good number of students. BS has been initiated. So we revived this SESCO a couple of years ago when I got this again the opportunity to become chairman of the department. So it was a biochemical society that was going on. That was working well. But with the SESCO, we have a bigger dimension. So we want to go like different international, national chapters, and our more of it like alumni. They come interact with us to have more sorts of collaborations, internships for students, their new openings, scholarships for the students, and a lot of that. So to begin with, we of course have the SESCO elections, and in our any of the final annual function, we have the oath taking ceremony of that. So we will, although there is a little bit we are behind the time, but we will have an oath taking of our newly elected members of the SESCO. So we have a SESCO cabinet, president, vice president, and all that, and then we have the councillors. So owing to the number of students, there are like more than about 700 students in the department. So we have a lots of more interaction that is involved. So we have a big councillor section and we have a cabinet. So I ask all the councillors and cabinets to please come on the stage with your oaths, and I will request Professor Khalid M. Khan to please take oath from you. भाई साहब का oath किधर है? यहाँ पे खड़े होंगे, इधर खड़े इधर है वो सर आप नहीं बल्कि हमारा बनाया हुआ है बिल्कुल सर कॉन्स्टिट्यूशन है जी सर है उसी के मुताबिक हम चल रहे हैं बल्कि आपसे किसी को मैं शेयर भी करता हूँ
उसको देखें अगर कुछ चेंज करने वाली है
ठीक है ठीक है ये आप सबने देखा होगा
आप मेरे साथ पढ़िएगा मैं उसके बाद बैसियत फिर अपना नाम बोलिएगा अपना अपना नाम बोलिएगा उसके बाद अगले अगले अल्फाज जो है उनको दोहराइए अलफनामा बिस्मिल्लाहिर्रहमानिर्रहीम मैं अपना नाम लीजिए बैसियत साइंटिफिक एजुकेशन एंड सोशल काउंसिल ऑफ बायोकेमिस्ट अल्लाह ताला को हाजिर नाज़ जानकर ये हलफ उठाती हूँ कि मैं काउंसिल के आईन और दस्तूर की वफादार रहूंगा और तंजीम की खुशहाली और तरक्की के लिए कोशा रहूंगा अल्लाह पाक मुझे इस अहद को पूरा करने की तौफीक अता फरमाए आमीन ये ले
फोटोग्राफ जी सर ये हम साइन फिर बात नहीं करें Okay.
फातिमा का मैसेज है कि एक तो सेकंड टेक्निकल सेशन के अनाउंस करने की उसकी अटेंडेंस अलग से होगी, सर्टिफिकेट उसकी बेस पे होगी,
ठीक
और
है।
दूसरा पोस्टर वैल्यूएशन भी लंच ब्रेक के बाद होगी।
ठीक है। Okay, so we move on. This all the events that are being organized by the SESCO, of course, there is an enthusiastic participation of different teams. But especially during the last year, we deliver like certificates and other awards to our students off and on. But for this particular event, there are two persons which worked diligently and very hard work during the last 1 year. So we want to present them a shield of honor for two of these fellows. Everyone worked very well, but these two worked extraordinary. So there is a message for other students as well. So you work extraordinary, and of course, we deliver something like that to you. So I once again request Professor Khalid Saab to please come on the stage to deliver the shield to our participants. बल्कि सर आप लोग भी साथ ही आ जाएं अगर बेहतर नहीं, अभी काफी सारी शील्ड हैं तो आगे ही आ जाएं सब। जी अब आ जाते हैं। जी आप भी आ जाए इधर। This symposium actually was initiated by two of very active after SESCO members. One is Arham. He was with he did the internship with Alicia in Poland, and another one was Sufyan. But for this particular moment, we want to acknowledge the work done by Sufyan, very enthusiastic work during whole of the year. So I welcome Mr. Sufyan to get his shield. And Sufyan, mashallah, is so energetic. He's so energetic. ये मेरे पास इतनी चीजें लेके आता है, मैं हमसे पूरी नहीं होती. He is very wonderful. I acknowledge that. And the second person is who worked so hard during the whole of the year is the ex-president of SESCO, Mr. Haseeb. He was last year's president. बारिश हो, गर्मी हो, सर्दी हो, जो मर्जी, he is always working over there. And the motivation behind these SESCO is the Dr. Fatma, advisor of the SESCO. As I mentioned, we will be having alumni board for during this, so we can initiate work. We get some suggestions. We get some suggestions from members. I got suggestions, and this is the first alumni board who will work for betterment of our biochemists, and we can add more members later on the suggestion of our alumni. So for this first board, I got these suggestions by myself from different fellow biochemists, and I invite one by one. First of all, Dr. Abda Raza. So she will be our member alumni advisory board. Currently, she is working as professor of biochemistry in the University of Arid Agriculture. We have a very enthusiastic, energetic, and instigating type of, you can say, biochemist and SESCO member who always urge us to do such many things. Now I request Dr. Amjad Hamid from NIAP to please come on the stage as a member alumni board. Amjad Hamid. Next is Dr. Imran Amin. He is also very active, but today he has he couldn't make it yet, so due to some incidents. Our next is Dr. Yasser Saleem from PCSAR. And the person who gave this logo, Dr. Hafiz Dr. Muhammad Javed Iqbal, the pioneer. He is the pioneer member of the SESCO, founder member of the SESCO, and he always, जैसे डॉक्टर साहब ने कहा था कि the catalyst behind it. So he has always been working like that. Our next member, he couldn't make this today because of some urgent assignment. Mr. Abdul Halim Asghar, he is in Islamabad, Comstech. So we will deliver his shield over there. But he has always been very active with us. And our next alumni board member is Professor Dr. Mukhtar Ahmed. Please take your seat, Dr. Mukhtar sir. Now I request Professor Mukhtar. These are okay. And our next alumni board member is Dr. Simon.
दूसरी है चले वो दे दो
Okay.
नहीं वो पहले हम दे चुके हैं अब कोई दे सकता ये आगे से वापस भेज रहे हैं बाद आगे जब नीचे जाएंगे
Now I request Professor Mukhtar to present a shield to Professor Dr. Khalid M. Khan as pattern in chief of the SESCO.
We do need a big round of applause, students.
फाइल मेरी पड़ी है
Guests in order, anybody want to come over here? There are a couple of park holding and stuff. Okay.
Dr. Javed साहब खड़े तो नजर नहीं where is Johnny? Johnny, where are you, Johnny?
Now I request Professor Khalid Khan, Secretary in Chief, for his final comments for this session. के बाद की टॉक्स हैं शाम को हमारी गैदरिंग है ये शाम को कंफर्म किया हमने कुछ और करना से
Bismillahirrahmanirrahim. Honorable guests from abroad and fellows, Dr. Mukhtar Sahib, Dr. Tahra Iqbal Sahib, Dr. Jamil और बहुत सारे जो दोस्त हमारे मुझे चेहरे नजर आ रहे हैं पुराने इतने पुराने भी नहीं मुझसे पुराने तो नहीं है लेकिन बहुत पुराने चेहरे नजर आ रहे हैं आज बड़ी खुशी की बात है I'm sorry if I'm talking in the local language.
I understand.
यहाँ आकर हमेशा always it's a pleasure to come here to be amongst with the people of the Department of Biochemistry and Faculty of Sciences, where I've spent my best years of career, my youth here in this university. I'm always at their service. Whenever they ask me to come, I always try that I should be there. And it's a very happy occasion also that you have arranged such a grand function of a symposium, international symposium, which is of great importance of drug discovery and development. The world is coming back to the natural health cures, and it is impertinent that Pakistan should also have utilize its potential on the potential for the drug developments from the local medicinal plants and traditional drugs methods should be used rather than the drugs made by the synthetic methods. Anyhow, talking about the SESCOP, Society for Social Scientists and Social educational activities of biochemists. This was established in 1989-90 when we were here, when I was here as a professor. And then it was very active, as Dr. Amar Jamil has told us. Amar Jamil is the probably, along with the others, have been the founder members of the society. And he has been since then consistently in the department. Others have gone somewhere else to the other departments, to the other education institutions. But Amar Jamil has been here always. And but with time, as he has mentioned, it became little dormant. And when I left and the other people left, it became little dormant. But now again, it has become very active. And I'm pleased that the society is coming back for the benefit of the students, for the benefits of its fellows, for the benefit of its members. When we started this, there were very few students in the department, and alumni was also very little. Now, I suppose we have expanded so much. Our alumni has increased number, lot of number, and they should all become the members of SESCO. They are all the members of the SESCO. But I hope that last time when we met, we suggested that we'll keep a database of our alumni. I hope that there is some progress in this direction, that we have developed a database and we have a record of all our alumni so that we can come into touch with them whenever we need.
Yes.
Is that so?
Yes, sir. We have this online number around directory.
Well, I'm much meeting for me that online we have developed this, and I think it's a very good development. Also, any society in Pakistan has to be a registered society. This was also mentioned last time, but I hope that you have not. You speak there say, but you have to act registered as Societies Act, under the Societies Act, to get recognition, financial aid, and other benefits. You have to be registered society. Therefore, I will suggest that we move in this direction as soon as possible and become a registered society so that we can claim our share of benefits from the our organizations, national organizations, also international organizations like Science Foundation, Pakistan Academy of Sciences, Higher Education Commission, and so on. So they are all supporting societies, but only registered society. So I hope that we move in this direction quickly so that we can benefit others. I also hope that as Dr. Amar Jamil has mentioned, that we have been very active in the last year. Next year, there will be more activities in this direction. And there will be more meetings, more interaction between the SESCO members and the alumni, so that you see, this university has given us many things. And when we graduate, when we go out of the university, it's time for us to give back something to the university. So, and this is the way that we get together to get back and do something for the university. I was just reading here, Ruth something, her name, has donated $1 billion to the Albert Einstein Medical College. She was an alumni of the college. I don't expect Pakistani to donate so much, but now maybe not in cash, but in kind. we can do something for the university, or do something for the department, do something for our society. So I hope that when we get together, we'll think on this line also. I thank you because it's time for prayers coming up. We have to go back. I thank you all and hope that when we meet next time, we will come up with the much more material that we have done something more in this for this side. I thank you all.
Thank you so much, sir. It's such a great honor to have you between us. Okay, now we are going to observe a break. But here's an announcement. Actually, there are two important announcements before going to break. That now we are going for the prayer break. It is about to start at right after 1 minute till 1:45. And all of you are instructed to get back at 1:45 in the ground right in front of the library and the VC building. So we will have lunch there. And and after that, we are having another technical session. Poster evaluation will also be done at that time. And the second technical session will have its separate attendance. And the certificates which you are going to get after this whole symposium thing are going to be distributed on the basis of your all attendances, like of the first session, of the second session, and of tomorrow's day. So now I request all our worthy guests that they can leave for the lunch and for the break.
The question, basic question is the new drug approval. There are two types of drug, just I have informed. A new drug approval and the abbreviated new drug approval, that is generic. In foreign countries, like in USA, in Europe, and in all other countries, the clinical trial cost or bioequivalence trial cost is very high. In Malaysia, in Jordan, in Philippines, the cost of single bioequivalence study is about 6 million up to 10 million rupees, Pak rupees. I'm just talking about the Pak rupees. Some of the big guns in pharmaceutical industry like Getz, Pharmatech, and Sami, they do bioequivalence studies because they want to export their drugs or they want to participate in the WHO tenders. In recent last 2 weeks back, we have a conversation with Cape Town University because one of the bioequivalence was being failed by the WHO because of the high coefficient of variance between the inter-subject samples. This is not easy to say that every bioequivalence study will be approved until and unless you worked very well in vitro. Yes, it is possible that if we start the work in Pakistan on bioavailability and we, inshallah, we are going to recognize our center with WHO and FDA USA. Once it got certified, the cost of bioequivalence will reduce to less than half. Here, the remuneration for the subjects, the cost of research is very much less than the Europe, but we have achieved the standards as per US FDA. So Pakistan, when you start, at the moment in Pakistan, only generic drugs are developing and certified by the GRAP. They got the license to manufacture and to the market. Not a single drug as an innovator drug. It says an innovator drug has not been till introduced from Pakistan. And by adopting the bioequivalence and clinical trials method in Pakistan, we can, inshallah, we can start the innovative drugs from Pakistan.
Jirak sir, thank you. Actually, it's not a question, it is a comment. I think nowadays the technology is totally changed. The world is moving to artificial intelligence, AI. I think maybe in future we can save a lot of time because you mentioned that drugs took a lot of time, even many years. So I think now this is a artificial intelligence is very fast with everything. So I just have a comment that just think about like this one also.
We have worked on it already, and research will also be done on AI in comparison of clinical trials and bioavailability. The generic area is totally different from area to area, but even then the response may be different because AI doesn't calculate the bioresponse from within the body and the impurities that have been developed due to the degradation of the product. There's only not a single factor. You can predict with AI, but what will be the biochemical reaction can take place with which impurity happens? Because each impurity has its own identity. It's in a trace amount. The same drug, when passes, for example, if I'm importing a raw material, active raw material from outside country, the way it is imported, either it was been managed, the temperature controls, पता लगा कि ये वो तो 2 दिन एयरपोर्ट के ऊपर ही ना धूप में पड़ी रही है. Nobody knows. So the impurities produced within that period cannot be detected by AI or the computational methods that what will happen with these impurities. So the bioavailability needs healthy volunteers and real-time testing on the healthy volunteers.
So one last question.
Sir, my question is that the professor is already asked AI is used in a drug discovery and development. My question is that if we genetically coded impurities all automatically removed, and we can also predict like how far it goes. For example, now AI is using in a diagnostic purposes. So we can scan the whole body and then we recognize after 10 to 20 years what will be the disease and before that the particular disease. So can we use AI? So because
just tell me one thing. I would like to ask one thing. In our genome, in our era, we administer simple paracetamol all the peoples for headache. But some people got allergic of that. And nobody knows what might be the reason to get the allergic for any chemical. Well, then what will you do?
my answer is that, sir, when we
I think there is some communication problem. So we can have now, this is more discussion we can later on. So with this, thank you very much, Dr.
Maybe one comment.
Okay.
Professor, maybe.
Then very interesting presentation and generics are, I think, very important things for the world. Maybe I share with you some experiences from Poland about the generic drugs because the largest Polish pharmaceutical company produces mainly, if not only, generic drugs and export it to the 35 countries, Polish government promotes companies which introduce generic to the market. We have regulations, we have benefits for this company which introduce generics to the market. And last, even pharmacists, every patient during the service in the general pharmacy, pharmacist must recommend cheaper generic, of course, if it's available. And this causes that the medical expenses of Polish patients are decreasing. But I agree with Dr. Muhammad Ali that safety is the most important thing. Not only expenses, decreasing the expenses, but safety. And I think that biobank test, not only artificial intelligence, of course, artificial intelligence will be very helpful, but I think that even for 10, 12, 50 years, we will have to check it in vivo or in human body, like
I just want to add one thing that lab on a chip can also reduce the cost of clinical trials. And one thing is that in Pakistan, I started to work with academia and industry linkages to build something and then try to develop the contrast agents. Because this is the item that is being, because of course, Pakistan. Coming back to the previous question by our student that हम लोग क्यों नहीं ये import मतलब हम कर लेते हैं तो क्यों हमारी जो cost जो है ना बहुत महंगा पे पड़ती है तो इसलिए हम import कर लेते that should be the solution. I just started working with the contrast agents. Just an example of the barium salt, and barium salt is only cost like 10 rupees. But when you talk about that. When in the Corona times, all we import this item from the, we export from India, and India is like, then there was no, trade between these two countries, and there was a shortage of contrast agents in the country, and all the CD and MRI and these agents were not available, and that was the a very with also many other drugs also. So that is the thing that at least if you are not going to work on drug discoveries, you are not going to develop the new drugs, at least we can work on the available drugs that are that doesn't require so much infrastructures or like this, but of course the GMP facilities or other things that are the requirement, that is the regulatory requirement. So I visited many industrial areas in our in British side also the Rawat industrial area and other things, and they were very much like happy. Okay, we can, you just give us the protocols. I just realized why we are unable to deliver our products to the industry. That we always try to think on the that what we are going to work do some job on the on bench, and all the protocols that we optimize that we state in our thesis or in our publications, that is of no use of the industrial persons because they need some protocol that are really like the commercial levels. They need some, the steps are 1, 2, 3, like simple steps. They should not be like the instrument that are जो कि आपको बहुत उस पे available ही ना कर सके. So there were many issues that we have to look into that how this the research that has been conducted in the university can be just transferred to the industry, and for that matter industry should take on board what the industrial partner really needs that. And this is not a solution that कि हम हर चीज जो हम बाहर से ले लेंगे. Of course we are not going to talk about all the like a very paclitaxel or doxor, obviously, like so much cancer drugs, but even then the smaller simple, like I have given you example of barium salt. So it is like the simple things that we can just do. So पाकिस्तान को चाहिए कि नॉट सो थिंकिंग कि बहुत बड़ी बड़ी चीजें हम सोचे, हम कभी छोटी चीज भी नहीं कर पा रहे, तो छोटी चीजों पे हम स्टूडेंट्स भी अपनी इस पे सोचे कि they can also.
Sorry,
कंप्लीट कीजिए मैं आपको एक बात पे कमेंट देना
So
चाहता हूं।
this is the thing that अभी मेरे जो स्टूडेंट्स हैं उनको मैंने इसी पे ही कहा कि भाई अब कुछ सोचें, कुछ इस तरह से आप प्रोडक्ट्स को बनाएं, cosmeceuticals, nutraceuticals, and then many other areas you can just identify, and simple products that also needs many, like आप pharmaceutical पे भी मैं मत जाएं, pharmaceutical you know it's like बहुत ज्यादा जिसमें कि आपको जो इंटरनल यूज के लिए उसके लिए आप एक्सटर्नल यूज पे आ सकते हैं, फार्मास्यूटिकल में भी अपॉइंटमेंट्स हैं, उधर की चीजें हैं। सो देयर आर अदर एरियाज जो कि आपके लिए बहुत इजी है और उस पर इतनी इन्वेस्टमेंट भी नहीं है, और उसके अंदर आप इस तरह से प्रोटोकॉल्स को डेवलप करें जो कि आपको फिर ये जो इतनी जो आपको पाकिस्तान के ऊपर जो थोड़ा सा बर्डन है उसको कम कर सकते हैं।
Okay, thank you जी, लास्ट कमेंट।
एक लास्ट कमेंट दूंगा बिकॉज इंडस्ट्री में मैं बहुत अर्सा रहा, मेरे सारे मोहतरम दोस्त यहाँ पे मौजूद हैं, रिसर्चर्स मौजूद हैं। इन 15 years ऑफ माई इंडस्ट्रियल लाइफ, टीम में मैं GM रहा, जनरल मैनेजर रहा, कोई एक रिसर्चर हमारे पास नहीं आया ये पूछने कि अगर आपकी टैबलेट की डिसइंटीग्रेशन नहीं हो रही तो क्या हम इसके ऊपर आपको काम करके दें, हम अपना एक एमफिल स्टूडेंट आपके साथ अटैच कर दें और वो इसके ऊपर काम करेगा और उसका बकायदा सोल्यूशन आपको निकाल के देगा। हम ये करते हैं कि अपने हिसाब से टॉपिक ढूंढा, किया, थीसिस लिखा, पब्लिकेशन की, दैट्स ऑल। प्लीज गो टू द इंडस्ट्री एंड आस्क देम व्हाट दे वांट, फिर आपके पास सॉल्यूशन सामने आएंगे। Thanks.
Okay, Dr. Sahab, thank you very much. It's a long debate and long talk, and thank you very much. It was a good, thought-provoking lecture and talk. But it is a continuous debate between industry, academia, the problem solutions. So, Anji, next continue करेंगे मेरे ख्याल से।
So our second technical session will be chaired by Dr. Lucas and Dr. Abel. Well, now I would like to call Dr. Lucas, Professor Dr. Lucas, to please come here on the rostrum and first of all introduce himself, and then later on he will address to you.
Thank you. It's a great pleasure and honor to stand here and give you speech. So excellent audience, so excellent speakers before. My name is Łukasz Fiałkowski. I'm from Poland, from Bydgoszcz. Is a city around 400,000 people population, and work in the Department of Pharmacometrics and Molecular Modeling in the Nicolaus Copernicus University. Something about me, I'm a pharmacist, I'm a practitioner, I'm owner of 3 pharmacies. Then I try to connect work in business and work in the science. I have 115 publications and conference speeches. My impact factor is 66. In the field of pharmacy, health science, in cooperation with the students, with the PhD students and other scientists, I conduct the molecular modeling experiments, including interactions of the ligands with receptors and proteins. I'm also a member of the crystal engineering and advanced solid state characterization research group. One more thing, I love to teach students. I think that is the reason that I work in the university, and I'm very glad that I see so audience of the students, and maybe of the records. I encourage you to encourage students to make research even during the studies, and maybe I show something like that. I don't know if the Einstein did say it, but my favorite quote is that if you can explain it simply, you don't understand it well. Then I try to show you molecular modeling in the simple way, and believe me, Of course, the advanced methods you have to do in the calculation laboratory, but the simple methods you can do it yourself in home. First thing, something about Poland, my home country. Poland is the country in the Central Europe, the Kujawsko-Pomorskie Voivodeship, where is our university, is the center of Poland. And our university, I can say it's special because it's settled in the two towns. 40 kilometers each other is the length between the cities, as you can observe. Is the very nice landscape. Is a mix of woods, steppes, fields. Then when I see you fields, I feel a little like in home. And in Bydgoszcz is medical campus, and in Toruń is there are other faculties. And I want to show you two symbols of the Bydgoszcz and Toruń. Bydgoszcz symbol is archer, and Toruń, where Nicolaus Copernicus were born, is the known, I think, on the world. But not only geography, but we go to the chemistry. First of all, as a member of Pharmaceutical Society, I'm very thankful for all biochemists, because I think that when you want to be good pharmacist, when you want to introduce the drugs into the market, you have to be also good biochemist, or you should know good biochemist. Yeah. Then without connecting the biology and the chemistry, we will not able to introduce the new drugs or even generics. It has to be multidisciplinary. Then something about serotonin, the happiness hormone. Yesterday and today, we have a lot of talks, and I think that in my body, the level of serotonin is very, very high. And I think it's a good condition, because when we have disturbation in the serotonergic transmission, it could develop anxiety, pain, depression, panic disorder, or obsessive compulsive disorders. Then I think that serotonin is very important hormone. When we have lack of serotonin, and we could observe after the onset of the COVID-19, where we were closed, when the travel wasn't possible, we have articles in literature that in 2020, we observed an increase of 27%, and also an additional 53 million cases of major depressive disorders globally. Also, an additional 76 million cases on anxiety disorders globally. It's increased about 25%. Then psychiatric disease was very challenging problem. I think that after COVID-19 pandemic, the importance of the psychiatric disease increased. What about depression? We have a lot of hypotheses of depression. We have monoamine hypothesis, neurogenesis, neurobiology, inflammation, or stress hypothesis. Today, I focus on the monoamine and neurogenesis hypothesis and try to speak something about it. Then, what theory is more probable? Of course, the monoamine theory is very simple. That when we interact with serotoninergic receptors, or when we increase the concentrations of the serotonin in the synapses, we will observe that the signal transmissions will be increased. And one thing about my research about molecular modeling, because it's a challenge, yeah? How to connect real life with the virtual reality, yeah? Because I can show that some molecule interact with the protein, but I don't know it will have the effect, yeah? Or what will be the effect? But, for example, in monoamine transporters, monoamine transporters is like tube. Then, when we block this tube with the physical interaction, when we see that we have affinity to the protein, and when the canal will be blocked, we will be sure that we will have therapeutic effect. Yeah. Then is the reason that in silico methods, in silico molecular docking, in transporters, in biological canals, I can say, is more probable that when we observed affinity in silico method, we also observed the clinical effect. And what about neurogenesis theory? The main compound today will be paroxetine, and after administration of paroxetine, we don't observe the effect after 1 day, several hours. We observe the effect after 2-3 weeks. Even we will see that in synapses we have increased level of the serotonin. Then this therapeutic delay suggests that slow adaptive change in the neural circuits during long-term pharmacological treatment possibly involve change in gene expression and protein translation. And depression is also caused by disruption of functional and structural connections of the neural circuits, and that underlie the regulation of mood. Then neurogenesis theory is worth to develop and to check it well. Biochemistry, chemistry, then I want to show some chemistry of paroxetine. Active substance is enantiomally pure. It's a phenylpiperidine derivative, secondary amine residing in the piperidine ring, which in turn is connected to benzodioxole and fluorophenyl groups. And what is very important and interesting, that each of 3 rings is located in the different plane. So the structure of paroxetine is highly non-planar. It's also lipophilic benzamine with both hydrophobic and hydrophilic moieties. It's also very important to when you compound should permeate the barriers. And paroxetine is slightly soluble in water, sparingly soluble in methyl dichloride and in ethanol, but is entirely soluble in methanol. I think the most important thing is that the chemical properties of paroxetine make it easy to modify and preserve drug-like properties that paroxetine could be good draft to develop new drugs. We have many registered generics of the paroxetine. I show you some preparation from Poland, also from Pakistan. It's very popular drug. When I used to work in the general pharmacy, I said it well and many patients bought it. And maybe we start about paroxetine as SSRI. It's the main mechanism of action of paroxetine. We focus in the three monoamine transporters with serotonin transporter, dopamine transporter, and noradrenaline transporter, and also fourth gamma-aminobutyric acid transporter. But these three first one, when we block this transporter, we can treat many psychiatric disease, and unfortunately, compounds with this activity are used as illicit psychostimulant drugs. What is worth to notice that paroxetine is an selective serotonin reuptake inhibitor that exhibits the highest no-binding affinity for the center site of the serotonin transporter compared to any other currently prescribed antidepressants. And is paroxetine not only active for depression? We made a review and found that paroxetine could have many other activities. For example, activity in the cardiac disease, because paroxetine exhibits 50-60-fold higher selectivity for GRK2 kinase versus other GRK. Paroxetine is the modest inhibitor. Other compounds from the SSRI group have shown no effect in vitro kinesthesia or ex vivo in vivo myocyte contractility. Then it's possible that paroxetine could be used in heart failure treatment in the future. It should be matter of the research. What about activity of paroxetine for the cancer? You know, when someone gets ill, get cancer, depression is very common disease, because the first thing is that, oh God, I will die. Yes. Then depression is very common, and it's observed that in some specific cancers, patients who administered the paroxetine had better clinical effects. What is the cause of it? It was observed that paroxetine could interact with tyrosine kinase, could be tyrosine kinase inhibitor, and could be gynous kinase inhibitor. And we check it, and we could observe, as you see in the picture, that in superimposition, it's quite similar structures, and we have the same element in the three drugs, then paroxetine could be involved in blocking some tumors. Then, okay. What is, maybe I show you only narrow part of my studies, but I want to show how our docking procedures, how our research is conducted. Then main question in our research was, is it possible for paroxetine to act in a neurogenic mechanism? The reason of choose this and try to answer this question was that we have lack of pharmaceutical data on the molecular level of the developing neurogenesis. We checked the interaction between paroxetine and 4 transporters. We used many computational software, which was supported, of course, by in vitro tests. Maybe I talk maybe I speak more about in silico studies. Then we applied docking procedure with the typical for processing of docking studies in this way. We prepared properly ligands in the Autodoc tools software. Sorry. Okay. All the molecules were geometry optimized in water described by the PCM model using the Gaussian software. Subsequently, torsional and number of active torsion for ligands were defined, and the Gestalt chart were assigned to paroxetine. What about proteins? Proteins were, some of them were downloaded from the protein data bank. And for example, serotonin transporter was downloaded from the protein data bank, but dopamine transporter, noradrenaline transporter, and GABA transporter was downloaded from Swiss Pro database. There was homology modeled channels, but with very high level of compliance with the real protein. We also conducted the similarity and superimposition study. And what about result? First, we check the clear, maybe very obvious thing. If paroxetine interacts with serotonin transporter, we compare the crystal with the paroxetine inactive site and our model of docking. And we were not surprised that the interaction of paroxetine in the crystal and interactions in our experiments was very similar. Comparison between calculated and crystallographic data confirmed that we can go further. Then we check the exact structure of dopamine transporter homology model, and we found that energy of binding the ligand is -8.35. It's the affinity of the dopamine, of course, is not so strong like in paroxetine, but still is very high affinity. And we could observe that crucial interactions in the active site were between asparagine acid 476 and other 3 amino acids. What about noradrenaline transporter? Affinity energy was quite high, and this confirmed that we have partial activity for the inhibition of noradrenaline neurouptake. And finally, we checked the GABA transporter, which was not so clear because, as we know, paroxetine is the inhibitor of the GABA transporter, but in our research, we observed that the energy of binding maybe were not so, the affinity was not so high, but still important and could cause the blocking of the channel. Of course, we validate our experiment. We made redocking. All the docking results we checked in other procedure, in other software. This software was Vina. And as you can observe in the picture, the validation process indicates a high similarity of paroxetine corresponding the process obtained using Autodoc and Vine. Then from virtual reality to in vitro, thanks for cooperation with scientists from Denmark, team of Professor Petrine Wallendorf made for us in vitro studies. And as you can see, all experiments showed that paroxetine has ability to inhibit the uptake of the gamma-aminobutyric acid and increase the positive signals in the neural, in the central nervous system. All experiments were performed with technical replicase in 3 independent experiments, and the ISD was in good value. Then I go to the conclusion. Based on our research and calculation, we concluded that drugs used in the pharmacotherapy of disease with etiology resulting from a decreased level of neurotransmission with the participation of basic mediators like serotonin, noradrenaline, dopamine, or GABA may potentially act in the neurogenic mechanisms. Data from molecular studies indicate a potential neurogenic effect of paroxetine, which may be mainly due to the strong inhibition of the human serotonin transporter. We found some structural similarities in the cell carriers and the transporters. Sorry. And I think it is an additional conclusion, but very important, that prospective achievement of this research was the confirmation of some activity of paroxetine towards human gamma-aminobutyric transporter. This enables the development of research based on the modification of the paroxetine structure to increase the affinity for particular step of gamma-aminobutyric transporter, which is well treated, for example, in epilepsy. Great pleasure. Thank you for the attention. Shukriya.
So, many thanks, Professor Likoshi Fukoshi. And now the house is open for discussion. And I think you must all have now enjoyed a pure biochemistry talk by a pharmacist. So it's like if you have any questions, so all the biochemists can ask. It's about happiness, it's about depression, it's about, many, many things. I think it was a very nice talk and too much knowledge. They have just, guessed it after a lunch. So all are like, feeling sleepy.
Less serotonin, more gamma-butyric acid transporter, I think.
Maybe.
Oh,
Dr. Javed.
thank you, Professor.
Professor, your talk was just good and we enjoyed. In fact, we took a heavy meal and we were sleeping a little bit, but we enjoyed. Our body, how decide to produce serotonin and other hormones when we have, we have some severe or climatic changes on our body, on our system. How we got the shutdown and how we start those thinking about the production of that serotonin.
Thank you for this question. One of the lecture before, it was about nature. Yes, about nature, compounds, and blocking of the serotonin uptake. All the serotonin uptake inhibitors you see, we not exogenic compounds interact with the receptor, but we physically block the uptake from the synaptic cleft. It's the reason that we don't put the synaptic exogenic compound and allow nature compounds to more react. Our research doesn't check how to produce more the serotonin because our neurons are able to produce enough amount of the serotonin, I think. But the clinical problem is when too much serotonin is pumped out from the synaptic cleft. Then it's the reason that we should block this pump like I showed in my presentation.
It's such a fascinating presentation from a handsome man, and who is a great pharmacologist, a pharmacist. You are happy because you know everything, okay?
No, I know. I don't know everything. Who know everything is died, I think.
Now, the main thing is, you might have seen this product Neuralink by Elon Musk, like people who are totally crippled, they can run the computer. You might have seen that. So now I think the technology is coming at the level where they can, every time that somebody is creating serotonin and is happy, and like Javed is happy all the time, because he has too much, people who keep him happy around, So, yeah. So, what will happen? Like if there is every time there is a serotonin, every time there is a happiness, what will happen? What will be the world with too much serotonin?
I can say that when you eat too much chocolate, in Poland used to say that chocolate is the industry who produces for us the serotonin, yeah? And it could be addictive. And as that Paracelsus, everything is the poison, is nothing is the poison. It depends from the dose, yeah? Then even serotonin, we need appropriate dose of happiness to live properly, I think.
Thank you very much. Thank you so much, Dr. Sandra. One more question?
Sir, I want to ask, how is peroxytin kills the colon cancer cells? And what is the mechanism behind those?
The mechanism of what? Could you repeat?
How it is fighting with colon cancer?
Colon cancer.
As you have mentioned.
How the serotonin is fighting for the cancer?
For the colon cancer, okay. Then some cancers, like the colon cancer, is related with some specific kinases. I think, like a biochemist, you know better than me. And we could observe that paroxetine has similar structure to sunitinib, for example, and it could block some signal pathways, could develop the carcinogenesis. Yeah. Then, of course, as you see, it's only review. It's not my research. We found it in the literature, and it's good start to maybe make in silico methods. Why in silico methods? It's good, for example, for students, because except one software from it is freeware. You can do it yourself in laptop in home. Then every of us can do introductive studies, and maybe it will be helpful with the finding and developing new drugs. Then this was from the literature, and it was only it not was my study. Yeah, then I only show it's in our publication, but it's in the review. I have this publication. I can show exact position in the literature when it was described.
We can have one comment from, maybe you can have.
Doctor, my question is slightly different from the topic, but I can ask, why sleeping pills remain our blood for more than a week? Why sleeping pills remain our blood for more than a week?
Sleeping pills?
I didn't understand well.
Why sleeping pills
Remain in our blood more than
remain in our blood for more than 1 week? Oh, it's all about the half-life, dear.
Yeah, biopharmacy.
Simple.
Yeah, it depends, and it could be depend from the individual specificity, because everybody could have has other gene experience, gene expression. Sorry, then it can cause that the half-life time will be different in the various persons.
I have some quick comments to protection, because not everybody knows that serotonin is mainly produced in our gastrointestinal tract.
Gastrointestinal tracts, yeah.
It's produced over 80% serotonin in our intestinal tracts. Only 20% serotonin is produced in our brain, and when we eat and have some good taste dinner, we naturally feel some happiness.
Like today.
Like today, that's right. And I think when we have some problems with gastrointestinal tract, we have some depression. It's naturally connect this process in our body. Thank you.
Yeah, it's very much important. very much interesting. You know, I just came to know that, you know, that why, you know, in our saying that the good food, if you make someone happy, just give a good food, especially for the husbands, you know.
Colin would have some sentence when we fall in love, the best recipe is track is track to the
Through the stomach to the lung.
the house, from the stomach to the house, you know. Good recipe.
So today we came to know the scientific reason, you know. Thank you very much. Yeah. Okay, thank you very much. So.
Thank you so much. And now our next speaker is Professor Dr. Muhammad Nadeem Akhtar, who got his PhD in 2004 from Karachi University. Please give a big round of applause. Moreover, he did his postdoc from Germany with Professor Carsten Krohn at the University of Paderborn in 2005. Currently, he is working as a professor at the Ghazi University, Dera Ghazi Khan. His research group is actively involved in the structural elucidation of natural compounds from medicinal plants and the synthesis of bioactive compounds, especially in drug design and development for cancer. The University Putra Malaysia, the international center, he has research collaborations with the University of Malaya, the University Putra Malaysia, the International Center for Chemicals Sciences, HEJ Research Institute of Chemistry, the University of Karachi, Pakistan, and GC University Lahore. He has published more than 102 research papers in the prestigious international ISI journals. He also received Best Publication Award in 2015 from Malaysian government. Please give a big round of applause.
Thank you for your kind introductions, and also I'm very thankful to Professor Dr. Amir giving me opportunity to have over here to present my some recent investigations. So my topics basically purely links the drug discovery and development. So this is the picture of Malaysian medicinal plants, which we are working from the last 1 decade. Very famous plants, which is called durian over here. I think I want to move forward to show you some slides. So today I work on research on this plant, which is called Morinda elliptica, and I will show some more slides. Of course, medicinal plants are the rich source of drug discovery. Very famous drugs which have been discovered from the last centuries, like morphines, very famous drugs, and we have a codeine, we have diazepines, morphines, quinines, and also the Taxol, which is very famous anti-cancer drugs, so that have been discovered from medicinal plants. Actually, we target breast cancer, and it's very common disease in the women. In 2020, there are 2.5 million women diagnosed with breast cancers, and 685,000 deaths globally recorded. At the end of 2024, 8.9 million women are expected to be diagnosed. So and National Cancer Research Report shows that among the cancer deaths, the breast cancer are very common, like about 45,000 have been death recorded in the last years. So every years, if you see that this graph, almost every years about 10,000 people, women are diagnosed as cancers. So these rates I've been that going to be very rapid, and the people have to be think about the seriousness of the breast cancers. Among cancers, other cancers, breast cancer is very high in women, In Pakistan, because we have only the Shaukat Khanum Memorial Hospital, which can detect some of the early stages. And as the record of Shaukat Khanum hospitals shows that 30% of all breast cancers can be diagnosed in the early stage. Mass screening of breast cancer is not possible in Pakistan because of lack of facilities. Even our women cannot reach to the doctor because they are very reluctant to approach to the health professionals. So if you see the some of the symptoms when the women have breast cancers, so they can feel some of thickness in the area, and changing in the breast also seen, and nipple turns inward and also develop sores. Something like inflammation also seen in the breast area, and also the bloody discharge from the nipples. So that can be the see the symptom of the breast cancers. If you see the some causes of the breast cancers, so normally it is known that 40 to 55 years a woman can see that this breast cancer symptom because of the lack of and disturbance of estrogen hormones. Lifestyle can also cause the also cause the breast cancer, like alcohol, not feeding milk properly, high fat diet, physically inactive, smoking can also cause the breast cancers. So prolonged medical treatment like chemotherapy, radiations, aggressive cancer treatment, hormone replacement, also the one of the cause. If medical history, immunosuppressive drugs, and long-term use of oral contraceptive also cause the breast cancer. History if you have a history of the in your family, can also be the one of the cause of the breast cancers. So now I want to come to my research because we have been get the compounds from the medicinal plants, and after that we can make the drugs in our laboratory. We can synthesize in our laboratory, and then we can test some more details. Because this plant is very Malaysian medicine plant, and it's called Morinda lactica. And this plants already we published some of early results that this plant shows the some of activity against MCFC, which is called breast cancer cell lines, and also reports some activity against HIV. And this the extract of this plant also shows the immunomodulatory activity. So today I will we get two compounds, which is called dominicanthals and also the nordominicanthals. So this one basically we get the active compounds from this medicinal plants, and we plan to synthesize this one. So for the synthesis, these compounds we design in our laboratory that this compound one of compounds is called dominicanthal can be synthesized from phthalic anhydrides and also the toluene derivative. So we start synthesis because these compounds are commercially available. So we can get these compounds. So we start synthesis from phthalic anhydrides and also the toluene derivatives. And then when we treat this compound in molten state in a reaction flask, so we get the this anthraquinone derivative. So this was the key step in our synthesis of these drugs, and we get a good yield of also about 80% yields. The mechanism of this reaction is very famous reaction, which is called Friedel-Crafts alkylations. After that, we compound number 3 was treated with the methyl iodide and potassium carbonates. So we obtain the 2-methyl-1,3-dimethoxy anthraquinone derivative. So methylations, we get these compounds, and the yield was 84% of this derivative. After this, we do N-bromosuccinamides. we got NBS like N-bromosuccinamides. So we get the bromination of compound number 5. So at this stage, we obtain several products, like we get dibromo, we get C brominations. So at this stage, we get several products, which is not our desire that we should have a good yield also. So next time we use this NBS very slow portion wise, very slow addition of this NBS, and we got 90% yields. In this way, we enhance the yield of these drugs and we get 90% of this product. After this ones, we make one more modifications. We use the dry tetrachloroform, and we get 90% yield. And the structure of this product was confirmed by single X-ray crystallography. After this one, this compound number 5 treated with the 80% acetic acids, and we got compound number 6, which is called the alcohol product, and the yield of this product was 78%. So that was also the excellent yield. Further, we treated this compound with the PCC, which is called pyridium chlorochromate, and we get acidic product, but that was not our desired product. Then we treated with the PCC in a very low quantity and add slowly. Then we get compound numbers aldehydes, about 92% yields. So finally, the compound 2 was synthesized, which is our desired product. That was demethylations. And demethylation was performed by the aluminum chlorides by using in dichloromethanes, and we get the dihydroxy product. And this was a very important step in which we do acetylations by using the acetic anhydrides and potassium carbonate in acetone reflexes. And then we get the monoacetyl product that is called compound number 8, which is called 58.7% yield. Again, we methylations take place on the OH group, that is called the monomethylations. And then we treat compound with NBS like chloroforms, carbon tetrachlorides, and then we get the bromination product. After that, we have two-step reactions. In the first steps, we do the like so-called with the base, and also we use in the ethanols. We get the acetylated product. Acetylated product. Ester product. After that, second step, we remove the deacetylation by using the SCLs. So finally, we obtain our compound number 1, which is our desired products, which is known as a good anti-cancer activity. We treat with this one by 80% acetic acids, and then we treat it with the PCC, and we get our product like 64.5%. So this was the first total synthesis of these drugs, and we published this one in molecular journals, which that was. So we test all our compounds, and we observe that a compound which is like a 5, and it has like good yields. I mean the good activity, like IC50 value is 5.7, and also the 8.5, which is a very low IC50 values. That was the second highest active compound against the cancer cell lines like MCPF7. So this one, we found that if it is active, we take one more bromo compounds, and we publish one more product like this ones, which means that bromo derivative shows the potential activity against the breast cancer cell line like MCPF and MDPA, MBP231s. So we make many derivatives like circulant derivatives, and then we make SCR, which means that every some like methoxy shows the activity, and also the if we have aldehyde product, aldehyde like this one, compound 13A also shows the activity, good activity. So another area, and we published several papers on this derivative because this shows a very good activity and also the good class of natural products. So another area which we discover some very lead compound, it's called curcumin, because हल्दी जो है वो आपके ब्रेस्ट कैंसर के लिए बहुत ही अच्छा एक वो क्या कहते हैं which we should say that is very potential because if someone have early symptoms in their bodies, so they can use the curcumin, which is called haldi, can use for the treatment of breast cancers. Early symptoms, that's just by discovery also. So apart from the curcuma langa, we observed that there's three main compounds in which is called haldi, that is contain three main compounds like curcumins, deep toxicurcumin, and also the bis deep toxicurcumin. So if we see from the last years, about 10 to 15,000 papers published on single curcumin. How much the active compound? Because curcumin, if you see the literature review from the SciFinder, every year almost 10 to 15 papers published. But if you see, it shows so many activity like it can be used like health bounds, respiratory, anti-inflammatory activity, antioxidant activity, anti-cancer, wound healing, so many activities. This ones. But even these compounds shows many activity, but in some of the report also shows that these compounds have poor absorptions and also the rapid metabolisms, low level serums, and limited tissue distributions. So what we have planned, we make some modification. Why this inactive compound? There's some of issues because it have enol form. When it has enol form, it has some solubility issues. So we make some modification. We remove this diketone moiety and we use single monoketone product. That's why, because we want to enhance its activity and we want to make some more better result as compared to the curcumin itself. So we make modification structure like this one. We use acetone, we use cyclohexanone, we use cyclopentanone for making more analogs. So we observe that we make about 10 derivatives of cyclohexanones, then we make some more series like acetone series. We make more about 10 compound like this one, and also we make more by using the cyclopentanone, so that we can check which one is more active, which one is how we can overcome this problem. So we test this compound against different cell lines like HeLa cell lines, KM562, MCF7, MDPA, MDP, which is linked to the breast cancer cell lines. And we check the, we tested this compound and we observed the results. Like here, you can see the results. But we found that which compound have methoxy like 2,6,9 methoxy, and also the like we can see the compound number 5, which has a similar structure as the curcumin. Because the compound 5, you can see that it has the same structure as curcumin except the diketone. So this IC50 value is much better than the curcumin itself. You see the IC50 value of this compound like 6.75 and 2.3 and 1.65, which is very low microgram per ml. So this was much better activity than the curcumin. And one more thing is that because fluoro, as mentioned the prof, the fluoro compound also have a good activity like very low IC50 value about 3.02 and 1.3. So this meant apart from the curcumin itself, if we add some bromo derivative, if we had some fluoro activity, it shows also the good activity. So in monoketone series like acetone series, we see the same case because the substituent something is also very important to enhance the biological studies. Again, we see the fluoro and then also the same methoxy, rhodoxy compounds, very potent as compared to the curcumin itself. Same as we can see the cyclopentanone series, we see that if the structure makes similar with the curcumin, we have a good activity. The structure of some of compound which we observe single assay, which help us to make some docking study also, and we done also the docking study as we see against kinase ones. So I'm not much So finally, the most potent compound was like if you have a 2-methoxy and 4-methoxy as very potentials, and monomethoxy also very potential. And finally, the compound C, the half structure like curcumin is very potential for the against breast cancer cell lines. So we publish this all the compounds in the journals like which have a central journal chemistry, which have a good about 4 point something impact factors. So finally, I just want to show you a very interesting example. So finally, we make more modification. We make more simplify the structures. We remove the double bond hydrophobic port, and then because docking study shows that diketone is very important part if there is no enolizations. If we remove enolizations, then we can see the more activity. We synthesize very simple structures, but this one is also very active against several cell lines. We study it in like we do a study in different in vivo study. We done in vivo study and we found that this compound is much better result as compared to the other derivatives because of simplicity. And we published more than 10 papers about this studies like DK1s. This is just few example. We observe and we test anti-metastatic effect, anti-angiogenic effects of curcumins, then DK1, and we compare in vivo study that this compound is much better than the ketone DK1, which means that the nature like simplicity. Simplicity is also one of the factors that also good for to enhance the activity. So I'm very thankful to my Malaysian collaboration research like Dr. Noor Jahan's, Dr. Sui Yong Yep, Dr. Nadia from Ministry of Malaysia. And thank you very much for your kind attention.
Many thanks, Professor Dr. Mohammed Ali Akhtar, and for a very nice and comprehensive presentation, especially on curcuminoids. And we all know how this important, the spice, the turmeric is the part of our routine kitchen items. But, we are not well aware of the best use of this spice. If you have any questions, please ask if he is available. I think that we have one more question.
Well, thank you very much, Dr. Nadeem, for your nice insights. I have a concern. One of the leading surgeon from the Light Hospital, Faisalabad, commented one thing. He stated that if a woman give birth more than 10 kids in his life, in her life, he guaranteed that woman will never develop breast cancer. Kindly give your inputs on this.
Yeah, because that was not the cause. Cause is like because as I mentioned, like some other, if family history okay, if like no any smoking, no alcoholic, not other any issues, then there should be no problem. In some cases, it has been reported in the literature that if pregnancy is some old age, not the young age, okay, but if it has getting old age, older, then might be can cause the breast cancers.
What could be the precautionary measures as in both are sitting and listening to you?
Sorry?
What could be the precautionary measures?
Precaution, yes, because I'm not
after developing of the cancers, but we should teach our youth so we can prevent from spreading such.
Yes, I think this is a good question. I think, as I mentioned, because we have to be very careful by utilizing the contraceptive and also the right time, feeding should be proper way, and also the cleanliness also important. Because why occurring? Because in our village movements, even they have some symptom, they never care about this words. Even they don't want to go for the practitioners. So this should be, I think, to be done by properly everything.
Comment from Alicia? I have some comments to your presentation. As you told, sorry, as you told, curcumin have low bioavailability, but from pharmacological point of view, the better bioavailability for curcumin is observed when we connect curcumin with paper. Is about 2,000 times bioavailability.
Low biology.
Yeah.
Yeah. Yes, that's right. Because we can say that it has so many activities. It's good for many mechanisms. Even because in certain cases it has so many potential activity, but not everywhere. You cannot say that is the drug of universal. Yeah, because it's some mechanism, madam, it can very good, no problem. Depends upon your mechanism of action, mechanism. But in certain issues that I mentioned over here, because of poor absorption, low serum tissue culture, in some of assay, some of mechanism, but we cannot say that overall. No, overall is very excellent because natural remedy. But because in certain issues we have seen that this one is not okay because penalizations. In some way, it's a good pH 7 and sometimes shows 7.5 pH, 8 pH. This pH can make some problem in some of the mechanism of actions.
So can you just give some elaboration of that, what you have just given the comment of the paper and some like a comment?
In Poland, for example, Łukasz prepared me some capsule when he mixed curcumin with paper, black paper. And this curcumin is good bioavailability in my body, and I feel so better. When we, when we're looking for some drug, we don't, we can't look only by some synthetic drug. I think we need some take lesson from nature. I think maybe it's good direction if we go connect curcumin with paper because it's good proof in the literature about this type of study. If you want, I can send.
Because it has so many applications. Even I can show you because in our the industry we are making is perfume, we are making for dyes, we are making some other recipe for the flavoring.
So
So
many thanks. Many thanks.
Dr. I have a question, please.
So
I have a question, please. आपने curcumin पे बहुत अच्छा बताया, you have a lot, but my question is, क्या इस पे कोई ऐसा काम हुआ, you find the toxicity level of the curcumin, इस dosage form में कितनी हम लेंगे तो वो toxic नहीं होगी, and secondly, if there is a comorbidity with the cancer or the patient is taking the allopathic drug or pharmaceutical drugs for the cancer, what is the interaction of that particular curcumin with the drug interaction? Have you studied on?
Because if you read this paper, because I have not done the detail. My colleagues have done the detailed work. Like I think I just showed up 4 or 5 paper last ones. They have done. They have.
We can share paper with you.
Because they have that because they have maintained the dose. They have tested on the rat animals. They have done the experiments. But as you say many times that we should move to the clinical trials. That is other story.
What is the interaction of the drug with that particular spice? So it may be harmful, it may be useful.
No, sir, that's very common because if we use as we see that little dose much better than by use the high dose, that's very normal. But if we can say that we have tested this dose, we have not done this experiment so far.
And again, one more question
Okay.
and one comment,
Okay.
very short. Breast cancer, thank you for the presentation and draw attention to the breast cancer because in Europe, breast cancer is the second reason of the woman death.
Yes.
Second reason. And I have question about molecular modeling. What software, if you can only mark, because I'm very curious of it. Because docking with curcumin with large molecules is quite difficult.
Yes.
And I'm very curious what the software.
That was done by my colleague, Dr. Zainul Kasmi. So actually, he told me that diketone is very important for the interaction with the ligands. So just he let me know that he said that this diketone is very important part. For other part, like the double bond is not much active, as you mentioned, just slide as lipophilia. That's why we remove that one. We keep the focus on the diketone.
Okay, thank you.
So thank you very much.
Thank you so much, sir. This was Professor Dr. Mohammad Nadeem Akhtar. And now our next speaker is an ex-agrarian. And I assure you that her lecture will be much more energetic, that the dizziness will vanish away. Her name is Dr. Saima Siddiqui. She did her MSc from this very University of Agriculture in 1995 and PhD in 2008 under the supervision of our Professor Emeritus, Dr. Khalid Mahmood Khan. She is working currently as a Deputy Director in Institute of Biomedical and Genetic Engineering, Pakistan. She has almost 100 international publications, and most of all, she is identified as rare phenotype, which she named as Siddiqui syndrome. Please, ma'am, come up on stage. Give her a big round of applause.
Assalamu alaikum. I hope everyone is sleeping.
Not everybody.
Okay, so let's start with the last talk. And for this, I think, session, and that is drug discovery and development in genomic area. By definition, drug is a chemical substance when consumed cause changes in the organism physiology and psychological changes, or the best thing is it's a substance that is used to prevent, diagnose, treat, or relieve symptoms of disease or any abnormal condition. The disease is actually an abnormal condition or any phenotypes or any symptoms that a person is feeling. And when we are to relieve that symptoms, that the substance that is used for it is called as a drug. Drug discovery and development have several steps. Generally, it is called as 5 to 7 steps. It has 5 to 7 steps, and it is described as the whole process is described as a sum of the total steps taken from any research initiative. Basically, it starts with a basic research. Basic research is the foundation for the drug discovery. Nowadays in Pakistan, mostly the people, they don't like if they see any project about the basic research. They say that no basic research, it should be practical, more and more practical, or more and more transformative, or it should transform, like it should bring something in industry or anything. But everything starts with the basic research. Basic research we need basic research to find out our target. When we find the target from, let's say, if we talking about any disease family, so we look into its genome, its proteome, or its clinical phenotype to see any target or anything that we can, we can correct. We can see different ways to correct it, or we take it in a preclinical research. Then we develop some clinical hypothesis out of it. Then we review those hypotheses and devise the ways to correct that phenotype. And then if there is something that we found, that need to be approved as a drug. This again shows the several steps of drug discovery and development. As we are living in the genomic era, where we have we have the whole map of our own genome, so we know where are the we have the genes which give us power. We have the genes and we are like this because of this. There is some certain characteristics in our genome that give us that particular features. So in the genomic area, basically we look for our target, disease target in the genomics through proteomics or through bioinformatic analysis. That later on developed step by step in vitro, through in vitro or in vivo studies into a definite model, which is called as a, which we take as a drug. The process of drug discovery and development is not a short process. It takes sometimes the whole life of an individual, and sometimes, like for generalized, it takes about 10 to 15 years. There is some time which is required for the basic research, but that time is also not very short. It takes a lot of time to take a disease and go with it for to look for its real cause and solution for it. Then sometimes we can design a drug for it, and then we do experiment in vivo and ex vivo. Then there is a long process of like its validation in trial, preclinical phase 1, phase 2, phase 3, and these trials are very essential to prove its efficacy and safety in human and animal models. Then we come up with a registration. There is also a short process, like once we develop drug from the scratch, then it takes so many years to develop that. But there is a process that, which is called drug repurposing. When we have two similar processes, we say this drug A is effective against this condition. This might affect the condition B also. So we try that, and this is called drug repurposing. And this is sometimes, if your luck matters, you can come up with a very short time with a new solution, new treatment, and new drug. Human genome, as we all know, there are 23 pairs of chromosome, 3.2 billion of nucleotides. So we, like this effort started to map the human genome in 1990, and after in 13 years, in 2003, we get the whole map of human genome. This genome this map started then the new avenues of exploration, like we start looking for the real cause or diagnosis of any disease into the genome of an individual. We can say that whenever there is a problem, there is a solution simultaneously in that organism. Like there are plants that give you solutions that give you relief, and there are things that make you that create problem for you. The genomes give us DNA as a target. DNA we find changes with as when we compare it with the reference genome, we see the changes, and these changes, if they are responsible for the disease, we call it as a mutation. And we target these mutations, we do something to correct these mutations, correct the gene. So it's that will go towards the therapy, and this is called gene therapy. When we talk about RNA, we find some changes in the RNA or in the three-dimensional structure of RNA so that it cannot perform its function. So there are certain ways like CRISPR-13 system, Cas system that affects the, that can edit the RNA. Then there are protein. The proteins, if we find defects in the protein in any level, then proteins can be replaced like enzyme replacement therapy, or in any way we can treat any disease. After like validating or identifying our target, like if there is a defect in the genetic level, so we have now a CRISPR-Cas9 system to that cut out the faulty gene from the genome, and we can replace the healthy gene of the individual so that
we can find a permanent solution. That it's not very easy, and it took several years to come up with this. So there are no novel therapies which are available like Zentaglo and Cascavy. They are very open and FDA approved in the market, but they are very, very expensive. So there is a time so that we should also go into these fields to help our own patient and give the reasonable, like we cannot afford the $1 million medicines. So we have to do it over here so that we can minimize the cost and give relief to the patients. These are some of the examples that now cell and gene therapies which are in the market, and there are many which are based on small molecule chemistry, monoclonal antibodies, and some are on biologics, and some are cell and gene therapy. You can see that from 5% to there in 2023, it goes up to 13%. That is monoclonal antibodies, sorry, cell and gene therapies from 2% to 10%. So it's gaining its importance, and this is a one-time therapy, and it corrects your problem for lifelong. Orphan drugs are another very good candidate to be targeted by gene therapies because they are due to the defects in the genome. There is a market size for the global orphan drugs. Here in Pakistan, we have very high percentage of orphan diseases. We do not, maybe we do not have exact figures because we don't have such studies which give us the real figures about their prevalence, about their typing, about what kind of diseases are here. But it's a fact that we have a lot more patients than it's anticipated in any level. So globally, 28% of the market blood-borne diseases are corrected by therapies, gene therapies, and 20% central nervous system disorders are being corrected by this, then endocrine, and then other are very less. Beta thalassemia is one of the examples that now have approved gene therapies. And it is estimated, like in Pakistan, we have like very large populous country, 5th most populous country, and we have like 5 to 7% frequency of beta thalassemia. So there are 10 million, more than 10 million carriers in our population. So the treatment which is available, these are basically these patients need constant blood transfusions. That's not easy, not for patients, not for their families, and it's very costly and it's very painful. So one-time solution or gene therapy that is available for these are Cascavy and Zentaglo, but it is like one dose is around 2 million, and it takes several months to treat a patient. Then the other second most common is a rare diseases, which are also a candidate for gene therapy. With the gene editing technology, scientists are working to correct more and more diseases every day, every month. This is the landscape where the world is going. Like they are the companies, very big pharma companies in this game. Like they have many, how can I, it's a small, I think there are many, these all are the drugs or studies that are going on worldwide to, they take a one, like each one is taking one as a target, one thing, one disease, one problem as a target, then gradually it's moving towards the center. In the center, the drugs that are shown in the center are those that are FDA approved now. One is, I can read Zentaglo, I can clearly read, and the other one is Cyclo something. If I can enlarge this image, I don't know where to do this. Can be enlarged? No. So it's like Dr. was in the morning, he was asking about the great invention of 2023. Of course, it is the treatment of sickle cell anemia and beta thalassemia, and that is by Zentaglo and Casgevy, the two drugs. And here the Casgevy act as a like for the two, one single solution for the two targets, beta thalassemia and sickle cell anemia. This is after the genomic, like we have found the whole genome map. The things are going in a very high speed. There are more and more diseases which are diagnosed each day, each year, and the global testing market is going very high. It's a 16.69 billion market now for till 2032. The rare diseases, the both gene therapy for the rare and diseases and all other diseases are going side by side. So it's not that these diseases are rare, so they may not had affected the market, or they may not have been used for the therapies, but they are going side by side with all other diseases. They are gaining equal importance. So these studies or these trials are mostly done on different race and populations, different age distribution and ethnicity distribution. So far, the studies that have been done is you can see mostly are done on 76% and done on white population. Asian, only 11% Asians are included. So Black, Hispanic, and other. So we need to include our own population for such kind of studies, because we also need solution for them as well. These are for for all of your young students who can join these things, because it takes years to reach any conclusion or to any achievement. This is the same, like more and more studies are done on white people and very little on Black and Asians. Drug repurposing are the same. Here, I want to show you that in our institute, we have started genetic evaluation. Like we have instruments like microarray and Ion Gene Studio S5, and we have started the services. This is very much ready. Like we are doing samples for diagnostic purpose also, and these are these instruments are very basic, and these are very basic to find your target. Like if you are working on the large pool of cancer patients, so you have to do go through your samples to have a microarray analysis for your samples. You can easily have your main target. Microarray does the 7.5K array analysis, like 750,000 SNPs are. He read the SNPs throughout the genome of an individual, and then he does not miss anything. He does not put any gap. You can see even small deletion, duplication, mosaicism, and everything you will have in a one window. So these are very, very important instruments to give you a real hint for your further work. So if you are to plan some projects for research, we are very happy. We will be very happy to support you in this way. And we have the instrument, we have the capabilities, and we are running all these things. And I think this was all.
There is any question, or I think people are
want to sleep.
But thank you very much for this wonderful talk. So we can have more. You can have questions if you're interested, especially some collaborations, so we can talk about at the dinner as well. So our last presenter today is yeah, you can announce it.
Dr.
You can announce.
You can announce.
Then the next person who will be presenting is Dr. Kira Ija. Yes. Welcome, Doctor, from Pak Austria, Pak Hochschule, Aripur, Pakistan. Austria is near to Poland, then we're very interested about the cooperation. We're looking forward for your presentation.
Thank you. I'm Dr. Hirai Chas from the Park Austria Fachhochschule. First of all, I would like to introduce my institute. Fachhochschule means in Germany, it is the German word means Institute of Applied Sciences. And it is first initially introduced a concept in the Germany, then in Austria. Now in Pakistan, this concept was introduced in 2019. And it is the first model institute in the Pakistan. And then this is the infrastructures and this is the aerial view of our institute. Now come towards the topic of my presentation or talk, development of polymeric network for controlled drug delivery. Now the formulation scientists face a lot of problems while developing stable formulations. Many stability issues comes like interaction of drug with the excipients, degradation of acidic drugs. Degradation drugs are very heat sensitive. In the same way, I have used pridopyl albumin, which is used for the treatment of hypertension. Pridopyl albumin shows various limitations like it is a highly sensitive drug, acid sensitive drug, heat biased drug. When it come in contact with the various pharmaceutical processes like milling, mixing, granulation, it undergoes degradation. So in order to overcome all these issues, it is important to develop a stable dosage form. Objective of the study is to use a graft polymerization to protect the drug from acid degradation, from the heat degradation, and to protect the drug from the different pharmaceutical processes, and to overcome the cost of the therapy, to improve the patient compliance, and to attain the desired therapeutic effect. As I have told that the pridopyl albumin is the acid sensitive drug. So when we take pridopyl albumin in the tablet form, in the stomach pH is 1.2. When our drug comes in contact with the acidic environment, it undergoes degradation. So major purpose of the study is to protect the drug from the acidic environment of the GIT. This is the workflow. Various formulations were formulated by using xanthine gums, which is used as a polymer, and the vinyl monomer, which is acrylic acid, and MBA was used as a cross-linker. MBA means methylene bis acrylamide. And in vitro evaluations include swelling studies, on-off switching studies, soldier fraction, drug loading, estimation of drug loading. And the instrumentational characterization includes FTIR, scanning electron microscopy, and dissolution studies. This is the structure of the xanthine gum. It is actually a hydrophilic polymer which swells in the basic media at pH 7.4. And then I use acrylic acid, which is the monomer. It is actually a vinyl monomer, which is pH sensitive or pH responsive. It undergoes swelling in pH 7.4. However, it prohibits its swelling in pH 1.2. Next is a cross-linker. I have used methylene bis acrylamide. It is a branch cross-linker which provides efficient cross-linking between the polymers and the monomer. This is the hydrogen bonding between the polymer, which I am using xanthine gum, and the monomer, which is acrylic acid. Methodology. First of all, I have prepared solution of polymer and then added initiator into it. I have named it as solution 1. Second solution was prepared by adding close linker into the monomer. After a certain period of time, both solutions were mixed on a hot plate, and then this mixture was transferred to the test tubes, and one test tube was kept in the water bath for about overnight, or you can say 24 hours, and resulting in the formation of hydrogels. Hydrogels are then removed, cut into pieces, and dried. In vitro characterization includes swelling studies. Swelling studies we are using for estimating the targeted drug delivery. Why we are using targeted drug delivery? Major aim is to track the drug in the stomach, which is pH, where the pH is 1.2. And our aim is to target the drug in the lower portion of GIT, where the pH is 7.4. Pulsatile behavior, which is used for studying the controlled drug delivery. Next is the sol gel fraction. It indicates the degree of cross-linking between the constituents like polymer and the monomer. Next is the drug loading. And the last point is the estimation of drug loading. Instrumentational analysis include FTIR, which is used to study the presence of different functional groups on our of the polymer, monomer, or the formulation. XRD studies was done to check the crystalline and the amorphous nature of the drug, polymer, and monomer. Scanning electron microscopy was used to check the topology as well as morphology. And the dissolution studies were used to check the drug release over time in pH 1.2 and as well as in pH 7.4. This is the composition of hydrogens. 8 different formulations was formulated by using varied ratios of polymers, monomers, and cross-linkers. This is the presumptive diagram. First of all, swelling studies. You can see here swelling studies was performed in pH 1.2 and pH 7.4. In case of formulation, xanthenegram Xg1 to Xg3. As the concentration of xanthenegram increases, the swelling increases in pH 7.4 as well as in pH 1.2. But the swelling was significant in pH 7.4. In the same way, as the monomer cross-linker concentration increases from in pH Xg5, the hydrogen's swelling decreases. This results in the formation of dense network, means it has less porous architecture. In next formulations containing varying concentration of acrylic acid, Xg6 to Xg8, as the concentration of acrylic acid increases, the swelling increases in pH 7.4. The increase in swelling or decrease in swelling is related to protonation and deprotonation of carboxylic acid group. Next is the on-off switching. On-off switching studies was performed to check the control release behavior. Okay. First of all, hydrogens was allowed to swell in pH 7.4. After a certain period of time, 15 or 20 minutes, the hydrogens are transferred into the beaker containing pH buffer solution of pH 1.2, and the de-swelling occur. Here, the protonation of COH group occur. Again, in pH 7.4, deprotonation occur, and deprotonation led to the generation of COO negative group, which resulting in the anionic repulsion, and the electrostatic force of repulsion results in the generation results in swelling. Sol gel fraction indicates that as the number of constituents or the ratio of substrate increases, gel fraction increases. Xc5 containing higher concentration of substrate, polymer, monomer, and the cross-linker show greater gel fraction and the minimum sol fraction. This is the FTIR spectra showing various function groups in the printopril albumin, which is our model drug, xanthenegram, unloaded nexus, and the drug loading nexus. And again, XRD shows xanthenegram has a crystalline nature, which show by the peaks. And again, the printopril albumin XRD shows peaks, few sharp, few dense, which indicate the crystalline nature of the printopril albumin. However, these peaks were diffused and minimized in the drug-loaded nexus, which means that our nexus convert the drug from the crystalline to the amorphous form. Next is the in vitro dissolution studies. In vitro dissolution studies are in line with our swelling studies. Hydrogen swelling was maximum in pH 7.4. Same in case of as that of swelling, and again it was decreased as the concentration of our as the concentration of cross-linker decreases, and again drug release increases with increase in the concentration of monomer, which is the acrylic acid. This is the same micrograph shows that our hydrogel poses rough, irregular, porous structures, which allows imbibition of water, resulting in the increased swelling and followed by the increase in drug release. This is the stability studies. Stability studies were performed up to 6 months. And at a accelerated stability condition, which is 40-degree temperature is kept as 40 degrees C, and the humidity was kept at 75%. In the humidity chamber, after 6 months, our hydrogels were removed. This was performed on the optimized formulation, which was XD4. And again, dissolution studies were performed, and dissolution studies shows that our formulation remains stable over 6 months. Key points of the studies was that swelling index depend upon the pH of external media. For example, we have used two pH, pH 1.2 and 7.4. As the pH increases, swelling increases. Soil gel fraction depend upon the composition of network. And also fabricated nexus expects good pH sensitivity required for the controlled drug delivery. Grafting of acrylic acid to the xanthane gum enhance the fundamental properties like hydrophilicity and ionic nature. FTIR spectra confirm the formation of highly cross-linked network. Conclusion: the intention was of designing the research studies was fulfilled by designing the novel controlled drug delivery system, which released the drug for the extended period of time. We have to take only one hydrogel formulation, and the drug release occur up to 24 hours. It means it improves the patient compliance. The microporous architecture provides sustained release effect. As I have already told us, swelling occur up to 72 hours and the drug release occur up to 24 hours. And next is a future perspective. And as these hydrogels can be used as carrier for the preparation of for the delivery of various bioactive molecules, or it can be used for the delivery of various heat sensitive or heat labile or acid sensitive molecule. It can be used for the delivery of insulin. As we know that insulin also acid sensitive. It degrades when we take it orally. So it can be used for the delivery of insulin. It can also be used for the treatment of the colon cancer. In colon cancer, we have to target our drug in the lower portion of the GIT, which is the colon where the pH is 7.4. We can use this carrier for delivering our drug to the lower portion of GIT where the pH is 7.4. In the same way, colon cancer is also one of the most important causes of death worldwide. These are the references. And these are publications. This publication was published in the polymer plastic technology and in engineering, and its impact factor was 3.2. Other relevant researches are in International Journal of Polymeric Materials and Polymeric Biomaterials, Drug Development and Industrial Pharmacy. Thank you.
Thank you, Doctor. Very interesting presentation for pharmacists, particularly. And there are questions. And the question.
Mike there.
Thank you so much for your nice presentation.
Thank you, sir.
The two concerns this total research presentation, the drug load and sustained release of the drug. Do you validated the drug release, sustained release with the passage of the time, and how you validated it?
We have performed drug dissolution studies at 2 pH. I have already told in pH.
Every time, no, this is the in vitro.
Yes, in vitro.
Something else.
In vitro drug dissolution studies.
I'm just asking, the sustained release of the drug, have you validated that sustained release every time?
We have performed these pharmacokinetic studies in vivo also. And the drug same shows its effect as that of in vitro with minor variations.
Is there any, on drug load, is there any effect of concentration of drug?
Yes, yes. We have to adjust the concentration as per dose of the drug.
Any more questions? Okay.
Oh,
the last row.
Question? No,
No question. Great.
Maybe I have one question, if of course you are able to answer. Because Perindopril is very common drug in Europe
Yes.
in hypertension treatment.
Yes.
And preparation registered is perindopril mesylate. What is the difference between your salts?
Sorry, this is actually a chemistry topic. I have totally drug development and drug delivery research. So I guess chemistry people can better explain about the salts and its modification. Yes, we can do salt modification for improving the solubility, stability, bioavailability.
Okay.
But in Pakistan, mostly pindopyl albumin salt is employed, and I have arranged this salt from the France. Actually, it was prepared by the CCL Pharma, and they only get imported from the France. So I managed to get from the France that salt.
This is the question to be asked to Dr. Nadeem. Okay, thank you very much.
Thank you.
So please wait. She was a little late, so she couldn't join the morning session. So we will present her shield. So I request the honorable guest to please present the shield to Dr. Hira. So you come over here.
Okay.
You have some announcement to make? Okay.
Okay.
Running through hands, yes?
Yeah.
Yeah.
Sure.
Okay, then thank you all for the attention. And I think we are waiting for the dinner, yeah?
Yeah.
The serotonin.
Now it's serotonin time.
Yeah.
Thank you all. And I close the meeting and maybe give speech to Professor Amar.
Thank you very much.
Oh, good morning, everyone, and Assalamu alaikum. So I am Dr. Muhammad Ali, the assistant professor in the Department of Biochemistry. And I'm happy that we all are inviting foreign guests. So we are starting today's technical session, and with the first presentation from the Professor Dr. Ata Athar. He is from the Michigan Technological University, USA. So I would request Mr. Dr. Ata to introduce him a little more because I don't have his introduction. So I'm requesting to introduce himself and then start the presentation. So it is over to Dr. Ata.
Thank you very much, Dr. Muhammad Ali. Actually, I was born and raised in Faisalabad because my dad was in army, so keep moving various part of the Pakistan. And I graduated with a master degree from University of Agriculture, Faisalabad, and then I went to Karachi for my PhD. I did my PhD from HCG Research Institute of Chemistry, University of Karachi, and then I came to USA for my postdoc at Florida Atlantic University. And then in 2001, I got a job as assistant professor at University of Winnipeg, and I ended up staying there for almost over 23 years. And then last August, I got an opportunity to work at the University of Michigan Tech. And that was a wonderful institution because it is R1 institution. R1 institution means it is research intensive university, and when you produce certain number of PhDs, then you are classified as R1 in USA with research intensive. So we have a lot of funding from the national funding agency, especially National Institute of Health. So this is the reason I developed an interest there. I said, why not? I can spend some time in Michigan Tech and work there. So I am head of the department over there, and we have actually, I will show you guys how it look like. So Houghton is a very beautiful area. It's a small town. It's a rural part of USA. It's a very beautiful area. So I would like to request Muhammad Ali Sahab to move my next slide so I can show you guys actually how Houghton look like. And so my next slide, can I have my next?
Yes. So for all participants, the slides are moving. Is it full screen of the slides for all the participants?
No, sir, it's not the full screen.
No, sir.
So, okay. It's not the full screen.
I think you have to click the screen. If you see the bottom, you will.
So even you cannot see the slides. What is visible to you? What you are
No,
looking right now?
we can see the slides. Full screen is not going on. Like side by side, the slides are going. वो जो full screen option, I think you have to turn it on.
Okay, okay, I do something.
Yeah,
Okay.
just away from you can zoom it. Beside that, you will see the icon of the full slide.
So now the full screen, right?
Yeah.
Okay, that's
Maybe
great.
try to put F5.
Yeah.
Yes, yes, yes. This is right. I even hit the presenter's view. Now is it full screen now?
Not yet.
Not yet.
Not yet.
Okay.
Maybe you are sharing the not the screen, but exact application, exact software, exact the window, yeah. And it's the problem sometimes with Teams, try to share the screen or alter window in sharing.
Oh, no.
Now we can see presenter better.
Entire screen. So I share the screen now. Anything
Yeah,
on
I
the screen
think
will be visible.
Yeah,
I
now
think
it's
you are
going on.
going closer now. It is still is presenter's view. I think now you is it's not now. I think now it is done. Yeah.
Now
Now it's
okay.
done.
Yeah.
Finally.
Okay, so thank you very much. This is the first slide, and I will be helping you actually for moving the
Sure.
slides.
Thank you. So we can move on to the next slide. So I can share you guys some beautiful Houghton. You guys can see in the summer, it's very beautiful. You will see a lot of trees and you can easily walk. This is a nice side of the campus, going to different buildings because each building has its own department. So you will see that. Can I have my next slide? Yeah, this is actually Husky. Husky is the logo for the Michigan Tech actually. And if you see this building, just behind the Husky, this is the Department of Chemistry building actually where I live. My office is on 6th floor and my lab is on 7th floor. So it's a very beautiful area. And this is all the summer. And in the summer, you will see a lot of flowers. So can I go to the next slide? And you guys can see now actually this is overview from the plane and you can see the nice lakes passing by. We can call it canal because it connects the lake and actually it's because we are living near the Lake of Superior. Lake Superior is actually one of the largest lake in the North America between US and Canada. And if you cross the lake, you can easily come to Canada, but the distance is almost at least 200 kilometers. It's not easy. So, and these are the different, all the buildings you can see these belong to the Michigan Tech. In a short form, we call it MTU. And I can tell you, you will see a lot of activities during the summer. So can I go to the next slide? Winter activities are also fabulous. You can see actually all winter activities. Winter actually, being a Canadian, I always enjoy winter because we have a lot of snow activities all the time. So you can, do not think we hibernate during the winter. We always go outside and enjoy our life during the winter also. Can I have next slide, please? Yeah, this is another view. You guys can see a lot of trees. And as I mentioned, it's a purely, we call it rural area of USA. And plus you will see beautiful drive alongside the, canal and the lake. So you will see actually, because this area is in between the lake and the canal. So it's a beautiful area actually. And you will, if you visit, but most welcome to come. I will explain you guys the opportunity, how especially graduate student or senior professor can come. So can I have next slide? I think yes. As I mentioned, winter, you don't think we sit inside. We always go outside, enjoy our life, and walk on a snow. It's really fun. You can ski. You can do a lot of skiing and skating. So it's a beautiful, and you can do a lot of fun. It's a lot of exercise.
हमारी मैडम ये बोल रही है
This is our new actually building that we established beside this, as I previously showed you guys, just behind the Husky, you see chemistry, and this is also STEM building. We are moving different departments here, and this building will be soon functional. And because we are moving in this month, and definitely from next month, it will be functional. And it has a state-of-the-art building, and so we built almost with over $9 million, and that has all the facilities that you require in the sign. So can I have my next slide? Actually, again, same buildings. So in the winter, it look like, you guys can see all the time, look like this way. Roads are clear, but you can have, snow you will find everywhere. So can I have my next slide? This is actually main entrance of the Michigan Technological University, but we have different areas to enter, so you can go from different directions. And it's one of the safest area in US, I call it, because it's, as I mentioned, a rural area. So here, actually, very safest area. You will see it's very safe campus and a very nice, beautiful area, and also almost you will see a lot of international students there. So you can see there. And especially, actually, if the students or the faculty want to spend time with us, they can try to apply for the Fulbright scholarship. And it's easy. And just let me know in chemistry or biochemistry. I would be happy to, find a faculty member for you guys and connect you guys. And so he can be your host. And then you can apply for the Fulbright scholarship. You can come and work here. And then if you want, you can submit the thesis in Pakistan, or do this, or the other possibility, you can use the HEC indigenous scholarship to they usually send for 6 months. But what can I do in our university? I can request so they can waive the bench fees. It will be more easy for you guys to come here, and you guys can work with any professor, and definitely it will help you guys to gain some expertise and top class publications from here. Because if you see, as I mentioned, we are R1 University. So if you can just try to Google it, most of the publications are coming one of the top leading American Chemical Society journals. So you will see there. Okay, so after that, I think you guys can see me now. So I have to go back to the slides. So can I have my next slides? Actually, uh, this is actually Winnipeg winter activities. I was just thinking I will be away from the snow. No, I'm again in the snow. So I always enjoy snow. Snow is one of the best part of my life since I came to Canada. So it's always there. So let's see, can I have my next slide, please? I believe we can start the science that we have to discuss it. As I mentioned, if you can see, the title of my talk was Natural Product Based Drug Discovery, and how the pharmaceutical activities operates. So drug discovery, actually, how the drug discovery works. So first of all, you find a selection that find a target, for yourself against which disease you have to discover compounds. And then you design the bioassays. Bioassay can be of two types, one of the enzyme-based bioassay, or other one is the cell-based bioassay. Most of the organic chemists you will see, they will do the enzyme-based bioassay. Why we do the enzyme-based bioassay? Because they are easily safe and you can easily handle in the lab. And if you see, most of the diseases are caused by the malfunctioning of enzymes, and lot of pharmaceuticals you will see in the clinics, they are enzyme inhibitor. One of the enzyme inhibitor is lovastatin, that inhibit the biosynthesis of the cholesterol in the body. If you see, it's a very complex mechanism, but it inhibits the one step, and that help to control the cholesterol biosynthesis in the body. So based on this, you will see a lot of so mostly in my lab we work on the enzyme inhibitors. So you will see it will provide us the source lead compounds, and then we can the lead compounds can come from the organic synthesis or the natural product sources, and we screen them using these bioassay. And then once we have the bioactive compounds, then we do some further pharmacological assays, and so we can find its practical application, so we can hand over to pharmaceutical industries. So can I have my next slide, please? Why we work in the area of natural products, preference is the natural product chemistry. We do synthesis, but I mostly prefer to assign the project related to natural products in in my lab. Why? Because if you see the literature, you will see more than 60% of the drugs that are available on the market are of natural product origin. Only you will see close to 37% come from the synthesis source. So this is the reason natural products are very important, because you will see examples include Taxol, that is used as anti-cancer agent. Penicillin is an example. It's a purely natural product, and though they have modified structure using organic synthesis. And another common example is the morphine. If you see, is still active in the clinic as a painkiller, because after surgery, doctor will put you on the morphine, and then after he will try to ask you guys after a couple of days, because morphine has addictive nature, so he will ask you guys go back to the normal painkiller like Tylenol, paracetamol, so you should not be addicted to that. But natural products definitely have pharmaceutical activities. So if you can go a little bit further, can I go to my next slide, please? Yeah, as I mentioned, actually, we work in the area of natural products because natural products provide the structural diversity. The structural diversity is difficult to achieve in through chemical synthesis or even combinatorial chemistry. So this is the reason, because if you see how many compounds synthetic chemist can make in the lab, you will see in his entire life he will be able to make maybe 1,000 molecules, and 1,000 compounds he can make different compounds. I'm talking different structures. So, but if you see the plant can make 1,000,000 compounds in 1 week. So compared to, or because nature is an excellent chemist, so due to this reason, you can acquire the structural diversity that you cannot achieve through the organic synthesis or even combinatorial chemistry. Because when I started my PhD, when I was almost started my professional career after PhD, the combinatorial chemistry was discovered. And in combinatorial chemistry, you can make millions of compounds in 1 day rather than isolating one natural products over a couple of years. So I was thinking, I think the era of natural product is gone, and natural product chemistry has no future. The way it is going, combinatorial chemistry was discovered. So what if you see the literature, combinatorial chemistry, I did my PhD in 1996. So you guys can see it from 1996 until now, combinatorial chemistry has provided only one compound that has anti-cancer activity, and that compound actually is in clinical trial, but it has so many side effects, so people are not using it. So due to this reason, I would suggest if you see pharmaceutical industry, they are coming back to natural products, so they can start working in the natural product chemistry to discover drugs. But the approach is different for sure, and we move more because natural product chemistry is a very labor intensive work. In order to avoid the labor intensive work, we are trying to discover actually LCMS, LCNMR, so that can help us to purify compound and it avoid the replication problem. Also, replication means how we can avoid the isolation of known compounds. So that's the reason we are working in the area of natural products, due to structural diversity that we cannot achieve through any other kind of science. Can I have my next slide? And as I mentioned, we work in the natural product chemistry basis. We do synthesis, but problem is that actually we do the synthesis if we have to synthesize some compound, or we have to, we have to modify the structure of bioactive compound to study the structure activity relationship. That we do that. So can I have my next slide, please? So problem with the natural product chemistry research, as I mentioned, actually in the plants, plants produce sometime compounds related to season. So it means their reproducibility is a major problem. Sometimes you isolate a compound and you cannot again isolate from the plant, until you use a similar condition when you collected the plant. This is the reason whenever we select a plant, we write the conditions, location, and the time, and the year, day, and the time, so we can make sure that we can get some reproducibility. Otherwise, you cannot do this. And this, I'm talking about plants here. If you go to marine organisms, you guys can see marine organisms stay inside the oceans. If you change 1 kilometer of the location of collection, you will see they are producing same species of sponge or coral will be producing different compounds. So due to this reason, we always say actually the lack of efficiency is a major problem and reproducibility is a major problem in natural product chemistry. So this is the reason sometimes we find some biotech method to produce bioactive compounds in the lab. Once we isolate the compound, we know that we may not be able to get that compound again from the nature. Selection of plants is another criteria. I will discuss the selection of plants. Different people are approaching different criteria for selecting the plants. So that I will discuss in next plant. Replication is major problem. Replication means isolation of a known compounds that already show a lot of activities. That is called known as replication. So replication you can actually avoid by using LCMS and LCNMR methods. So you can characterize those compounds, and if the plant, you can only work on the dereplicated extract. So dereplicated extract can help you to isolate the novel chemical entities to discover new drugs. And then another problem is synergetic effect. When you see the plant is significantly active, and when you do some bioassay guided isolation, you will see all the compound have some very mild activity. So we call it actually synergetic effect. So for synergetic effect, we always do the bioautographies to make sure that we can avoid the synergetic effect. So we can know the bioactivity. We also develop bioautography, means we develop the TLC based bioassay. So we can know that where the bioactivity is and how can we, how many compounds are showing the bioactivities and which one is showing the more bioactivity compared to the other, or only 1 or 2 molecules are active. If 1 or 2 molecules are active, that extract we usually work on that plant extract. Otherwise, we try to avoid that. Can I have my next slide? As I mentioned, I will be discussing in my next slide, the selection of plants. People select the plants actually by using folk medicinal applications, reported biological activities, or random selection, which is highly, not recommended. Why it is not recommended is just like fishing around. So, random selection will never give you anywhere to go. Bioinformatics is also not recommended. Why? Because very little genomic data on the plants are reported. So if we have very little genomic data, so it means we cannot actually target a plant for anything. However, it can help you guys to isolate known compound. But most of the time, people use these two for ethnomedicinal application of the plants or the reported biological activities. So it is our experience, as well as reported in the literature, if the chance of getting bioactive compound based on the ethnomedicinal importance of the plant is 70% higher compared to other reports. So we always use the plant, folk medicinal folk indigenous medicinal plants actually, so we can discover some new bioactive compounds. So can I have my next slides? And as I mentioned, actually a lot of problems in bioassay directed. You will see false positive results. We come up with because plants contain millions of compounds, not only one bioactive compound. Sometimes the false bioactivities are coming due to lignin. So what we do, we usually use we filter the extract using charcoal or the resin, so we can remove the lignin or other biopolymers, so we can see the bioactivity is associated with the small molecules. And then replication, I told you guys we do the LCMS or LCNMR. So can I have my next slide? So I will give you guys actually some idea what type of work we are doing. So for example, if we see glutathione, GST glutathione as transferase, this is a phase 2 detoxification enzyme, and it catalyzes the reaction of glutathione and electrophile to make glutathione adduct. And this glutathione adduct is a water soluble, and it can be excreted from the body. And what it does, most of the xenobiotics in the form of even the drug, for example, anti-cancer drugs or anti-parasitic drugs, they contain electrophilic sites. So what's happening in case of drug resistance problem, especially in anti-cancer drug resistance problem, So what's happening? Those drugs make adduct with the glutathione, and then actually it's excreted to the body. So we are lowering the effective dose of the in the body. If we are lowering the effective dose, so it means that those cells become, the cells are recognizing it. As I mentioned, nature is an excellent chemist, so they develop some enzyme to counter that drug. So what happened? They actually develop resistance to the drug, and then once those cancer cells grow, they become so nasty, so no drug works on them. So what happens? Eventually, people die. So due to this reason, we decided how we can overcome the drug resistance problem by using glutathione S transferase. And it's not only we have discovered based on the several scientific factors. There are several reports which shows that the glutathione S transferase is overexpressed when in the patient who are using the anti-cancer drugs. So due to this reason, you will see they have increased the four times more overexpression compared to the normal cells. So due to this reason, we decided we can discover some GST inhibitors, so we can inhibit the activity of GST, so that those compounds can be used as adjuvant during the cancer chemotherapy or any parasitic chemotherapy to overcome the drug resistance problem. So when we look at the literature, people were using long time ago FDA approved one compound, but that was very toxic. So we decided we can explore the natural products, because natural products have some less side effects compared to the synthetic compound. That was the ethacrynic acid. So can I go to the next slides actually? So I told you guys actually these compounds have application to overcome the drug resistance problem. So this is the reason we are interested in this area. So can I have my next slides? Sorry, Zahid Muhammad Ali sir, you'll be tired actually by changing the slides.
No,
Okay.
it's okay. I'm enjoying actually. Thank you.
Okay. So we develop actually, as I mentioned, actually now, as I mentioned previously, glutathione will make an adduct with the drug, because drugs contain electrophilic center, and that drug is a water soluble. It is excreted in the body, and then you cannot actually maintain the required dose in the body. So you will be decreasing the efficiency of the drug, and the cancer cells will develop resistance against those things, because they also contain the definitely defense system in their body. So we can go to the next slide. So we actually, we took actually some plants, and especially we collected some plants from Sri Lanka, and we found actually some, we did the bioassay guided isolation. When we did the bioassay guided isolation, we found actually, this phenylethanide, they inhibit the GST activities, and almost the IC50 value was close to the ethacrynic acid, little bit higher. So we kept the false positive results in our mind, and human error in our mind. So based on this, we are saying more closely to this. And we found actually this compound is water soluble, and these phenylethonides have a less side effects compared to the ethacrynic acid. So based on this, we are thinking we are thinking is a less side effect, because if you see this is present in cereal. These phenylethonides are present in cereal, present in the different grains. So based on this, we are thinking it will have, because we always eat, nothing happens to us. So we always think they have a less side effect. So based on this, we have provided this compound to our collaborator, and they are working on it to see its in vivo activity. In vitro, its activity is strongly done. And once when it has passed the pharmacological in vivo assay, then we are planning to take this compound to the clinical trial and to see whether it can be effective to overcome the drug resistance problem or not. And then in addition to this, we got actually other glycosides also. So, but they were not actually iridoid glycoside, but they were not as active as the phenylethanide is. So we found actually phenylethanide has significantly higher bioactivity. But if you see the higher activity of phenylethanide is due to, most of the time, it is also our experience that all the compound that contain the alpha beta unsaturated carbonyl carbon, they exhibit the anti-GST activity. This anti-GST activity, so we think actually alpha beta unsaturated carbonyl carbon is one of the required pharmacophore for the expression of the anti-GST activity in these compounds. Am I running out of time?
No, it's okay.
No, it's fine. I think
Okay. Okay. So then we found, as I mentioned, we have discovered. Once we have discovered it, so we have provided. I'm not able to show you the structures of those phenylethanide that we have provided for clinical testing to Pfizer. So they are doing it. So let's see what are the results there. So since we have discovered the active pharmacophore, so then we have decided move on to the next enzyme. We can discover it. So we are working in the long time ago when I came to Faisalabad, we started this project at that time, and we found actually, renin inhibitors have activity, can be used to discover compound against hypertension. Because why? Because cardiovascular is one of the leading disease, not only in the developing country, even in the developed world. So mostly it is caused by the high blood pressure. And so what's happening, if I can, what's happening inside it, so I can show you guys what's happening in my next slide. You can see the renin is produced at the surface of the kidney, and the liver produce the angiotensin enzyme. So angiotensin, renin converts the angiotensogenin into angiotensin 1. Angiotensin 1 actually is not active, but the converting, angiotensin converting enzyme converts into angiotensin 2. This enzyme lies on the surface of the arteries that control the blood pressure. If it is overexpressed, it will cause the high blood pressure. But if you inhibit its activity, so you can decrease the hypertension. So due to this reason, we have found actually any compound, the anti-renin compound have potential to discover compound against hypertension to overcome the problem of cardiovascular diseases. So when we did some literature search, we found actually L-ascarin, it's a synthetic compound, and it has, it is used to treat the hypertension. So-called it has mode of action is the same. It inhibits the renin activity. So IC50 value is 6.6 nanomolar, but it has several side effects. Due to this side effect, we decided nobody is working in the area of natural product chemistry to discover actually compounds against renin. So we decided that we can discover, we can explore natural product chemistry to discover compounds there. So we can see what we have discovered. Can I go to the next slide, please? We have a library of plant extracts, and so we found actually Nucleolothifolia. We screen all these plants in our renin inhibition assay. So we found actually yellow hits are positive. So I will hopefully share you guys the result of couple of plants here. So that is the Nucleolothifolia that has yielded us actually very mild compound, very mildly bioactive compound. So that I will share with you guys. So can I have my next slide, please? Can I have my next slide, please?
Yes.
Yes, sir.
Nucleolothifolia, we collected it from South Africa. This plant is also found in Nigeria, and it has very good activities against different diseases. Based on this, if you will click it, it will give you some more bioactivities. Yes, I told you guys that it has, and people use it to treat malaria and other infections. So when we disagreed this plant was active in our bioassay, we decided to do bioassay guided isolation on it. Can I go to my next slide, please?
Yes, sir.
Yeah, what we did, little bit showing you guys how to do bioassay guided isolation. We took the plant extract, defatted with hexane. We found the hexane extract was not active, defatted extract was active, and then we did the column chromatography, and we ending up with the very nicely two fractions that were showing activity, remaining fractions were inactive. So we combined these two fractions and again did the chromatography to isolate the compound. Can I go to my next slide?
Yes, sir.
I can't, yeah. We end up with you guys can see carbazole type alkalides, indole type alkalides. And these indole alkalides were moderately active. So now when we combine them, they were showing significant activity. But when we isolate them again, they were showing moderate bioactivity because IC50 value is significantly higher. If you see compared to our standard, that was nanomolar active. So what we did, we decided this plant is showing bioactivity against renin system due to the synergetic effect. So due to this reason, we actually did a lot of chromatographic work on different fractions. Are we missing some compound, but ending up with nothing? So we decided actually this bioactivity is due to the synergetic effect of the plant. So and we collected this plant based on the reported bioactivities. Then we also did some phytochemistry. Yes, I want to go to the next slides. Thank you. So then we went to the Mimus elongi. We collected this plant again from South Africa. They used it to treat the wound healing properties. The traditional healers told us that they are using excuse me, this plant to treat the heart diseases or to treat the blood pressure. Then we screened this plant was significantly active in our bioassay with an IC50 value of 10 microgram per ml. That's a very good activity compared to the crude extract, if we compare it with the pure compound. So we were thinking we will be getting very nice compounds. So we did our bioassay chemistry, just like I showed you guys a few minutes ago. We end up with the carbazole type alkalides. And these carbazole alkalides were significantly active. We found actually the biphenyl ring that has the R configuration, they are significantly active compared to the S configuration, biphenyl that has S configuration. So due to this reason, we have provided this compound to our pharmacology collaborators. They are doing the in vivo bioassay on it. So let's see what will happen now. So can I have my next slides? Another thing, we are working in the area of acetylcholinesterase, and we discover compound against Alzheimer because acetylcholine is a neurotransmitter, and acetylcholinesterase stop its function by hydrolytic destructions. And these compounds have several applications, but our target is to discover compound against Alzheimer disease. And so we, we have developed a very nice bioassay in our lab. Can I have my next slide, please?
Yes, sir.
Yeah, we have discovered a very nice bioassay in our lab. So we decided to do some chemistry on Buxus nathalensis. We collected again this plant from South Africa. The traditional healer were using this plant actually to treat memory loss in elderly people. When we screened this plant in our bioassay, and it was showing nice, acetylcholinesterase inhibitory activity with an IC50 value of 28 microgram per ml. I like to emphasize here that any plant that shows the bioactivity, with an IC50 value less than 50 microgram, we are very happy to do chemistry on it because we are 100% sure we will get something, and we always get it. So this is the reason we all, we always work on that area. So we can show you from what we got from Buxus. We end up with steroidal alkaloids, and we found actually one of the compound that contain, we name it as O2 nathafuranamine. Natha, we take it from the plant species nathalescens, and furamine means it contain furane ring, if you will see here. And then furamine is due to the amino group here, and its activity is almost close to the hiprazine. Hiprazine is a prescribed drug, FDA approved drug that they use it in China as well as in USA. So its activity is very close to this. But problem with this compound is that this compound is not in vivo active. So now we are trying to make phosphoester so we can get some bioactive compounds. So coming back to the conclusion of my talk, actually, you can see dimeric carbazole alkaloid we have shown you guys have very potent anti-renin activities. And Buxus alkaloid have potential to develop drugs against Alzheimer, but only thing you have to overcome the solubility problem, so it can work in vivo. So due to this reason, take home is that lesson is that actually you can see dimeric carbazole alkaloid that are present in several herbs. You will see they have some very good anti-renin activity. They can control definitely the blood pressure and other stuffs. So can I have my next slide, please? I like to acknowledge the peoples who have worked in my lab. Actually, this lab is from University of Winnipeg. As I mentioned, actually, I moved nearly 7 months ago to Michigan Tech. So my lab is still under development there. So these are the students who have worked definitely in my lab, and they are involved in different parts of the projects, and they have discovered definitely all these compounds. So can I have my next slide, please? So mostly funding came from Michigan Tech, though as I mentioned, I'm in the process of starting my lab there, and definitely Canadian Institute of Health Research has provided us funding, Research Corporation USA, NSERC, and definitely Pfizer, because Pfizer is collaborating with us for these compounds. So let's see where we are ending up. And these are the funding agency that has provided funding to keep our research running. And thank you very much for listening all this. If you have any question, I'll be happy to answer.
So thank you very much, Dr. Atta. Very nice presentation, very enthusiastic. So the session is open now for questions. If any question.
I have a question.
Yes, please, sir.
Hello.
Yes.
There is a long-standing.
Yes,
Dr.
Dr.
Atta,
Javed
Assalamu
Iqbal.
alaikum.
Walaikum salam.
Hi, sir. How are you doing? Very nice to see you. And you may not be able to see me because I am blocked. my maybe. Ah, here I am. Here I am. Thanks a lot.
Yes. Yeah,
This was a wonderful activity here to join the symposium organized by Dr. Amir and his team. It's always wonderful and always wonderful to listening you and learning from you a lot of things. Alhamdulillah,
Thank you.
you are doing very good over there, and we are trying to be good here. My question regarding your presentation is this: how we can avoid basically the angiotensin converting enzyme ACE inhibitors. I mean, replacing the therapy from the from the chemical toward the some biochemical or from the plant source. I mean, directly how we can use some source of plant. I mean, as you discuss about the Nigerian plants and all that African things. But I mean, how we can replace this, I mean, the ACE inhibitors from the system. And one more thing, that your work related to the Alzheimer is just wonderful. I mean, you are planning and thinking over there, and we are also, because dementia and all that things we are feeling in our system now. A lot of things, elderly people, they have such kind of things in the system here.
Yeah, thank you, Dr. Javedi. Well, first of all, actually, we cannot use the plant. That's a major problem here, because plants contain million of other compounds, and each compound target different bioassay, different biological systems, because body biological system is very complex. So they're using it. People are selling it. This is a major problem in the Europe also these days. They are selling the plants even in developed countries also. So, but problem is that we don't have any proof, or so-called the quality control, to see whether they are active, and this is the reason they have several other problems are arising, even in developed countries. So due to this reason, FDA have banned actually these activities to buy plants, unless some pandemic arises, just like COVID-19. They say, okay, you can do this, you can do this, you can do this, but otherwise they prefer to have the pure chemical entities. So if you have pure molecular level drug that we know that the target the particular, because that has been tested, that is target the particular biological system that we want to do, that's much more effective. That's the answer.
And the other thing, that Alzheimer and the dementia and other kind of stuff in our system, we are feeling that our elderly people are also, I mean, going through this process now. I mean, a lot of people are hospitalized because of this situation. And this is a poor country, and we cannot, I mean, provide all the necessity thing for a medical help for those people. So some shortcut thing, if we can apply and easily, I mean, uh, cope with that situation.
Yeah, I think that's a good question. At least you guys can see some. We have isolated some compound against the acetylcholinesterase from the green pepper, bell pepper. You guys can think about that. And also okra, so-called ladyfinger, right? And they have actually, because I had one time undergraduate student, I asked him to screen different herbs, and we have discovered it. Even in the curry leaf, you can find some compound that has actually, it's exactly hyperazine type compound, and then you will see actually they have some anti-acetylcholinesterase activity. And this
Oh,
is the
that
reason, if you see a little bit go back, the people, our elders, they have a lot of good memory compared to us. Why? Because they were using those spices frequently. We are not. This is major reason we try to avoid the fast food.
Actually, those
Yes,
spices
we
were
definitely do.
added
Yeah.
to our recipes by.
Yes.
Okay.
We don't hear you well, Amr.
आपकी आवाज नहीं आमिर साहब आई.
So first raise hand from Professor Amar Jamil. So maybe
Okay,
he's out.
so I have one
Yes.
long-standing question. Is a wonderful presentation as always. You are also linked with your parent department, uh, HEJ. There is a big question. Yesterday it was raised in our live symposium as well, that they have been isolating these wonderful activities and all that. But why Pakistan is not presenting any of the pharmaceutical drug in the market? Your comment will be very valuable.
There should be actually, there should be some actually very drug design mechanism. HEJ is definitely doing good job there. I believe they have patented some compounds, if I remember correctly, and from the literature, and they have some good activities. I don't know why they are not pursuing. They should pursue for sure. Definitely take it further. And here actually in US we need definitely go through a lot of paperwork for clinical trial and a lot of channels to go for clinical trial. But in Pakistan, I think these hurdles can easily be overcome. So I think they can do this if first of all they have to do. I think problem is that I don't know what's the problem, but I can tell you which I guess problem. They do not do the in vivo assays. That's the problem. And they as soon as they discover compound, they should go after in vivo assays. That's my suggestion.
Okay, thank you. So the next raised hand, Muhammad Muzammil, you can have one quick question. Muhammad Muzammil, raise the hand.
Assalamualaikum, Dr. Tarata. It's amazing to listen you on this seminar. I was very interested to listen you live here in the session because you are working on acetylcholine esterase enzyme. It was amazing, especially by Bux's alkaloid grabbed my interest so far. Next up, did you observe some kind of Bux's alkaloids in Pakistani plants as well or not?
No, unfortunately, it is my wish. I think now to collaborate with definitely in Pakistan. This is the reason I went to Federal Urdu University for vice chancellor, so I can increase that collaboration, but I could not. So I think definitely I will be thinking to collaborate and especially with the agriculture department, in Department of Chemistry at University of Agriculture, Faisalabad. So that will help me to increase definitely to go in this direction to explore.
So that's are you going?
So one last question from our respected guest from
That's all
Poland, please.
I want to add. I want to add.
Ji Mohammad Muzammil Saab, you can add. So I think our respected guest from Poland, can you hear? You muted your mic. Suka, you can ask question. Okay, please ask, please. Last question.
I would like congratulate your gorgeous speech, and I have question. Did you make some molecular modeling study with your compounds or not?
Yes,
yet?
I did it computational chemistry. Definitely, without computational chemistry, it's hard to work these days. So definitely I did it, and they are definitely integral part of our research that allows us to confirm their bioactivity again. And some of the compound, they were not active. We try to do this, but problem is that actually in computational chemistry, that's our problem, because I'm not a computational chemist, and we found actually when we cannot add water molecule to them. So due to this reason, it's a major problem there in silico chemistry. So one of my collaborators, he's developing the some software so we can see the in vivo activities. Yes, we do, definitely. Then we could determine the linkage with the especially a particular enzyme in the enzyme, particular amino acid in the enzyme and active side, or whether it's active, or yes, we can do that. Yes, we do that. Yeah.
Okay.
So I could not present that, but we do. Without computational chemistry, we chemists are nothing.
I only want to say that computational chemistry will be helpful to predict what in vivo, with what compounds do the in vivo studies, because as you know, during the synthesis, you can obtain many compounds, and it's if we want to test every one compound, we waste of time. Then molecular modeling could save our time. Thank you, Professor.
Yes, I agree. I agree.
So thank you very much to Professor Dr. Atharata. So very nice presentation, and all who are working in the plant field area. So they know after this presentation that the crude extract may not be working. So some of the activating compound and some of the inhibiting compound at the same time may be present. That's why pure compounds are needed to be screened, and in the in vivo especially. So thank you very much. So we will move on to the next speaker. So hope you will be with us. Thank you very much.
Yes, thank you. I like to thank organizing committee to giving me opportunity to talk about my science, and thank you. And I think it's a wonderful symposium. I always love to be present there physically, but due to this, due to some commitment, I could not come there. Thank you. And thank you
We miss you,
for
sir.
inviting me.
Thank you, Jazakallah.
Thank you very much. So our next speaker is Professor Dr. Zhang, the Chinese Academy of Sciences. So he is already with us in this meeting. So I have already allowed all the participants to be as a presenter. So he can join and he can start his presentation at his own end. Professor Zhang. So.
Hello, can you hear me?
Okay. Welcome, Professor Zhang. So nice to
okay.
see you.
Well.
Hello.
Sorry,
Professor Zhang, you are mute.
Can you see my slide right now?
So I can see slides and I can hear you even.
Okay.
Also.
Okay. Thank you very much for giving me the opportunity to present my work. So I'm Huawei Zhang from China, and today I would like to introduce my work about the inhibitor AK42 towards the target chloride channel 2. So, but before that, I would like to give some introduction about the chlorine and its application in the drug discovery. So as we know that chlorine is a very new technique, and it wins the Nobel Prize in 2017 because of the resolution evolution for some breakthroughs in the hardware such as the direct electron detector and also some software. So cryo-EM techniques have several techniques. One of the important one would be the SPA single particle analysis. So for this one, it is suitable to investigate the molecular in vitro in the isolated stage. And another one is the cryo-electron tomography. It is suitable for the cells and cell organelles, such as the mitochondria. So in this one, we isolate the organelles from the cell, and then freeze the organelles directly. And then just like a CT, we collect the data at the different angles, and then reconstitute the 3D models of the organelles. Now the last one would be the CLAM, correlative light and electron microscopy. So in this method, people usually combine the light microscope, and also electron microscope. So firstly, we label the targeted proteins. We can see their localization under the fluorescence microscope. And then we freeze the sample and put it in the electron microscope. Using this method, we can have the high resolution structure of the local structure, and also the sample can be investigated under the large spatial space. And this is the workflow for the single particle analysis. So firstly, we prepare the sample. We express and purify the proteins in many different ways, such as the affinity column and size exclusion chromatography. And then we freeze the samples using on the cryo-EM grids, such as the copper grids. And then we image the samples and collect data at large scales. We process the data using several softwares, such as Crosspark and Relance. From this, we obtain the map of the molecules. Then we build the model based on the map, and then for further structural analysis. So right now, cryo-EM has played a very important role in the drug discovery. So in last year, we are invited to submit a review to summarize current applications and prospects for cryo-EM in drug discovery, MMR, very high impact journal. So there are several techniques that cryo-EM can be used in the modern drug discovery. The first one would be the structure-based drug design. So for this method, firstly, we usually got the target protein from for example, the PDB database, or we can even use the predict model using the AlphaFold tool. Based on the models and the structures, we can identify some pocket that the small molecule can bind to the protein. And based on the pocket, we can screen large scale of database and to get the ligand candidate. And then we can perform some analysis, such as the molecular docking, to see how the ligand will recognize and modulate the protein's function. And based on the lead compound, we can further optimize the compounds to further improve its effect. So here is one of the example using cryo-EM to determine the protein target named GPCR, and it also is small molecule ligands to see how they interact with each other. Another one would be the fragment-based drug design, FBDD. So it is pretty similar like the SBDD. However, for this method, usually we got small compounds for one pocket, and lastly, we link the ligand together to control the molecule. And also here is some examples using these methods to regulate the function of proteins such as the BGU. Next one would be the protic. So this is also a new method recently. So for this one, it's essentially a small molecule that have 3 parts. In the middle, it is the linker, and also there are 2 ligands. So one of the ligands will recognize the ubiquitin proteasome system, and another ligand will recognize the target protein. So the two ligands will bind their target together to put the target protein and the ubiquitin enzyme together so that the enzyme can degrade the target protein. So here's several examples using the PROTEC, using structural studies to review how the PROTEC molecules work. And other than this, recently CREM can also be used in the antibody development, especially in the pandemic of the COVID-19. There has been a lot of antibody development and investigate using the CREM to determine how the antibody recognize the target protein, especially for the S proteins. So in this pipeline, during the development of the antibody, you can use the CREM to investigate how the antibody recognize the target protein and what is the fingerprint that can for their interactions. And based on this, we can further modify to engineer the proteins to improve its effect. So, and last part would be the drug repurposing and the drug failure. So as we know that many drugs have very severe side effects, and many drug candidates can be failed during the clinical trials. So here people use the CREM to determine this protein target. It is gamma secretase. So APP is a normal substrate of the enzyme, but people find that some clinical drugs always fail. So people use the CREM to determine its complex structure, and they found that actually the notch, another substrate, can function as a similar way as APP, so that some inhibitors can inhibit both of the interaction with APP and the notch. So that is why some of the drugs that are failed in the clinical trials. And the next part will be the work of CLC2 chloride channel and its specific inhibitor AK42. So my research is actually mainly focused on the mechanism and drug delivery of memory proteins, especially for the ion channels. Ion channels plays very important role in the normal function of our main organelles such as the brain, heart, and the kidney. So because of their importance, so many studies on ion channel has been won the Nobel Prize. For example, in 1963, 1991, 2003, and 2021. So my research is mainly focused on the assembly and the functions of ion channels and to determine their structure and based on the structures to screen and investigate how the drugs can modulate its functions. So using the CREM as a tool, we have several outputs. For example, in 2019, in collaboration with Professor Wachu, Stanford University, we discovered how the chloride channel cytotoxin YA assembly into the hexamers and insert in the membrane channel. And also in last year, we determined the CREM structures of calcium channel panel 3 to review how it's assembly and how it determines the substrate specificity. And also in last year, we have determined the chloride channel CRC2 and in complex with its inhibitor K42. So here's some brief introduction about the CRC2 family. So this family has several members from 1 to 7 and also Ka and Kb. So CRC1 and CRC2 are from the same subgroups. So mutations on CRC2 can result in many diseases. For example, the loss of function mutant can causing early disease, and some mutations such A500V can be related to the loss of function mutant. while other mutants such as 172Q can cause in the gain of function mutant, where the CRC2 have increased activities. So in 2020, a research group from Stanford University, they used the high throughput screening to screen a molecule named FCFA. They found that this molecule can modulate the function of CRC2 channel specifically. So based on the backbone of these chemicals, they did a lot of work to improve the effect of these molecules. And eventually they found that the molecule AK42 can significantly inhibit the function of CRC2 channel. And also it shows a very critical specificity compared with the CRC1. So now the scientific question is that what is the underlying mechanism for the specificity of AK22 towards CRC2 but not CRC1? So to answer this question, we determine the structures of APO form CRC2 and also the AK42 bond form. So we prepare the sample, we collect the data, we did a 2D classification and a 3D reconstruction. We improve the resolution to about 35 to both of the forms. So structure analysis reveals that CRC2 is a homodimer. So here you can see these overall structures. So the protein consists of TMD domain and CTD cytoplasmic domain. So it have 18 helix named A2R. And this shows the local resolution for these maps. You can see that relatively the cytoplasmic domain have a lower resolution. So we mainly focus on the TMD domain, that is the transmembrane domain. So you can see that the model of the CRC2 can fit very well with the maps densities. And also this is the density for the K42 inhibitors. So this model can also fit well with the densities. Within analysis the channel, that is the how the chloride ions can pass through this channel. So there are 3 sets that the chloride ion can pass to this channel named as external, SEXT, set central, SECN, and also set internal, this one. So 3 residues named E25, S162, and Y553, they form a selective filter to control the substrate selectivity for this channel. And also in this region, the channel pool is very small. That is to ensure that the channel can be in the open form and the closed form in the regulatory way. So one of the residue E205 in our structure, it binds to this set. So that means the set train will close the channel. That also means that our structure is in the closed form. So we also compare our structures with other reported channels like CRC1, CRCK, and CRC7. We found that in CRC1, the side chain of the residues E232, that is the equivalent of this residue E25 in CRC2. So it points to other sets but not in the channel. So that is why the CRC1 is in the open state. And also in CRCK, the equivalent residue will be the valine 166. So it is a very short side chain. So that indicates CRCK is also in the open state. And in CRC7, another members in CRC family, so this side chain points to other direction. However, because of the other sets are very narrow in this channel, so it is also in the closed state. So we then analyze how the inhibitor AK42 interact with the CRC2 family, CRC2 channels. And here, the cyan color means the CRC2 channel, and this green color is represents the AK42. You can see that this molecule, this inhibitor, responds to the channel directly. So structure comparison between the Apple form and AK42 band form reveals that upon the binding of AK42, one of the residues M460 have a significantly change of the side chain orientation. But overall, the structure is very similar. So within analysis how what is the residues that is critical for the interactions. So we identify several residues.
So screen shared, stopped sharing. Dr. Zhang, we cannot see your screen. Maybe he's disconnected.
Rena?
Yes, you are back again. Please share screen.
Yeah, because I noticed I was removed. It's fine, it's fine.
You were removed, really? You were removed by someone?
I'm not sure, yeah.
Actually,
So,
all are present in this meeting, so anybody can do, but some mischievous.
Can you see
We are
my...
very glad that you are back.
Can you see my PowerPoint right now?
Yes, you can continue, please.
Yes, sorry. So based on the structure analysis, so there are two important patches that can be in the interactions of AK42 and also the CRC2 channel. So one of the pocket will be the hydrophobic pocket in the bottom. So there's a lot of hydrophobic residues such as F and also L. And another important set would be in the charge interaction with the key 394 residues. It is the charge residues. So we right now we have identified the residues, and then we compare with the residues from CRC2 and also with other members in the CRC family. We found that so for each row we can see that we show the equivalent residues, and that is compared with the CRC2. And the red color means they are different from CRC2. So you can see that. So for each of the member from the CRC family, so there's at least 5 residues that is different from CRC2 channel. So this result reveals the structure bias, how AK42 can specifically inhibit CRC2, but not other CRC2 members. So based on the interaction residues, we further validate our result using the patch clone electrophysiological recordings. So we did a lot of mutations for these key residues, and we found that for most of the residues after mutation, and their inhibiting effect will be significantly reduced. So this result further validates our result that this molecules, these residues are very important in the interacting with the AK42 inhibitor. And so previously, so in the peanut paper published by Stanford research team, they actually have screened a lot of other molecules that is similar as AK42. However, the effect is not as high as AK42. So based on the structures, we also review the behind the reasons why these molecules it's not as effective as AK42. So these molecules either modify the rings in the bottom or the rings on the upper. We can find that. So if you modify the rings in the bottom, so it will introduce the stereo inhibitors that reduce the interaction of AK42 with the CRC2 channels. And also previous study also used molecular docking to investigate how AK42 inhibitor CRC2. So they had a lot of posts. They identified several posts for this inhibitor. And also we compare our experimental posts with molecular docking posts. We found that none of them can fit well with our experimental structures. And also, although they identify several key residues based on the molecular docking results, two very important residues named F306 and F463, they are not identified. However, these two residues are identified in our cryo-EM experimental results. So during the functional analysis, we also found several interesting phenomena. So after we mutate several residues at G203 and F306 and L524, we found that the current pattern changes a lot compared with the wild type. So we further mapped these three residues to our structure, and we found that these residues are very close to the channel. So possibly these residues are important for the channel regulation and directly in the involved in the chloro transportation. So based on our structures analysis and function validations, we propose a model to explain how AK42 inhibits the function of CRC2 channel. So upon the addition of the application of AK42, this molecule, this inhibitor can block the channel directly. And during the binding, one of the residues, M460, will change the conformation of its side chain significantly. And also, we also collect the mutations that is related to the diseases. And we found that those mutations can be grouped into three parts. For example, one of the residues, GH98, it locates in the dermal interface and probably likely will affect the dermal interactions of CRC2 channel. And other residues such as G199A, it locates very close to the channel regions, and it may directly affect the channel properties. And other residues like R68H, it locates in the cytoplasmic region and possibly play a role in the functional regulation of these channels, such as voltage sensing. So here's a short summary of my presentation. So cryo-EM has been the main technique in the structural biology and plays a critical role in modern drug discovery. So our results also reveals structural biases for the AK42 specificity towards CRC2, but not other members. Yeah, so we would like to thank to our collaborator in Southern University of Science and Technology, and also professors in the Chinese Academy of Science. So our cryo data are collected in Southern University and Science and Technology, and our findings are from the NSFC China. Okay, thank you very much.
So thank you very much, Dr. Zhang, for a very nice presentation, very nice
Thank you.
information about the mechanism of the chloride channels. So any question, if anyone want to ask a question, can raise hands. The first question from Javed Iqbal, the first raised hand. Javed Iqbal. So Javed Iqbal can hear me? Okay. If not,
I cannot. Yeah, I cannot hear.
So Javed Iqbal, we cannot hear. You mutated your mic, maybe. Now. Now we can hear you.
Yes, yes. Thank you. Very nice to listen to you, Dr. Zhang. My question is about the CIC channel, how it affect the smooth muscle and the cardiac muscle. and how it can regulate that channel?
This channel can transport the chloride ions in between the extracellular space and also the intracellular regions. So it make a balance between the ion channels. So when the channel is opened, the chloride ions will be transported from the intracellular region to the extracellular regions. So that makes the hyperpolar of the voltage. Yeah.
I mean, this channel is regulating truly with the cardiac muscle. I mean, the arrhythmia are impulses to that.
Sorry, cannot hear you.
Hello.
It's fine right now. Yeah, right now it's fine.
Sometime this cord maybe creating. I want to learn. I mean, the channel which is regulating the arrhythmia, rhythmic impulses to the heart, cardiac muscle, and other smooth muscles.
Yes.
How this CL3 channel regulating? I mean, the link between these things.
So the this channel when it is open, it can transport the chloride ions from the intracellular set to the extracellular set and makes the cell in a hyperdepolarized state.
Okay.
Okay,
Please carry on.
Okay, thank you very much. Okay, the next question from maybe our guest from the Poland. Lukas may be asking question. He raised the hand. So all other participants can mute their mics. All other participants. Okay, Lukas, go ahead, please.
Okay, thank you for the very informative and so impressive presentation. Yesterday, I presented similar scope about the channels, but it was monoamine transporter channels, and we are very interested. First
serotonin,
question.
right? Is it serotonin?
And I would like to ask, it is possible share us your presentation?
Sure. So share.
Could you send to us your?
Of course, after the conference.
After
the conference, because we are very interested in your studies.
Yes.
That are very complex, very actual, because we will see that in literature. It's very, actual problem about chloride channels. Then we're very interested, and we will be very thankful when we can check your most studies and maybe contact in the future.
Yes, sure.
Thank you so much.
Thank you.
So thank you very much, Dr. Zhang. Very informative, very useful. So thanks again. So
thank you very much.
We move to the next speaker now.
Okay.
Thank you. So the Professor Azhar Hussain from Pak Austria Project, Pakistan. So he's online. Professor Dr. Azhar Hussain. So he can. Yes. Okay. Welcome, Professor Dr. Azhar Hussain. So I have little introduction about you. So
thank you for having me, and all the presentation that I have been looking at in the in your schedule, and since morning I have been listening to the learned presenters. My presentation is totally different than what is being discussed in your conference. Thank you for allowing me to discuss what I have. It's basically a perspective, looking at things at a broader level, not at the micro level, not at the molecular level, not at the cell level, but probably the impact is also there on the micro and the cellular level. So I would be talking about the accelerating sustainable development goals progress through research and academia. academia industrial collaboration for sustainable drug development. Next slide, please. I'm working as a professor of practice at Park Austria Fachhochschule, University of Applied Sciences and Technology. And the people who know the capital city of Islamabad in Pakistan, it's almost 90 kilometers from Islamabad. If you go through the motorway, or if you use the old Khanpur Dam road, it's almost 50 kilometers from Islamabad. So it's almost a 1 hour drive from Islamabad. Next slide, please. It's a new project. It's a 3-year-old project, and it is located in a very beautiful vicinity of a city called Haripur. It's basically on the CPAC track, and this is the aerial view of the university. We have a very beautiful small dam. It's a very green area, and with the academic blocks and all the different facilities that we have. Next slide, please. Okay, so we talk about the Sustainable Development Goals. The Sustainable Development Goals, also known as the Global Goals, were adopted by the United Nations in 2015 as a universal call to action to end poverty, protect the planet, and ensure that by 2030 all people enjoy peace and prosperity. These are 17 in numbers, and it is recognized that they are all integrated. I've lost the slide. Yes, they recognize that action in one area will affect outcome in others, and that development must balance the social, economical, and environmental sustainability at all levels. Next slide, please. So the role of academia, research, and higher education is very important in the attainment of the Sustainable Development Goals, and the role of academia and higher education institutions worldwide is of paramount importance in realizing the UN's Sustainable Development Goals through teaching, training, research, knowledge creation, community engagement, and finding innovative solutions to the challenges the world is struggling to deal with it today. Next slide, please. So the role of research in sustainable development is very important, and the research will aid the decision-making process of the policymakers at various cadres of the managerial leadership of the SDGs all around the world where the goals are intended for. The essence of research in enhancing growth and development of any system is very important and need to be focused. Next slide, please. The world's biggest challenge are global and interdisciplinary, requiring insights from multiple disciplines to fully understand and effectively address them. So it is very important to understand the perspectives of different stakeholders and how they work, and especially how they integrate to get to the right outcome, which is very important. Next slide, please. So these are the 17 Sustainable Development Goals. You can see it in the picture. And the transdisciplinary university industry linkage can accelerate progress towards solutions for SDGs. Over here, I'm focusing on 3 SDGs. SDG number 3, which talks about health and well-being. SDG number 4, education, and SDG number 9, innovation and industrial development. Next, please. So we also need to understand if the industry and academia are working together. So one of the outcomes that we would like to see is the number of publications that comes out as a joint authorship between academia and industry. So in 2019, Microsoft published an estimated 1,775 pieces of collaborative academia research globally. In the Netherlands, it was 5.8% of the publications came out between the collaboration of the academia and industry. And between 2015 and 2019, Philips was the top collaborator in the Netherlands, IBM in Switzerland, Bosch in Germany, Hoffmann La Roche in France, and Microsoft in the UK. So there's a lot of money which is available as part of the Research and Development Fund, but usually industry and academia, from where I'm talking about part of the country, the part of the world from where I am sitting in Pakistan, usually this collaboration between academia and industry, joint publication is seen very less. So this is one of the areas, if you really want to work with the industry want to work with the academia, they need to focus on joint publications. Next, please. So the academia industry synergy, if we can develop that, it can lead to combining theoretical insights with practical applications, resource optimization, accelerating research and development, developing the next generation of skilled professionals, sustainable business practices, and also in long-term investment in sustainability can also be seen. Next, please. So for the contribution by global academia and industry linkages, we can look into the four broader areas. Number one, emerging industries we can look at. Number two, importation and transplantation of the industries. Number three, diversification of existing industries into technologically related new ones, and upgrading of existing industries. So these are the four areas where we can focus on. In the emerging industries, we can focus on the forefront science. Okay, we can go to the next one. There's no problem. So this is one of the models. So in the emerging industries, we can focus on the forefront science and engineering research, technological licensing policies, promote, assist entrepreneurial businesses, developing incubation centers, cultivate ties between academia and industry, researchers and local entrepreneurs, creating an industry identity, participate in standard setting, convene conferences, workshops, all these standard things that we are doing and we are looking at. And in the type 2 importation transplantation of industries, we can have education, manpower development, responsive curricula. We cannot have a curricula which is in isolation with the industrial input, and then technical assistance for subcontractors and suppliers. And at type 3, diversification of existing industries into technological related new ones, bridging between disconnected actors, filling structural holes, and creating an industry identity. And type 4, upgrading of existing industries, we can focus on problem solving for industry through contract research, faculty consulting processes, education, manpower development. Global best practices can be scanned and can be used in the local context wherever we are working. Convening foresight exercises, looking into the future, what are the needs, and aligning yourself according to those needs, and convening user supplier forums. Next, please. So academia industry smart synergy model for producing drug innovators. This is one of the models that can be used. There's an industry and there's a university. University basically focuses on professional education and also on research. But though that research should be responsive towards the industry research, we need to have customized programs which are focusing on the need of the industry, and industry also need to focus on the type of collaboration that they can bring in with the universities. And in the end, if we do that, the university do that, they can have a more competent workforce. Industry usually works with the market demand. And for that, fulfilling that market demand, usually they focus on the research, and the research can come from the universities. But for that, they need to find some funding and support. The industry also gives certain certifications to the universities, and the curriculum development is very important, which is responsive to the latest needs of whatever we are doing in the real world. Next, please. So this is the prospective framework for the university industry collaboration. So there has to be a broader policy framework coordinated by the government. If it's the Ministry of Industries which is looking at it, if it is Ministry of your higher education looking at it, it really university industry collaboration should focus on the skill development. Usually in our part of the world, we are more focusing on the theoretical knowledge again, and usually the skilling part is usually not that emphasized at the educational universities. Innovation and technology transfer is very important. And then promotion of entrepreneurship and startup and spin-offs are very important. The generation and production of products and acquisition of all the resources which are required to create these synergies are very important. Next, please. So the collaborative model which needs to work on is focus on the identifying the gaps and developing the partnerships which are required. The collaborative model is network of research and development in collaboration of industry and academia needs to be established, and profit alone slogan needs to be replaced by the profit together, which can lead to the focus on the drug development. And the partnership needs to be developed between the academia, industry, CROs, patient organizations, and all the important stakeholders which we think are important, who are the end users and who are the developers that needs to be connected together. Next, please. So the role of research and academia in promoting SDGs, we need to focus on research and academia, drive innovation in drug development. And the critical role in ensuring the safety and efficacy of the products is very important, whatever it is coming out for the patients or the end consumers, and to promote sustainable drug development practices by conducting clinical studies, by conducting research, testing, evidence practice, using evidence-based practices, considering environmental impact, ethical considerations, and affordability. Next, please. So this is basically academia research support in clinical trials. Academia is good in basic research, and its early development can be done at the university level. And that research can be used by the startups and biotechs, and middle-sized and big pharma industries can actually use that knowledge at all phases, particularly strong from lead optimizing through market access. Next, please. So strength of academic research, academia has an excellence in basic research. Innovation for new therapeutic targets and pathways can be developed. Cutting edge of science in determining specific mechanism of actions can be done. Technical expertise are available which can be collaborated, and scientific network facilitating exchange of knowledge, that what we are doing, what university is doing right now, basically they are facilitating different parts of the world working on same and different things, having different perspectives. They can develop further collaboration. Next, please. So the importance of collaboration between academia and industry cannot be overemphasized. Combining academia knowledge and industry experience can foster innovations. It can lead to better interdisciplinary approaches and practical application of the knowledge that we have. Industry collaboration enhances academic impact. It translates research into real-world solutions and improves patient outcomes. And access to industry and resources and funding can enable research, development, and commercialization, and it can have a sustainable impact. So what are the challenges in collaboration for sustainable drug development? They're different in priorities and timelines. Universities have different priorities and industries have different priorities. Academic focuses on knowledge generation, and industry is driven by profit and market demand, and usually they have more focus on a product development. So industry and academia need to develop a certain synergy to look into this problem. Then they need to have a communication and cultural differences needs to be seen. Can you go back? So building strong, communication and cultural differences needs to be addressed. We navigate diverse organization structures and practices. We see academia has a different culture of working, and industries have a different culture of working. So that needs to be seen when we are working together. So intellectual property rights and commercialization concerns. What are the concern working with industries? Industry usually don't want to publish their work unless it is developed into a product and marketed. Although the academia works differently. So again, this is one of the challenges that is between the academic research and also with the industrial research. Next, please. So building strong partnership for sustainable drug development, we need to establish clear goals, roles, and responsibilities, ensure alignment and accountability throughout the collaborative processes. Effective communication and regular updates are very important while facilitating information exchange, problem solving, and decision making. And mutual trust, respect, and shared values needs to be established, which can foster a collaborative and inclusive work environment. Next. So if this happens, there will be a great impact of academia industrial collaboration on sustainable development goals progress and contributing towards sustainable development goals globally as well as locally by advancing access to affordable and quality healthcare, by promoting environmental sustainability in drug development and supporting economic growth and innovation. Next, please. So as a last slide, collaboration between academia and industry is very crucial. It cannot be overemphasized. There's a call of action for all the stakeholders, means all stakeholders who are involved in research, academia, policy making, whatever it is, need to work together. I don't see in my country a collaboration between Ministry of Industries and Higher Education. They work in silos. So if there has to be some policy where industry and academia need to work together, so there has to be certain policy at a higher level. And of course, if this happens, together we can accelerate toward the sustainable development goal progress. I thank you. That was the last slide. Thank you very much.
So thank you very much for this nice presentation, Professor Azhar. So if any question, Professor
I hope
Ahmad
it
Jamil.
made sense. I hope it made some sense.
Yeah. Okay, so I have a question like yesterday we were also discussing here why there is no like positive collaboration going on in our situation, like lots of efforts or emphasis has been put on by the government, by HEC, for academia industry for the last 20 years, but still we don't see what are the real bottlenecks and what may be the solution to those in our case.
Doctor, number one thing that I see, most of our academics are not practicing. They're not practicing. So if I am a pharmacist, I hardly see, I pursue my career from all my degrees in continuity, and I never worked. I did work in industry, but most of us, we don't have exposure with the university. What I can tell you, what we are doing at my university, it's Parkostia Fachhochschule. Fachhochschule is a German word which means University of Applied Sciences. And usually the concept is that the university is placed within the industrial zone. So where we are placed in Haripur, it is connected with the industrial zone, which is Hattar. So usually that's one of the steps that is being taken in the country. It's one of its own kind. Fachhochschule, one is coming up in Sialkot, one is coming up in Gujarat, another one is coming in KPK. So there are 8 Fachhochschule which are being designed on this concept that the university should be linked and placed within the industrial zone. So that's one of the things that can be done. Secondly, at the university level, we have very strong boundaries. We say that you can only teach at my university if you have a PhD. What we are doing at our university, that we have introduced the concept of professor of practice. Professor of practice is someone who don't have a PhD, but have spent good number of years working in an industry and is eligible to teach at my university. So that's the second thing that we are doing. Thirdly, what we are trying to do is that from the first year of the education of the students, it is mandatory for the students to go for an internship. Usually our university carry out their internships in the final years or after the completion of the degree. So for our university, 500 hours of internship is mandatory to get a degree. So that's one of the things that we can do. And thirdly, when our students go for internship, our faculty also opt for an internship within the university. So if I feel that there's a gap and I want to work with your university or some industry in Faisalabad or some industry at Karachi for 6 weeks, I also go for that internship, sort of internship or refreshing my knowledge, whatever you say. So probably this is what can be done at your university. We are trying to do at our university. And secondly, higher education, Ministry of Higher Education, whatever you call it, and Ministry of Industries need to work together. I hope probably that can help, Dr. Saab.
All right. Thank you very much for elaborative talk and elaborative
Thanks, sir.
reply.
So thank you very much, Professor Azhar. So our next speaker, there is not much information. He has not contacted yet. Dr. Junaid Afzal, if he is with us, he can raise the hand. Otherwise, we will move to next speaker. Dr. Junaid Afzal is with us from the Kashmir University, India. Yes, Dr. Junaid Afzal is with us. So I unmute his mic. Okay, so Assalamu alaikum. Welcome, Dr. Junaid Afzal. So I allow you to become the presenter so you can then share the slides and you can continue with your presentation. So I have only your name and your topic. Your topic is for today's presentation is chemico-biological interaction in the drug discovery. You can give us a little more introduction about you. So it's over to you, Dr. Junaid Afzal. Okay, that's really great. Okay, you can continue. Jazakallah.
Okay, sir. Thank you, sir. So my topic was all about chemical-biological interactions, and this is my personal information. And I am currently working on this project as designing supra-decorative allosteric ligands. And here is a small information about my supervisor, Dr. Masood Ahmad Rizvi, currently working as associate professor in Department of Chemistry, Kashmir University. So here are a few contents about my presentation. The introduction, the chemical-biological interactions in drug development, what are the materials for chemical-biological interactions, perspectives of drug target interactions, what are the methods, and there is a small overview about molecular docking and some case studies. So what actually chemical-biological interactions mean? It refers to recognition of interactions between chemical compounds and the biological targets in the living system. Basically, in the drug development, once we synthesize any compound, we deal these two things. One is drug and another is target molecule. First, we must know what is our target, and accordingly, we should design our drug moiety. So what actually my presentation is all about, these interactions. What are actually these interactions? How forces are interacting and how these drug and target are actually interacting with each other. And here, during the drug development, we deal with biological targets, especially we deal with proteins and enzymes. And it is common that we know that enzymes are soft systems. Soft here, we normally mean that even a small condition, pH change or temperature change, it matters a lot in drug designing. So these are dynamic and tunable via non-covalent and chemical-biological interactions. During the drug development, we mostly focus on non-covalent interactions because if our drug is a good carrier, it must be a good releaser also, because it has first our drug should reach to the target site, and then at that time it should be a good releaser also. So its function should be dynamic, and its interactions should not be permanent. It should be non-covalent. Here I have mentioned some like ion-ion interactions are present, ion-dipole interactions, dipole-dipole, and mostly in protein molecules there are benzene rings normally, and we are normally see there are pi-pi interactions, and hydrogen bonding is normally commonly seen in drug-target interactions. The binding of bioactive molecules to their biological occurs family through non-covalent. What I was about telling you that these interactions are normally found in drug-target relations. So what is the need? One must know why there's a need for drug development. So recently, we have faced this COVID-19 pandemic disease that was challenging all over the world. So these are the preparedness. We should know if tomorrow there will be any big challenging disease like vulnerable any other disease. So we must know how to prepare ourselves, how to create such drugs which will be resist against such viruses. So these are some perspectives. These are some needs. Like to potentiate a chemotherapeutics, our drug should be drug resistant. It should specific, and these are normally what our mentors have already discussed about. It should be bioavailable, and it should have less side effects. There are the preliminary cases for drug development. And these are some properties while we designing the drug. It should be low molecular in weight because it has to travel throughout our body, and it should have a balanced hydrophilicity and hydrophobicity according to our need, and it should be stable under physiological conditions. Physiological here means our drug is not working in outside conditions. It has to function inside our body. The parameters are different, the conditions are different, and it should be non-lethal. And as a drug chemist, we should know it should not be, it should not cause any lethal effect in our body, and it should not accumulate, and its toxicity should be minimum, and its pharmacokinetics, it should be acceptable. It should not damage our body. It should not show any negative response to our body. So here are some materials for what are the materials for chemical-biological interactions. Once we design any drug, how to design any drug? What should be our prerequisites regarding any drug? So first of all, I have mentioned already that here during drug designs, once we design any drug, we have to create or we have to design it accordingly to create some functional sites on our drug moiety. Normally, we design it on the basis of suprafunctionalities. We put some supra designed sites like hydrogen bonding, hydrogen donor sites on our drug moiety. Accordingly, it is working on our drug molecule. So introduction of supra type function is capable of inducing non-covalent interactions. Already mentioned that while designing drug, it has to deal with non-covalent interactions. It should not, the interactions should be in balance. It should not have any permanent interaction with our carrier molecule or with our target molecule, because it has to accordingly function. First, it should be a good releaser, and then it should act on our target molecule. Then it has to again empty the site for the next molecule. So it is interactions should be non-covalent in nature, and more the number of supra sites. Here we are dealing supra, here means supra type of functionality means supra bonding. That means non-covalent interactions. The more sites we have, the more is the chance of interactions, and the more interactions we have, that means our drug moiety definitely will be a potent drug candidate. So here I have mentioned about is that it is directional. Once we design a drug, we know about its target, we know how to deal it with. It should be all directional. It should not be random. So here in this lab, we are currently dealing with these normally these biotargeters like HSA, human serum albumin, or BSA, bovine, and DNA is our target. Catalyst is our target. Acetylcholine ester is our target. And our formula connection should be like this. How bioavailable is this? What is its biocompatibility? And regarding this is regarding with HSA and BSA, because these are carrier proteins, and our drug demand from this carrier protein is always it should be a good binder and it should release kinetics. So what I actually mentioned that if our drug is binding with these proteins betterly, then it should release also. It is not all about to bind, it is about to release also. So it should be a good binder and good releaser also. Next, our second target is DNA, and here we deal with DNA cytotoxicity, the cancer, commonly dreadful disease, and its apoptosis. Here our drug demand should be always, it should bind to it and it should cleavage it. It should limit its what we can say proliferation, or it is irregular division. And normally I have worked on this catalase enzyme, and catalase, it is an antioxidant enzyme, we know, and it releases oxidative stress, and it is working on H2O2, and it breaks down H2O2 into oxygen. So our drug demand is always, because catalase is working against the reactive oxygen species. So our demand is always its efficiency should be increased. Here our demand is catalase activity promoters. We have to design those drugs which increase the catalase activity. So catalase is antioxidant. So those compounds which increase its efficiency, here we deal with this. And we accordingly, once we design a drug, we put those functionalities there which are interacting with this catalase enzyme, and accordingly its efficiency will be increased. And there is one more, acetylcholine esterase. Normally it is higher concentration or lower concentration. It is dealing with neurodegenerative diseases. And here our demand should be accordingly, if there is any lower concentration effect, our demand should be that time it is inhibition. So here are some parameters how these biotargets are shown in our books. So inherent complexity of biological targets. The way we are showing these biotargets like catalases or even DNA it is not as simple as it is shown in books. Actually, book version is this type, the crystal structure is this type, and actual structure is really, it is much more complex in nature and it is much more dynamic in nature. What are actually descriptors of chemical-biology interactions? How can you check what are the actual descriptors? How to, once you design a drug, how can you say that my drug is efficient, my drug is binding or not? So here, first of all, we deal with that whether our drug is binding to our target or not. Is the first perspective. Next, it is strength. How much it is strength it is while it is binding to our target molecule. Next is when once it is binding, is leading to any change in structure or not, and there is thermodynamics. How thermodynamic it is efficient to interact with our target molecule. So these are some methods how to check these. What is our how to check this affinity, how to check this binding. These are some parameters like biophysical methods. We check with we deal with absorbance, fluorescence, CD mechanism, and commonly recently the current era is dealing with in silico methods like molecular docking and molecular simulations. And there are some biological methods for the biological students like Western blotting, electrophoresis, and MTT-IC. And during these methods, what we deal with during absorbance, fluorescence, or CD, we change these, we interact our drug molecule with these our target molecules. And after these methods, UV methods, we check how its intensity is changed. Is it leading to any hyperchromic shift or hypochromic shift, either blue or left shift? These shifts definitely will give you an insight about the interaction of your drug and target molecule. And after performing these analytical methods, we go through enzyme kinetics, and there definitely you will get information about either it is a promoter or inhibitor. And through Michael-Menten equation, we get these results. And these are some applications I have mentioned, whether drug is binding to a target. If there is any change in intensity, if there is change in any spectrum shift, definitely you will get information about your drug target interactions. So this is how actually we deal with UV visible methods. And there is wavelength shift, hypsochromic and hyperchromic, and absorbance shift, bathochromic and hypochromic shifts. These shifts definitely give you an insight that your drug is interacting with your target molecule or not. So here are some shifts of our T30 molecule. What my supervisor already worked on this moiety, that is the organic moiety, and here are some shifts regarding this molecule. So what are these applications? Applications is normally a number of binding centers on our target molecule. First, we go through this. Next point is whether our ligand is binding to a target or not, and what is the strength. Strength of via conjugation. These are actually these analytical methods. And binding with is if it is binding, there are two possibilities always. If your drug is binding with your target molecule, either it is leading to any possible change in structure or not. So this information, these superior methods will give information whether it is leading to any change in structure or not. And through Stern-Volmer conjugate construct, you will get information about this thermodynamic. Is thermodynamically applicable or thermodynamically feasible or not? It is not always that your drug is interacting with your target mode. It will definitely lead to any efficient or any change in efficiency. No, it is not always possible. Sometimes your drug will interact with your target molecule, but there should not be good thermodynamics. So it overall, this sum up overall as a whole, whether it will lead to a good target or whether it will lead to a good drug molecule. So ground or axis interaction is normally due to fluorescent intensities. This is the general information of circular dogma analysis. Normally, this through this CD mechanism, we deal with the secondary structure of our target molecules. Is our secondary structure stable or not? Through these interactions, we can get information about this. So here I have discussed about something like molecular docking studies. In the field of molecular modeling, docking is a method which predicts the preferred, because once you are dealing with drug and target, you don't know what is the actual pose, how actually our drug is interacting with the target molecule. So through these computational methods, we get know exact information, what is the actual pose, how actually our drug is interacting with the target molecule, because there are different sites available in our our target molecule. So how actually our drug is interacting with our target molecule, through computational methods, we can get the best information and use to predict how a protein interacts with small molecules. And definitely this, maybe you might have heard about this. And we deal with this software. Normally Autodoc is known for simple docking. Simple docking here we mean that our drug is interacting with the target molecule, but through simulations and simulations we get exact information. Is this efficient? Is this thermodynamic feasible or not? All those parameters can be analyzed through MD simulation. So if this is, so here I have just taken this video. This is actually how molecular docking is working, how our drug is working with this target moiety, because target moiety is very complex in nature. So it finds a best pose. It finds our drug is finding always a best site to accommodate there. So actually this is working like here in this small video. It will show you how our drug moiety is interacting with this target molecule. And you must know it is all dynamic in nature. It is not as simple as it is shown in. And these are actually these supra interactions which are effective or which are applicable always in these drug target relations or drug target interactions. So accordingly, you can show, there are lots of things in this video. You can show how actually our drug is interacting with the protein moiety. So here is a simple information about this catalase target. Actually, the catalase function is all about to break down hydrogen peroxide into water and oxygen. And we all know that reactive oxygen solutions are toxic to our body. And here our drug demand is always that it is efficiency or its activity should be promoted. So we have interacted our T30 molecule on BSC through these UV absorption methods. We have it is shown that it is showing a good mobility or good efficiency with this catalase enzyme, and it is increasing its efficiency. As we have increased the concentration of T30 molecule, it definitely has led to increase in BSC activity in a dose dependent. It should be a concentration dependent. That is the point what I have mentioned here. So these are some spectrum what I have discussed with this T30 molecule. It leads to change in, and through these methods we calculate its thermodynamics. We calculate is this binding efficient or not. So these are some parameters which actually deal with analytical methods. So what is the conclusion of this? First off, we will get information about the interactions between drug and target proteins, because drug and being a key in drug not only help us to understand the disease mechanism, but to identify therapeutic activity and adverse side. We can get both the informations through this way. As COVID-19 has recently, it has accelerated our scientific world to be active with this current challenges, the timelines of discovery and development of drugs and vaccines. So that is the reason that we are here. So new, effective and less costly. These are some designing parameters. Therefore, in this moiety, we have seen that this T30 molecule which we have designed here. It is shown. It has proved. It has shown an efficiency against this reactive oxygen species, and it has led to enhancement of the enzyme activity of this catalase enzyme. So here, these are some references why I have taken my presentation. Thank you, sir. Thank you. Thanks a lot.
So thanks for the nice presentation. Thank you very much, Dr. Junaid Afzal. So any participant or faculty member can raise the hand and I will allow the mic for the questions.
Anji, meanwhile, I can ask one question. Yes. So, where are you, like, particularly, do you think that we are standing in terms of some commercial avenue towards this drug development using your techniques you are working on?
Yes, sir. Yes, sir. I'm not actually getting you, sir.
So question can be repeated, can please?
Okay, I will repeat. So first of all, I, as a principal organizer of the symposium, we are thankful to you, as Dr. Ali has already said, for joining us today. So we wish that next time you may be joining us physically in some other endeavor, and you are doing very good job. So I was wondering, where do we stand with reference to the commercial point of view, like the drug development process you are working on, how it can lead to the commercialization of the product?
Definitely, sir. Your question is really important for us. And currently, I am a project scholar, and I am actually working on this, how to design actually our drugs. And I don't have much information actually on the basic levels. But what can I say? Look, on the basis of some problems, we can design the drugs. And there are lots of parameters where we have to discuss about it. And it's commercially much more effective. I think so. There are lots of commercial applications while designing a new drug.
All right, thank you very much.
Thank you, sir. Thanks a lot. Thank you, sir.
So thanks once again, Dr. Junaid Afzal, for joining us. So our next speaker is Dr. Aisha Ghaffar, University of Agriculture, Faisalabad. If she is with us, she can raise hand. I will allow her to become the presenter. Dr. Aisha Ghaffar from the University of Agriculture, Faisalabad. So, Dr. Aisha Ghaffar. So, Aisha Ghaffar is with us in this meeting. So, no hand is raised. So, respected sir, can we move to the next speaker? We can again.
Yes, move to the next speaker. So today, due to certain assignments, there are some speakers, they cannot join today. So like Dr. Rahila, also she had some. So we go to the next, Haram Afzal, I think.
Okay. Okay. So next speaker is Dr. Haram Afzal, University of Agriculture, Faisalabad. He can raise hand. Okay, one hand is raised. Thank you.
All right, she can go.
Okay. So I'm making her the presenter. So welcome, Dr. Haram Afzal, University of Agriculture, Faisalabad. Her topic today is about the recent technological advancements in the mRNA vaccine development and its potential applications. So she can a little more introduce herself. I don't have your introduction, and then you can continue to your presentation. So up to you, Dr. Haram Afzal. So you can share the slides then. You can continue.
Hello, Assalamu alaikum. Can you hear me?
Yes, we can hear you.
So this is Haram Afzal, student of Doctor of Pharmacy, 6th year, and I'm not a doctor right now. You were calling me a doctor, so I'm not a doctor right now. I'm a student of 6th semester right now, and my topic is recent technological advancements in RNA, messenger RNA vaccine, and its development. Then okay.
Yes, now
Is
it's
it right?
fine.
Sir?
Okay.
Okay, thank you,
Yes,
sir.
yes.
Thank you. Okay, madam, go ahead.
Okay.
Haram Afzal. Yes.
Okay, so I'll be talking about recent technological advancements in messenger RNA vaccine development and its potential applications. So starting with the introduction, we know that how vaccination process works. We use weakened, killed, or attenuated viruses, and we administer it into the body of a patient, and that virus develops the immunity in the person, and as a result, when the subsequent attack of a virus occurs, so there is already immunity in the body, and it can kill the virus. So that's how our conventional vaccination used to work. So there is a recent process that's been employed, that is messenger RNA vaccination. And in this technique, we don't actually use a virus kill or attenuate it. We actually use a similar protein that is present on the virus, and you use messenger RNA to develop that protein. So when the subsequent attack of virus occurs, our body is already familiar with that kind of protein, and there is no use of the virus in itself. So how it works is it boosts our immune system. The disease causing virus is actually absent. So messenger proteins similar to unique design of viral proteins are administered. In case of specifically coronavirus, there is a spike protein on the virus, and that protein is created with the help of messenger RNA, and it is administered into the person's body. And as a result, the person is familiar with that protein, and when the virus actually enters the body, it recognizes, our immune system recognizes that protein, and they start producing antibodies against it. So in this technique, we can avoid the risks of actually administering the virus into the body. So it is very useful technique. But the current vaccines that we use for this process are Pfizer, BioNTech COVID-19. I myself got the vaccination of Pfizer, and it's 3 doses, the booster dose as well. So it is used to provide protection against COVID-19 virus. The recent vaccines that used were Pfizer and Moderna. There were other as well, Novavax, Sinopharm, but these were not based on messenger RNA technology. The messenger RNA technology was used by Pfizer and Moderna specifically. So these were helpful in combating that global pandemic of coronavirus. It was actually very helpful, but there were still complications that were very minute. I was going across some studies, and I found out that there is a very small ratio, but there is a risk of myocarditis, thrombosis, and myocardial infarction related to messenger RNA vaccines specifically, because the virus, when we give that protein, messenger RNA protein, it can confuse the heart muscles for the viral protein, and the body can produce antibodies against the heart muscles and actually weaken them and cause myocarditis. So these are the complications. Although the drug regulatory authorities have said that it is not, the risk to benefit ratio is much lower, and it's not that risky, because having coronavirus is much more riskier, and myocarditis risks are much greater with coronavirus without the vaccine. But the risk cannot be ignored still. I saw what people had to say about it, and people were not already very confident in getting the vaccination at that time. And still people were saying opinions like, oh, see, we told you guys, and we are the pure blood enjoyers, and those who got vaccination will see what happened with you now. So there was already many speculations and many problems with people getting the vaccine done. And with such news, it is already very, it is becoming a problem for future. So people will not trust the vaccination process. And when if God forbids it happens again, a breakout happens again. So these complications need to be addressed, and there is so much room for improvement and research in this field. The management of complications, we need to identify the risk factors, that what are the environmental factors that are causing the myocarditis and inflammation of heart muscles and these problems. We need to optimize the vaccine design, because the vaccine design that was previously present was attacking the heart muscles. The immune system was attacking its own heart muscles. So there need to be a better design for vaccine that can be very specific about the attack on the virus protein only. So there is another room for improvement in the developing adjuvants with reduced immunogenicity. Adjuvants are added in any medicine or vaccine to improve its availability and to make it better for the patient. So we need to develop better adjuvants that can actually help the vaccine, so it is not acting against the heart muscles, and we need to explore the different delivery methods. The previous vaccines were given by intramuscular pathway. So there's room for research in the delivery methods like nasal methods, spray methods, or intradermal patches can be used. So different approaches to avoid the benefits, the risks that are attached to this, messenger RNA vaccination, and different pre-treatment strategies and medications can be given to the patient, which are not currently employed yet, but research can be made on these topics, that we what are the possible medications that we can give to the patient to avoid these certain risks. The future aspects include it can fill the gap between infectious pandemics, and it can help us with the plentiful supply of effective immunizations, safe and long-lasting immune response in both human and animal models. And messenger RNA technology can also help in treating diseases like cancer. Cancer, we know that we don't have a very effective treatment against cancer. So it can we can produce individualized medicines with individual patients, and our body can produce antibodies to kill the cancerous cells. So it has a lot of future aspects and applications, and room for research and improvement. So that's all about my presentation. If you have any questions, please go ahead.
So thank you very much for your presentation. You are working on the vaccines and the RNA vaccines. We all know that RNA vaccines are producing the immune response within 3 days, I have heard. So that's why maybe you have selected the RNA-based vaccine. So what kind of adjuvants you want to use, and can the efficiency of vaccines be increased, the RNA vaccines, by the use of adjuvant? What kind of adjuvant you are using?
This is my prospective research right now. I will be doing the research in my final year project. So that's all about it right now. Yes.
Okay, okay. It will be good for even our students. Some of my students are producing the hyperimmune sera, and they are using their adjuvant, and they may be contacting you. This is a nice presentation. So thank you very much. So I hope you will
Thanks
be
so
with
much.
us. Thank you. So our last speaker, if she is joining us, Aisha Ghaffar, if she is with us in this meeting, she can contact, she can raise hand actually. Aisha Ghaffar, the last speaker for today's session, for the symposium. Aisha Ghaffar. So I think she may have some responsibility today. She may be busy. She did not join us. So with this last presentation, we conclude the symposium and we conclude the today's session. And we are thankful to all the presenters, and especially our guest from the Poland. We are really thankful to them. And they presented and they participated in all presentations, and they were really cooperative, and we hope our collaboration will continue. And we are also thankful to all the teams, the student organizers and the faculty members who help us in organizing this event. We are also thankful to the hall management. Maybe they will be listening us. They help us in managing the hall and the presentation and the stage. And we are thankful to the Dean, Faculty of Sciences, the respected Honorable Professor Dr. Ikar Ahmed Khan. They provided us this opportunity, and definitely the Chairman, Professor Amar Jamil, helped us in organizing this. So now I hand over this mic for the final comments to the Professor Amar Jamil. So over to you, respected sir. Thank you.
Okay, thank you very much. So I congratulate all the participants and all the speakers that we have a very successful this symposium. Dr. Ali already has thanked all our committees, all of our administration, and especially we are thankful to Endowment Fund for putting in funding for us. And I'm also very thankful to all of the keynote speakers, all oral presenters, and our delegate from Poland. And for today's session, I'm especially thankful to Dr. Ali. He has managed it very well, not only today, but yesterday, the online participation that was also managed by Dr. Ali. And thank you very much, Dr. Ali. You managed it well. And I am also, I appreciate the efforts once again by all the my team members, including the department faculty, department staff, and the SESCOB, a whole of the SESCOB, and all the students who participated it. So today there was a good presentation coming up, like Dr. From the UK. But he, unfortunately, yesterday he had some dental surgery, so it is very difficult. So we will ask him, because it is a political economy with reference to the drug development. It was a important talk. We will ask him some other time, some online seminar with him. So with all these, I think we conclude this session, and I hope you enjoy the weekend. Thank you very much.
So thank you very much. So then we say bye to each other. Hope our collaboration will continue. So thank you very much. Allah Hafiz.
Okay, Allah Hafiz. Goodbye.